Articles by HIV/AIDS researcher Peter Duesberg. Part 1. A Challenge to the AIDS Establishment. By Peter Duesberg. Bio/Technology, November 1987. Acquired immune deficiency syndrome, AIDS, has become a cause célèbre for journals, companies, and scientists, and the deadly AIDS virus has been sold to the public as the cause of AIDS with the confidence and authority that is usually derived from absolute scientific proof. The bases of the virus AIDS hypothesis are that this retrovirus was originally isolated from an immune-deficient patient, that 80–90% of AIDS patients have antibody to the virus, and that there is a reasonable correlation between antiviral antibodies and persons in high-risk groups for the disease. The hypothesis is appealing because it appears to fit a 100-year-old tradition of triumphs in medicine that proved viral and bacterial parasites to be the causes of contagious disease. But this appearance is deceptive. 3 criteria need to be met before a virus can be said to function as a pathogen. It must be biochemically active. It must infect or intoxicate more cells than the host can regenerate or spare. And the host must be genetically and immunologically permissive. Yet almost none of the accumulated data on the human immunodeficiency virus, HIV, demonstrate that the virus satisfies any of these criteria. HIV is latent and inactive, not only in the 1 to 2 million Americans who test positive for antibody to the virus, but also in the 10,000 who annually develop AIDS and the 5,000 who die from it. Yet all other pathogenic viruses are known to be metabolically active when they cause disease. Latency is the mechanism by which parasites typically survive as passengers in asymptomatic hosts. HIV also fails to meet the second criterion since it actively infects fewer than 0.01% of susceptible lymphocytes and since 5% of T cells are regenerated during the 2 days it takes the virus to infect the cell. Moreover, it is truly paradoxical that HIV is said to cause AIDS only after an asymptomatic incubation period of at least 5 years, although antiviral immunity is induced within a few weeks. Ever since Jenner discovered the principle of vaccination, antiviral immunity, the basis of the AIDS test, has been considered the ultimate weapon against rather than an indication for future disease. Consistent with the presence of antiviral antibody, there is very little direct evidence for the presence of the virus in persons with AIDS. There is not one report of a virus titer from an AIDS patient. Indeed, virus can only be isolated from 50% of symptomatic and asymptomatic carriers, and then only by techniques originally designed to activate latent viruses. The methods are to grow millions of cells in culture, away from the immune system and as yet unknown suppressors of the host, until at least one previously latent virus becomes active. This will then be multiplied by adding uninfected cells until a detectable titer is reached. Thus isolating virus from 50% of AIDS patients implies that 50% carried less than one latent virus in several million cells. In fact, viral genomes have only been detected in about 15% of persons who test positive for HIV antibody. In these persons, about 1 proviral genome is found in 100,000 susceptible lymphocytes. In other words, 85% of antibody-positive persons carry either less provirus than this or none at all. Moreover, most of these proviruses are dormant since only 1 in 10,000 to 100,000 susceptible cells express viral RNA in both symptomatic and asymptomatic carriers. In contrast, the titers of other known retroviruses are between 10 to the 4th power to 10 to the 10th power infectious units per ml of serum or tissue when they function as pathogens. The virus AIDS hypothesis also totally fails to explain how the virus depletes T helper cells, and why it takes at least 3 to 5 years to do so. Unlike all other animal viruses, retroviruses need mitosis to initiate infection. Moreover, no HIV gene remains inactive during replication, which takes about 1 to 2 days, as with all other retroviruses. Thus HIV would be expected to kill T cells and cause AIDS when it first infects an organism and not 5 years later when it is biochemically inactive and suppressed by antiviral immunity. The 5-year latency presents proponents of the hypothesis with 2 bizarre options: either old T cells die 5 years after infection, or the offspring must die in the 50th generation, given a 1-month generation time for the average T cell. Nevertheless, killing of T cells within weeks, not years, after infection has been observed in cell culture in apparent agreement with the claim that the virus kills T cells. But this type of killing is fundamentally different from the unconditional cell lysis achieved by true cytocidal viruses. It involves cell fusion mediated via HIV antigens on the surface of infected cells, and is conditional on the cells and virus isolates used. Further, it does not occur in chronically infected human T cell lines that grow indefinitely in culture yet produce more virus than any other system, nor has it ever been observed in blood taken from an AIDS patient. In fact, limited cell killing by fusion is a common feature of retroviruses, none of which have as yet been claimed to cause AIDS. It seems clear from the foregoing that the virus AIDS hypothesis fails to make a case for sufficiency. It offers no explanation for why less than 1% of antibody-positive persons develop AIDS and why the mean latency between infection and disease is 5 years, whereas antiviral immunity is established in a few weeks. A latent period for pathogenicity that exceeds the latent period for immunity is unambiguous evidence for a CO factor or another causative agent altogether. Finally, the hypothesis cannot support a claim that the virus is even necessary for AIDS in view of the fact that it is barely present and consistently latent even in persons with the disease. Since the transmission of AIDS depends on frequent contacts involving the exchange of cells, the case for a viral cause remains open *. End of article. AIDS, non-infectious deficiencies. Acquired by drug consumption and other risk factors. Ph. Duesberg. Department of Molecular and Cell Biology. University of California. Berkeley, California 94720, USA. The first 5 AIDS cases, described in 1981, were all male homosexual drug users with pneumocystis pneumonia and acute cytomegalovirus infections, Gottlieb et al., 1981. Since then, over 100,000 cases of about 25 conventional diseases including predominantly pneumonia, Kaposi's sarcoma, lymphoma, dementia, tuberculosis, candidiasis, and diarrhea have been recorded as AIDS diseases in the U.S., CDC, 1990. Over 90% occurred in 2 major risk groups, CDC, 1990, namely in 20 to 40-year-old intravenous drug users and male homosexuals who also consume drugs, Gottlieb et al., 1981, Lauritsen and Wilson, 1986, CDC, 1987, Darrow et al., 1987, Haverkas and Doherty, 1988, Rappaport, 1988, Adams, 1989, Chasen et al., 1989, Weiss, S., 1989, Friedman-Kien et. 1990. Because acquired immune deficiency is thought to be the common cause of these vastly disparate diseases, they were grouped together as the AIDS syndrome. There are 2 competing hypotheses as to what causes AIDS, the virus AIDS and the risk AIDS hypothesis. The virus AIDS hypothesis, currently held by most medical scientists, in particular virologists, proposes that the retrovirus HIV, human immunodeficiency virus, causes AIDS by killing billions of T cells but only on average 8 to 10 years after infection. Institute of Medicine, Natal Academic Sci., 1986, 1988, Coffin et al., 1986, Gallo and Montagnier, 1988, Blattner et al., 1988, Duesberg, 1989a. HIV was originally discovered by Montagnier and CO workers in 1983 as a latent retrovirus in AIDS patients, Barsinisi et al., 1983. Tracking latent viruses, like HIV, depends largely on technology that was only developed in the 70s and 80s. Prior to that time, a latent virus such as HIV was practically undetectable. It is for this reason that the virus is perceived as new. Gallo and Montagnier, 1988, Blattner et al., 1988. On the basis of the virus AIDS hypothesis, about 25 conventional diseases are now defined as AIDS if they occur in the presence of antibody to HIV. Institute of Medicine, Nat. Academic Sci. 1986, 1988, CDC, 1987, 1990, Duesberg, 1989a. However, except for antibody to HIV or rare elements of the latent virus, there is no known distinction between conventional Kaposi's sarcoma, Friedmann-Kien et al., 1990, emphoma, dementia, tuberculosis, candidiasis, diarrhea, etc. and their AIDS counterparts. The fact that antibody to HIV is found in the US predominantly in AIDS risk groups, and that about 1.5% of the 1 to 1.5 million antibody-positive Americans develop AIDS diseases annually, Duesberg, 1989a, are cited as the primary arguments for the virus AIDS hypothesis. Coffin et al., 1986, Institute of Medicine, Nat. Academic Sci. 1986, 1988, Gallo and Montagnier, 1988, Blattner et al., 1988. Yet a correlation, in particular one with antibody to a virus, is not an unambiguous argument for viral causation of a disease, Duesberg, 1989a. Moreover, the relevance of this correlation to the etiology of AIDS is questionable because it is 100% by definition, Institute of Medicine, Nat. Academic Sci. 1986, 1988, Duesberg, 1989a, rather than by natural coincidence between the diseases and HIV. In addition, a viral etiology of AIDS is claimed based on anecdotal cases of transmission, Institute of Medicine, Nat. Academic Sci. 1986, 1988, Gallo and Montagnier, 1988, Blattner et al., 1988. It is claimed that because of infection with HIV, antibody-positive hemophiliacs or other antibody-positive recipients of transfusions have developed diseases otherwise typical of their various conditions, although these diseases appear on average only 8 to 10 years after antiviral immunity and only in 1–3% of them per year, Duesberg, 1989a. Likewise, it is claimed that HIV transmission from mother to child is the reason some antibody-positive babies have developed typical pediatric diseases on the average 2 years after perinatal infection, Duesberg, 1989a, but not the diseases typical of the major AIDS risk groups such as Kaposi's sarcoma, Duesberg, 1988, Birol et al., 1990, CDC, 1990. Yet there is not a single controlled epidemiological study to confirm the postulated viral etiology of AIDS transmission, comparing for example the incidence of ALDS-like diseases in two groups of matched hemophiliacs that differ only in antibody to HIV, Duesberg, 1989a. Moreover, the virus AIDS hypothesis is not compatible with orthodox viral pathology. It is paradoxical that a. Unlike all other pathogenic viruses, HIV hardly infects any cells when it is claimed to be pathogenic, Duesberg, 1987, 1988, I989a. During AIDS, 1 in 500 lymphocytes contain a latent HIV provirus and only 1 in 10,000 to 100,000 contain an active provirus, just as in millions of asymptomatic carriers, Duesberg, 1987, 1989a, 1989b, Schnittmann et al., 1989. Therefore, it is difficult to explain the loss of billions of T cells said to be the hallmark of AIDS, Institute of Medicine, Nat. Academic Sci. 1986, 1988, CDC, 1987. Like the rest of us, even viruses need to do something to get something done. b. In view of the claims that in vivo HIV kills T cells directly, Baltimore and Feinberg, 1989, Ho et al., 1989, over 99% of the few T cells that ever become infected survive infection to become reservoirs of latent HIV, Schnittmann et al., 1989. c. In view of recent claims that viremia is necessary for AIDS, Baltimore and Feinberg, 1989, Coombs et al., 1989, Ho et al., 1989, the number of infected cells remains low in all AIDS cases, Duesberg, 1990. D. AIDS diseases by definition, Institute of Medicine, Nat Academic Sci., 1986, Coffin et al., 1986, only occur after the onset of antiviral immunity, a positive AIDS test, Duesberg, 1987, 1988, 1989a. All other viruses cause diseases primarily before antiviral immunity when they are biochemically most active, Evans, 1989. E. AIDS diseases only occur after long, unpredictable latent periods, averaging 8 to 10 years with a minimum of 2 years in adults, Duesberg, 1989a, while all other viruses including pathogenic retroviruses cause primary disease within weeks after infection. Such long latent periods are particularly paradoxical if one argues that antiviral immunity is essential for pathogenicity, Duesberg, 1989a. F. The same virus would take an average of 2 years to cause diseases in children and an average of 8 to 10 years in adults, Institute of Medicine, Nat Academic Sci., 1988, Duesberg, 1989a. G. The same virus is proposed to cause neoplastic diseases such as Kaposi's sarcomas and AIDS lymphomas as well as necrogenic diseases such as T-cell death or AIDS dementia, Duesberg, 1989a. This, in particular, is implausible, since neither Kaposi's cells nor neurons are even infected by HIV, Duesberg, 1989a. Indeed, neurons cannot be infected by HIV because retroviruses depend on mitosis to initiate infection, Rubin and Temin, 1958, and neurons don't divide. H. Immune deficiency should cause neoplasms such as Kaposi's sarcoma and lymphoma, Duesberg, 1989a. T-cell deficient humans or animals such as nude mice have no more tumors than immune-competent counterparts, Kinlan, 1982, Sharkey and Falk, 1984. I. The most common AIDS disease in the U.S. is pneumocystis pneumonia, while the most common AIDS diseases in Africa are slim disease, diarrhea, and fever, although pneumocystis carinii is ubiquitous in all humans, including Africans, Duesberg, 1989a. J. AIDS diseases would occur in the U.S. to over 90% in males, but would be equally distributed between the sexes in Africa if they were caused by a sexually transmitted virus introduced into each country about 1020 years ago, Duesberg, 1989a. K. Among all risk groups in the U.S., HIV would cause Kaposi's sarcoma almost exclusively in homosexuals, Duesberg, 1988, 1989a, Birol et al., 1990. It is also incompatible with viral etiology that Kaposi's sarcomas in American homosexuals with HIV are indistinguishable from those without, Friedman-Kien et al., 1990. The virus AIDS hypothesis is unproven, because, 1. HIV as the hypothetical cause of AIDS fails to meet many classical criteria of viral pathology, 2. HIV has not been shown to have any unconventional properties to resolve the above paradoxa, Duesberg, 1989a, and 3. There are no controlled epidemiological studies showing a role for HIV in AIDS. The view that HIV is not sufficient or even not proven to be necessary for AIDS, Duesberg, 1987, 1988, 1989a, have been voiced by a number of scientists including, Bialy, 1988, Eigen, 1989, Evans, 1989, Duesberg, 1989c, Gilbert, Hall, 1988, Liversidge, 1989, Griffin, 1989, Haas, 1989, Holub, 1988, Ruth Bernstein, 1990, Rubin, 1988 AB, Schwartz, 1989, Sonnabend, 1989, Stewart, 1989, Weiss, R., 1989, and at least one non-scientist, Fosse's sister Denise, Fosse, 1989, Duesberg, 1989d. Moreover, Friedman-Kien et al., 1990, and Birol et al., 1990, have now called HIV into question as a cause of Kaposi's sarcoma, which used to be the hallmark of AIDS in the early 80s, Birol et al., 1990, for the same reasons I have cited above and previously, Duesberg, 1989a. The risk AIDS hypothesis suggests that AIDS is caused primarily by non-infectious agents. These include psychoactive drugs, overmedication with antibiotics, Rappaport, 1988, Rubin, 1988a, Adams, 1989, Sonnabend, 1989, and above all AZT, a chain terminator of DNA synthesis administered to treat HIV infection since 1987, Duesberg, 1989a, Farber, 1989, Lauritsen, 1989. Consumption of psychoactive drugs is often associated with traditional causes for immune deficiency such as protein malnutrition and parasitic infections, Seligman et al., 1984, and AZT is directly immunosuppressive because it is designed to kill lymphocytes, Duesberg, 1989a, Farber, 1989, Lauritsen, 1989. This hypothesis is compatible with views stated by Ruth Bernstein, 1990, Rubin, 1988ab, Sonnabend, 1989, and Stewart, 1989. The risk hypothesis explains a. Why AIDS is limited to risk groups rather than random in the population, as would be expected from an infectious agent. B. Why natural vaccination against HIV, a positive AIDS test, does not protect against AIDS. C. The long and unpredictable latent periods between HIV infection and AIDS, averaging 8 to 10 years in adults, as the individual reaches a pathogenic threshold of AIDS risks. The shorter latent periods in children averaging about 2 years would reflect prenatal and postnatal risk factors, as 90% of babies with AIDS are either born to mothers who are drug addicts or prostitutes or both, or are hemophiliacs, Duesberg, 1988, CDC, L990. D. The enormous diversity and risk group specificity of AIDS diseases in terms of the diversity of risk factors. For example, the incidence of Kaposi's sarcoma exclusively in homosexuals correlates and declines directly with their group-specific use of nitrite inhalants, Lauritsen and Wilson, 1986, Haverkas and Doherty, 1988, Rappaport, 1988, Duesberg, 1989a, Birol et al., 1990, regardless of the presence of HIV, Friedman-Kien et al., 1990. The nearly complete difference between the major AIDS diseases in the US and Africa could be explained as a consequence of drug consumption in the US and of malnutrition and conventional parasitic infections in Africa. The paradox as to why HIV, unless an innocent bystander, can afford to infect actively 1 in 10,000 and latently 1 in 500 susceptible lymphocytes even in those dying from AIDS, Duesberg, 1989a, Coombs et al., 1989, Ho et al., 1989, Schnittmann LA/. 1989. F. The recent emergence of AIDS diseases in the U.S. as a function of the enormous increase during the last 10 to 20 years in the consumption of psychoactive drugs, Nisi reports, 1978-1988, Rappaport, 1988, Adams, 1989. For instance, cocaine consumption alone has increased fivefold during the last 10 years in the U.S., Nisi reports, 1978-1988. Indeed, about a third of the American AIDS patients are confirmed intravenous drug users, CDC, 1990. In addition, many, perhaps most, of the male homosexuals with AIDS appear to have used various psychoactive drugs, Gottlieb et al., 1981, Lauritsen and Wilson, 1986, Darrow et al., 1987, Haverkas and Doherty, 1988, Rappaport, 1988, CDC, 1990, Adams, 1989, Friedman-Kien et al., 1990. Together, these two risk groups represent 90% of the U.S. AIDS cases, CDC, 1990. Moreover, since 1987, 20,000 antibody-positive symptomatic and asymptomatic persons in the U.S., Marks, 1989, and 50,000 worldwide, Deer, 1989, have been treated with the DNA chain terminator AZT, which is directly immunosuppressive, Duesberg, 1989A. Thus, the use of drugs appears to be a common denominator of most AIDS cases in the U.S. The preferential occurrence in the U.S. of HIV in AIDS risk groups can also be reconciled with the risk hypothesis. Since HIV is not widespread in the U.S. and also very hard to transmit due to its chronic latency, Duesberg, 1989A, only those who have intimate contacts with many others are likely to be infected, as for example promiscuous homosexuals, drug addicts sharing needles or practicing prostitution, and hemophiliacs. Thus HIV would serve as an outstanding, but not an absolute, Friedman-Ken et al., 1990, surrogate marker for AIDS risks, Darrow et al., 1987, Duesberg, 1989a, Weiss, S., 1989. It is concluded that in the US and probably in Europe, AIDS is a collection of non-infectious deficiencies of which about 90% are acquired by drug consumption associated with malnutrition and parasitic infections and other specific risks, such as chronic transfusions for treatment of hemophilia. The 10% of AIDS cases occurring outside the risk groups in the U.S. CDC, 1990, are consequences of the clinical definition of AIDS, namely conventional diseases occurring in persons accidentally infected by HIV. The African AIDS epidemic would also appear to be the product of the all-embracing AIDS definition, namely a widespread heterosexual distribution of the newly detectable, dormant HIV together with old African diseases like slim disease, fever, and diarrhea. For example, about 1020% of the 30 million Zairens carry latent HIV in Africa, but only 335 developed AIDS in the period from 1985 to 1988, Duesberg, 1989a. Like nearly all retroviruses in wild animals, endemic human retroviruses are probably transmitted perinatally, Duesberg, 1989a. This would more convincingly explain the heterosexual distribution in Africa of HIV, and hence diseases now termed AIDS because of HIV, than theories about peculiar sexual practices. G. Klein, 1988, Institute of Medicine, Nat. Academic Sci., 1988. End of article. AIDS epidemiology, inconsistencies with human immunodeficiency virus and with infectious disease. Peter H. Duesberg. Department of Molecular and Cell Biology, 229 Stanley Hall University of California, Berkeley, California, 94720. Contributed by Peter H. Duesberg, October 11, 1990. Abstract: the newly defined syndrome AIDS includes 25 unrelated parasitic, neoplastic, and non-infectious indicator diseases. Based on epidemiological correlations, the syndrome is thought to be due to a new, sexually or parenterally transmitted retrovirus termed human immunodeficiency virus, HIV. The following epidemiological data conflict with this hypothesis. I. Non-correlations exist between HIV and AIDS. For example, the AIDS risks of infected subjects vary tenfold with their gender or country. Abnormal health risks that are never controlled as independent AIDS causes by AIDS statistics, such as drug addiction and hemophilia, correlate directly with an abnormal incidence of AIDS diseases. Above all, the AIDS diseases occur in all risk groups in the absence of HIV. 2. American AIDS is incompatible with infectious disease, because it is almost exclusively restricted to males, 91%, because if it occurs, then only on average 10 years after transfusion of HIV, because specific AIDS diseases are not transmissible among different risk groups, and because unlike a new infectious disease, AIDS has not spread exponentially since the AIDS test was established and AIDS received its current definition in 1987. 3. Epidemiological evidence indicates that HIV is a long-established, perinatally transmitted retrovirus. HIV acts as a marker for American AIDS risks, because it is rare and not transmissible by horizontal contacts other than frequent transfusions, intravenous drugs, and repeated or promiscuous sex. It is concluded that American AIDS is not infectious, and suggested that unidentified, mostly non-infectious pathogens cause AIDS. Epidemiology is like a bikini. What is revealed is interesting. What is concealed is crucial. AIDS is a newly defined syndrome of 25-fold parasitic neoplastic, and non-infectious diseases, including in the United States 53% pneumonia, 19% wasting disease, 13% candidiasis, 11% Kaposi sarcoma, 6% dementia, 3% lymphoma, and 2% tuberculosis. 1. These unrelated diseases are grouped together because they are all thought to be indicators of an acquired immunodeficiency. 2. In America, AIDS is almost completely restricted, 91%, to males. 1. About 90% of all AIDS patients are 20 to 40-year-olds, 30% are intravenous drugs users and their children, 60% are male homosexuals and some heterosexuals who frequently use oral psychoactive drugs, 3 to 7, and 7% are hemophiliacs and other recipients of transfusions. 1. As of 1982, the Centers for Disease Control, CDC, considered AIDS infectious because it appeared to be transmitted among intravenous drug users and homosexuals by sexual contact or by contaminated blood. 8. Among infectious agents, cytomegalovirus and various bacteria were proposed as causes of AIDS. 6. 8,10. In 1983, Montagnier and co-workers, 11, suggested lymphadenopathy-associated virus, now termed human immunodeficiency virus, HIV, and Gallo et al., 12, human T-cell leukemia virus, HTLV, as causes of AIDS. However, psychoactive drugs, like aphrodisiac nitrite inhalants, poppers, were also proposed as causes for Kaposi sarcoma and pneumonia in homosexuals, 3 to 7, 9. In April 1984, the Secretary of Health and Human Services announced that HIV was the cause of AIDS, and an antibody test for HIV, termed the AIDS test, was registered as a patent by Gallo and collaborators, 13 to 15. This happened before even one American study on HIV had been published, 13. According to this view, HIV is a lymphotropic retrovirus that is sexually transmitted, 16 to 20. On average 10 to 11 years after infection and appearance of neutralizing antibodies, HIV is postulated to cause immunodeficiency by killing billions of T cells, 16 to 21. Only then, indicator diseases are said to develop from which patients die on average within 1 year, 21 to 26. Thus HIV became the first virus for which a positive antibody test is interpreted as an indicator for primary diseases that have yet to come. Antibodies against conventional viruses typically signal protection against disease, and those against some persistent viruses also signal a small risk of secondary disease upon virus reactivation. 27, 28. Although no retrovirus has ever been shown to be pathogenic in humans, 29, HIV is thought to be 50 to 100% fatal, more than any other human virus, 16 to 21. The novelty of AIDS is postulated to reflect the novelty of HIV. The large variety of indicator diseases are postulated to reflect underlying immunodeficiency, and the almost exclusive concentration of AIDS in 20 to 40-year-olds, 1, is postulated to reflect sexual or parenteral transmission of HIV, 16 to 20. This virus-AIDS hypothesis was accepted by most medical scientists, in particular virologists, by 1986, 16 to 18, 30. Accordingly, the virus was named HIV by an international committee of retrovirologists, 30, and became the only basis for the definition of AIDS, regardless of the presence of other causes of immunodeficiency. In the presence of laboratory evidence for HIV, any disease listed indicates a diagnosis of AIDS. 2. AIDS is now diagnosed whenever antibody to HIV is detectable along with any of the 25 indicator diseases, even if no immunodeficiency or opportunistic infections are detected, as in cases of Kaposi sarcoma, lymphoma, dementia, and wasting syndrome. 2,18, 23 to 26:31. Moreover, infection in the absence of any clinical symptoms is now termed, and often treated as, HIV disease, 18. However, all efforts directed by the virus ALDS hypothesis for over $2 billion annually, have failed to contain or cure AIDS, 32-33. Since 1987 I have challenged the virus ALDS hypothesis because HIV is latent and is present in only 1 out of 500 T cells during AIDS, because retroviruses typically do not kill cells, and because AIDS appears on average 10 years after the virus has been neutralized by antibodies, 729-34. In response to this challenge, it was agreed that the hypothesis cannot be defended in terms of orthodox virology, based on known genetic and biochemical properties of HIV, 35-47. However, it was claimed that epidemiological evidence supports the virus ALDS hypothesis, 35-36, 38-41, 44-47, and that Gallo, 48, Montagnier, editorial comment in Ref 7, p. 5, and possibly others, editorial footnote in Ref 34, p. 755, would prove it. Here this epidemiological evidence is called into question. Non-correlations between AIDS and HIV. AIDS risks of HIV-infected persons differ 10 to 65-fold depending on their country. If AIDS is caused by HIV, the ratio of infected to diseased carriers should be similar in different countries. However, in the United States about 10%, or 100,000, 1, of 1 million HIV positives, 16,18, 49, 50, have developed AIDS since 1985, but in Uganda only 0.8%, or 8,000, of 1 million, 51, and in Zaire only 0.15%, 4,636, 52, of 3 million HIV positives, 34. Although AIDS surveillance by some African countries has been questioned, surveillance by Uganda is reported as highly successful, providing the highest number of cases in Africa, 51. Since the HIV epidemics of both the United States and Africa are said to be new and to have an African origin, 17, 20, 36, 45, but the AIDS risks of infected Americans are 10 to 65-fold higher than those of Africans. Country-specific pathogens are necessary for AIDS. Moreover, if AIDS equaled opportunistic infections resulting from immunodeficiency, more, instead of less, AIDS per HIV carrier would be expected in Africa than in the United States. AIDS risks among HIV-infected Americans differ about 10-fold based on gender. Since 1985 the U.S. Army has tested 1.15x 106 male and female 17 to 19-year-old recruits for antibodies to HIV. Every year 0.03% of the males and females in this age group were found to be HIV positive, 53. Although HIV has been distributed equally between the sexes among 17 to 24-year-old Americans for the last 5 years, about 10 times more AIDS cases have occurred in males, 53. 95% of these were either intravenous drug users or homosexuals, 53. Abnormal health risks correlate directly with the incidence of AIDS diseases. Since 97% of the American AIDS patients come from behavioral or clinical health risk groups, and since the incidence of AIDS indicator diseases in risk-matched, HIV-free control groups is never reported, 1. It may be argued that pathogens associated with abnormal lifestyles are causing AIDS, 3-7, 32, 34, 54, particularly because the diseases are risk-specific, see below. In response to this, it has been pointed out that AIDS occurs outside the major risk groups in Americans and Africans. However, to date only 3,900, or about 3%, of all American AIDS cases have come from groups without health risk, 1. These represent 25 conventional diseases that occurred in a country of 250 million inhabitants over the last 9 years, 1. To determine whether these diseases are due to HIV, their incidence must be compared with that in the normal, HIV-free population. However, this has not been done, because these diseases are only reportable and recorded by the CDC as AIDS if HIV is present. 1. Thus there is no controlled epidemiological evidence that HIV is pathogenic in the normal United States population. The same is true for Africa. The cumulative incidence of AIDS from 1986 to 1989 in Uganda was only 8,000, 0.8%, out of 1 million HIV positives, 51, or about 0.2% annually. Most AIDS statistics are cumulative and thus always increasing. Since 90% of these cases are common African diseases like Lyme disease, fever, diarrhea, and tuberculosis, 34, 51, and their incidence in HIV-free controls is not reported, it is uncertain whether HIV is pathogenic in Africa. Further, it has been argued based on anecdotal cases that AIDS did not exist prior to HIV in clinical health risk groups such as I. Hemophiliacs, 2. Other recipients of transfusions, 3. Children of drug-addicted mothers, and 4. Drug addicts, 1,18, 35, 36, 38, 44, 45, 47. However, competing causes for AIDS diseases have not been excluded in any of these cases. I. Transfusions of blood and factor VIII and intrinsic deficiencies associated with hemophilia, acting over the long time periods said to be latent periods of HIV, have all been identified as pathogenic factors for AIDS-like diseases in hemophiliacs, 55 to 61. To determine whether HIV or clinical deficiencies and their treatment cause AIDS in hemophiliacs, either controlled epidemiological studies comparing matched hemophiliacs, with and without HIV, or epidemiological evidence that the mortality of hemophiliacs is increased by HIV would be necessary. Surprisingly, in view of the many claims that HIV causes AIDS in hemophiliacs, there is not one controlled study that has found HIV to be a health risk. There is also no report from any country that the mortality rate of hemophiliacs has increased due to the infection of hemophiliacs with HIV, 59. In fact, it appears to be lower than ever, 56, 59. About 75% of the 20,000 severe hemophiliacs in the United States are reported to be HIV positive at least since 1980 to 1982, owing to the administration of blood or factor VLLL, 16,18, 59, 61, 62. Because the administration of plasma fractions prepared from large numbers of donors started in 1965, many hemophiliacs may have been HIV positive for greater than 10 years now, 56, 59. Since 50 to 100% of HIV-infected persons are postulated to develop AIDS after an average latent period of 10 years, 18, 21, 34, at least half of the 15,000 HIV-positive hemophiliacs should have died from AIDS. Yet 2% of the 15,000 HIV-positive hemophiliacs in the United States have died with a diagnosis of AIDS in 1988 and in 1989. 1. The low annual incidence of AIDS-like diseases among hemophiliacs in the United States is likely to reflect hemophilia-related morbidity and mortality under a new name. Indeed, among American hemophiliacs infected for an average of at least 10 years with HIV, elapsed time as a hemophiliac stands out as the critical determinant for AIDS diseases. The median age of hemophiliac AIDS patients is 34 years, or 14 years higher, 59, than that of their asymptomatic peers, which is 20 to 22 years, 16, 59. 2. The thesis that HIV transfusions cause AIDS in other patients is also entirely uncontrolled, 36. Indeed, a controlled study might be difficult because 50% of American patients, other than hemophiliacs, die within 1 year, 58, and 60% within 3 years, 63, after a transfusion, long before the average 10 years that HIV is said to require for pathogenicity have elapsed. The pathogenic conditions that necessitated the transfusions are obviously deadlier than the hypothetical pathogen HIV. 3. About 95% of the children with AIDS in the United States were subject to pathogenic conditions other than the putative pathogen HIV, either drug addiction of the mother or deficiencies of their own that required blood transfusions. 1. The remainder probably reflects normal morbidity of infants born to healthy mothers who are perinatally, see below, or iatrogenically infected by HIV. 4. A rare controlled study showed that among 297 asymptomatic, HIV-positive intravenous drug users, the AIDS risk over 16 months was 3 times higher in those who persisted than in those who stopped injecting drugs, 95. Since the incidence of AIDS diseases in non-risk groups appears normal, and since the abnormal incidence of AIDS diseases in risk groups correlates directly with their abnormal health risks, there is no epidemiological evidence that HIV is pathogenic. Although longitudinal studies of selected risk groups claim some even without published data or references, 64, that HIV positives have more AIDS, none of these studies have controlled the negatives for all health risks and selection biases, 18, 47, 62, 65. AIDS indicator diseases occur without HIV in all risk groups. Tsukas et al., 61, observed severe immunodeficiency in 6 of 14 HIV-free and 9 of 15 HIV-positive hemophiliacs. Ludlam et al., 60, described a group of 15 hemophiliacs who had acquired immunodeficiency before they were infected by HIV. Others observed HIV in only 7 of 19, 55, or 10 of 19, 66, hemophiliacs who had very similar immunodeficiencies. However, direct correlations were observed between the degree of immunodeficiency and the amount of clotting factor consumed, in both HIV negatives and HIV positives, 60, 66. A prospective study from Canada identified immunodeficiency in 33 of 161 HIV-free homosexual men. 9 of these became subsequently infected by HIV, 65. Further, a recent study identified initially 6 and now 12 HIV-free Kaposi sarcoma cases in male American homosexuals, some of which occurred in the absence of immunodeficiency, 67, 68. Others recorded the occurrence of Kaposi sarcoma in AIDS risk groups long before AIDS, 57. And recently, CDC workers have postulated a Kaposi agent other than HIV, because of the almost exclusive occurrence of Kaposi sarcoma in homosexuals among AIDS risk groups, 69. Moreover, there is no trace of HIV even in the Kaposi sarcomas of patients in which antibody to HIV is confirmed, 34. Thus HIV is necessary neither for immunodeficiency nor for Kaposi sarcoma in homosexuals, although their association is perceived as the hallmark of AIDS, 68, 69. Among intravenous drug users in New York representing a spectrum of HIV-related diseases, HIV was not observed in 26 of 50 pneumonia deaths, 15 of 22 endocarditis deaths, and 5 of 16 tuberculosis deaths, 70. Likewise, no HIV was observed in 24 of 54 prisoners with tuberculosis in New York State, of whom 47 were street drug users, 71. Similar neurological deficiencies were recently observed in 12 HIV-infected and 16 uninfected infants from drug-addicted mothers, 72. In a group of 21 heroin addicts, of whom only 2 were infected by HIV, the ratio of helper to suppressor T cells was found to decline within 13 years from a normal of 2 to 1, 73, which is typical of AIDS, 16. Since all AIDS indicator diseases occur in AIDS risk groups in the absence of HIV, HIV is not a necessary cause for these diseases, except for their diagnosis as AIDS. Current AIDS statistics probably include additional HIV-free cases because HIV was confirmed up to 1989 in only about 73% of all American AIDS cases, and in only 39% of the AIDS cases from New York and 61% from California, 74. Moreover, statistics are biased in favor of HIV-positive cases because AIDS is reportable whereas most HIV-free indicator diseases are not, 57. Inconsistencies between AIDS and infectious disease. AIDS diseases that are not male-specific show a tenfold preference for males. The distribution of conventional sexually transmitted diseases is almost even between the sexes, 75. By contrast, American AIDS occurs almost exclusively, 91%, in males, although none of the 25 AIDS diseases is male-specific. 1. However, African AIDS appears largely compatible with infectious diseases that strike without preference for sex risk, and age groups, 17, 18, 51, and that hardly overlap with American AIDS, see below. Latent periods of 10 years are not compatible with infectious disease. Viruses, retroviruses, and HIV are immunogenic and biochemically most active within weeks or months after infection, 27, 28, 34, 76, 77. There is no precedent for an infectious agent that causes primary diseases on average only years after it is neutralized by antibodies, 27, 28, 38, 39. Yet HIV is claimed to cause AIDS on average 10 years after transfusion in adults and only after 2 years in children, 18, 34, 62. The diversity of these latent periods is inconsistent with one infectious agent, and their magnitude is characteristic for diseases caused by chronic exposure to toxic substances. Specific AIDS diseases are not transmissible among different risk groups. If AIDS diseases are caused directly by HIV or are due to HIV-induced immunodeficiencies in available parasites, all infected subjects should have the same risk of developing those AIDS diseases that are not associated with group or region-specific parasites. However, 53% of American AIDS patients have pneumocystis pneumonia and 13% have candidiasis, 1, whereas 90% of the African AIDS patients have slim disease, fever, diarrhea, and tuberculosis but not pneumonia and candidiasis, 34, 51, although pneumocystis carinii and candida are ubiquitous in humans. Including Africans, 34, 78, 79. In addition, the AIDS diseases of American children are different from those of adults, namely, 50% pneumonia, 16% wasting disease, 12% dementia, and 20% bacterial infections, which are exclusively diagnosed in children. 1. Further, in the United States, Kaposi sarcoma occurs 20 times more often in homosexuals than in hemophiliacs and intravenous drug users, 69, often in the absence of immunodeficiency, 23 to 26, 67. Thus, specific AIDS diseases are not transmissible among different risk groups, suggesting that distinct, non-transmissible pathogens must be primary causes. Unlike new infectious diseases, AIDS does not spread exponentially. AIDS is said to be a new sexually transmitted disease, 17, 18, 36, 45. The epidemiological hallmark of a new transmissible disease is that it spreads exponentially until it has saturated a susceptible pool of the population, a process described by Farr's law, 80. Therefore, AIDS would be expected to increase in the sexually active population at an exponential rate, provided there is at least some promiscuity. Yet since the AIDS test has been established and AIDS was given its current definition in 1987, 2, AIDS has spread very slowly, claiming among greater than 100 million sexually active Americans only 20,000 to 30,000 cases annually. 1. Epidemiological evidence that HIV is not pathogenic. HIV is epidemiologically not new. Since 1985, when HIV infection became detectable with the AIDS test, the number of infected Americans has remained constant at about 1 million, or 0.4% of the population, 16, 18, 49, 50. Likewise, the percentage of HIV-positive male and female U.S. Army recruits has remained constant at 0.03% for 5 years, although 70% of 17 to 9-year-olds are sexually active and about 50% are promiscuous, 53, 62. The strikingly constant incidence of HIV indicates that it is epidemiologically not new in the United States and thus not a plausible cause for the new epidemic AIDS. HIV depends on perinatal transmission for survival and is thus not likely to be fatally pathogenic. Due to the absence of HIV in the semen of 24 of 25 HIV-positive men, with one provirus detected per 106 cells, 81, and due to the chronic latency and presence of HIV provirus in only 1 of 500 susceptible lymphocytes, 34, 82. HIV depends on an average of about 500 sexual contacts to be transmitted, 83, 84. Perhaps even more contacts are necessary, because only about 15% of the wives of hemophiliacs are HIV positive, 20, although about 75% of severe hemophiliacs in the United States have been positive for 8 to 10 years. Therefore it is unlikely that HIV could survive by sexual transmission. Further, it is unlikely to be preferentially transmitted by homosexual males, since about 10% of both males and females frequently practice anal intercourse, 62. Based on animal and human models, retroviruses depend almost exclusively on perinatal transmission for survival. They are very difficult to transmit horizontally by immune-competent animals and humans, because they are chronically suppressed, first by maternal antibody and then by the baby's own, 76, 77, and possibly also by cellular suppressors, 34. Even retroviruses with sarcomogenic or leukemogenic oncogenes have never spread horizontally in breeding colonies, 29, 85. Therefore, specific strains of mice, chickens, or humans from geographically distinct regions are often marked for generations by distinct strains of perinatally transmitted latent retroviruses, 85, 86. For example, HTLV is endemic in certain islands of Japan and marks specific ethnic groups among mixed populations in the Caribbean, 86. Wild animals, 29, 85, 86, or humans, 42, 43, 86, with an acute retrovirus infection are virtually never observed. Acute retrovirus infections result from experimental infection or horizontal infections among mass-bred animals, typically prior to immune competence with virus strains not covered by maternal antibodies, 76, 77, 85. Since perinatal transmission of HIV is at least 50% efficient, 18, 20, 34, 62, and sexual transmission is 0.2% efficient, it appears that HIV, like other retroviruses, depends on perinatal transmission for survival. Therefore, it cannot be fatally pathogenic in most infections within 2 to 10 years, as postulated by the virus-Ald's hypothesis. This provides the only plausible explanation for the random distribution of HIV in even as few as 0.03% of 17 to 9-year-old healthy Americans, 53, and in about 10% of Africans of all ages, 31, 34, 49, 51. This explains why no more than 2,456 AIDS cases have been recorded among about 75 million Americans under the age of 19 in the last 9 years, 1, although at least 0.03%, or 25,000, can be estimated to be perinatally infected, 53. It appears that 90% of perinatally infected Americans are asymptomatic for at least 19 years. Antibody to HIV is a marker for American AIDS risks. American AIDS risk groups and patients are marked by antibodies not only to HIV but also to many other viruses and microbes, such as cytomegalovirus, hepatitis virus, herpes simplex virus HTLV, parvovirus, Epstein-Barr virus, genital papillomavirus, treponema, Neisseria amoebae, candida, and mycoplasma, 1, 5, 6, 10, 12, 54, 57, 67, 75, 78, 87, 88. Among these, antibodies to HIV and HTLV are perhaps the most specific markers because their prevalence in AIDS patients is 73%, 74, and 25%, 87, respectively, but only 0.03%, 53, and 0.025%, 86, in the general United States population. Because AIDS patients carry antibodies to many more viruses and microbes, in particular, rare ones such as HTLV, than the general population, it is arbitrary to delineate HIV as an etiologic agent of AIDS by the presence or titer of antibody alone. In addition, this hypothesis is inconsistent with the typical sequence of events in which antibodies follow rather than precede viral pathogenicity, 27, 38, 39, incompatible with HIV-free indicator diseases, and implausible in the absence of HIV activity, 3482. As tens of thousands of positive tests for antibody and hundreds of negative tests for free virus have shown, 34, HIV remains typically dormant in T-cell reservoirs even during AIDS, 82. The simultaneous occurrence of HIV viremia and antiviral antibodies was reported in some AIDS patients in 1989, but this observation has not been replicated by others, 42, 43. More and more of the AIDS-associated parasites are now named as AIDS cofactors of HIV, most recently HTLV and Mycoplasma, 45, 88. A consistent alternative explanation for the high prevalence of antibody to HIV, and other microbes, in AIDS risk groups and AIDS patients proposes that HIV is a marker for American AIDS risks, 34. The probability of becoming HIV antibody positive correlates directly with the frequency of injecting unsterile drugs, 34, 62, 70, 89 to 91, with the frequency of transfusions, 59 to 61, and with promiscuity, 62, 65, 89, 92. However, in America, only promiscuity aided by aphrodisiac and psychoactive drugs, practiced mostly by 20 to 40-year-old male homosexuals and some heterosexuals, seems to correlate with AIDS diseases, 3 to 7, 62, 67. HIV would thus be a marker for these drugs and also for the frequent infections by conventional venereal diseases such as gonorrhea and syphilis, 5, 6, which are not part of the AIDS definition, 2, and for the corresponding therapeutic and prophylactic medications. In fact, HIV was named as a marker for homosexual promiscuity, 92, and recently for an unknown sexually transmitted agent that is presumed to cause Kaposi sarcoma in male homosexuals, 45, 67 to 69. However, not all HIV antibody positives above those expected from perinatal transmission, e.g. 0.03% in the United States, must reflect promiscuity and parenteral infection. Instead, perinatally infected persons may develop antibodies only with age, as latent proviruses become activated by transient immunosuppression or other stimuli. This predicts that the percentage of antibody positives among provirus positives increases with age. The lower incidence of antibody to HIV in 1 to 14-year-old Zairean children, 1 to 2%, compared with adults, 4 to 10%. 31, is a case in point. The incidence of antibody to HTLV also increases with age in countries where HTLV is endemic, although HTLV is just as difficult to transmit sexually as HIV, 86. An alternative hypothesis. Numerous correlations have linked American AIDS with the consumption of drugs. The CDC reports that 30% are intravenous drug users, L, but does not report that another 50 to 60% have used oral psychoactive drugs, 3 to 7, and medical drugs, above all the DNA chain terminator 3-azido-3-deoxythymidine, AZT, 7, 34. AZT is currently prescribed to 125,000 sick and healthy HIV-positive persons worldwide, including about 80,000 Americans, based on annual sales of $284 million and a wholesale price of $2,200 for a year of AZT at 500 mg/day. Burroughs Wellcome Annual Report 1990 End. Office of Public Affairs, Personal Communication. Therefore it is proposed that either drug consumption, frequently associated with malnutrition, by recently established behavioral groups or conventional clinical deficiencies and their treatments are necessary and sufficient to cause indicator diseases of AIDS. This hypothesis resolves the many paradoxes of the virus-Ald's hypothesis. American AIDS is new because of the recent dramatic increase in the consumption of psychoactive and medical drugs, 4, 7, 70, 91. For instance, cocaine-related hospital emergencies increased five-fold from 1984 to 1988, 93. 2. American AIDS is prevalent in 20 to 40-year-old men, although not one AIDS disease is male-specific and this age group is the least likely to develop any diseases. The reason is that men of this age group consume 80% of hard psychoactive drugs, 94. 3. The vastly different AIDS diseases are caused by different pathogens, pathogenic conditions, and their treatments. This also explains AIDS diseases that do not depend on immunodeficiency and occur without it, including Kaposi sarcoma, lymphoma, dementia, and wasting disease. 1, 2, 23-26, 34, 67. 4. African AIDS would be old diseases caused by malnutrition and parasitic infections under a new name, the reason why it is equally distributed between the sexes. 16, 51. V. The long and unpredictable latent periods between infection by HIV and specific AIDS diseases are the product of functionally unrelated events, the pathogenic events necessary to reach an individual's threshold for AIDS diseases, and infection by the marker HIV. End of article. Can alternative hypothesis survive in this era of megaprojects? By Peter H. Duesberg. The Scientist, July 8, 1991. Scientific megaprojects costing taxpayers billions of dollars are, in some instances, the only means of reaching achievable goals. But if a megaproject becomes an undertaking that prejudicially focuses on a flawed theory promoted by a special interest group, the project is counterproductive. The multi-billion-dollar wars on AIDS and cancer are a case in point. AIDS research is now based on the hypothesis that the disease is caused by human immunodeficiency virus, HIV. The war on cancer is based largely on the theory that cancer is caused by either activation of cellular oncogenes or the inactivation of anti-oncogenes. Both wars are waged by selfing groups on behalf of hypotheses that neatly fit their limited expertise. Among the casualties are scientific minorities whose alternative hypotheses threaten the professional and commercial interests of these groups. When a new theory deviates from that held by the majority, it is labeled controversial rather than original, and the controversial label is tantamount to a death sentence, manifested by non-invitations to meetings, non-citations in the literature, non-nominations for awards, and non-funding of research grants. Science journals participate in this process of excommunication, favoring papers that confirm the prevailing dogma. As a result, the underlying hypotheses of the AIDS and cancer programs have been perpetuated long after they failed to produce clinical benefits. Meanwhile, the rise and fall of 3 dissenting AIDS investigators illustrate how valuable research has been squelched. Luc Montagnier became a global leader in AIDS research when HIV, which he discovered, was accepted as the cause of AIDS by Robert Gallo in the United States and Robin Weiss in the United Kingdom. But when he concluded in 1990 that HIV would not be a sufficient cause of AIDS, he was thrown out of his own temple. No paper on his current AIDS hypothesis has yet appeared in the leading journals. Michigan State physiologist Robert Root Bernstein, a MacArthur Genius Award recipient, has concluded that AIDS diseases have occurred in the past and are the result of conventional clinical health risks, such as hemophilia, and of newly developed behavioral risks, including drug consumption and sexual promiscuity. On requesting support for his AIDS studies from NIH and NSF, he was advised that he was unqualified and should not bother to submit an application. Although his research has since been supported via industry collaboration, he is without federal grant support since JUNE1. After discovering retroviral oncogenes and the common genetic map of all retroviruses in the 1970s and 1980s, I became the darling of the emerging hypotheses that cellular oncogenes also cause cancer and that human retroviruses without oncogenes also cause cancer and many other diseases. In view of this, I received an NIH Outstanding Investigator grant in 1985. But 5 years later, after criticizing the cellular oncogene and the virus AIDS hypotheses, another NIH-selected committee terminated my grant. Although my grant did not propose even one experiment on HIV, the review panel included 4 HIV experts. My appeal, which pointed out the professional and commercial conflicts of interest of a number of the reviewers of my application, was disregarded by NIH. What can be done to save investigator-initiated research grants from extinction by theoretically and commercially fixed mega-projects? Among other things, scientists should have to reveal all income derived from their expertise when publishing papers, when peer-reviewing grant applications, even when giving informal talks. Asterisk. End of article. The role of drugs in the origin of AIDS. Peter H. Duesberg. Department of Molecular and Cell Biology, 229 Stanley Hall, University of California, Berkeley, California, 94720 USA. Received November 28, 1991, accepted December 10, 1991. Summary: it is proposed that the new American and European AIDS epidemics are caused by recreational and anti-HIV drugs rather than by human immunodeficiency virus, HIV. Chronologically, the AIDS epidemic in the 1980s followed a massive escalation in the consumption of recreational drugs that started in the 1960s and 70s. Epidemiologically, both epidemics derive about 80% of their victims from the same groups of 20 to 44-year-olds, of which 90% are males. In America, 32% of these are intravenous drug users and an unknown percentage are prescribed the cytotoxic DNA chain terminator AZT, as inhibitor of HIV. Direct evidence indicates that these drugs are necessary for HIV positives and sufficient for HIV negatives to develop AIDS diseases. The drug AIDS hypothesis predicts correctly that AIDS is new in the US, because the drug epidemic is new, while the HIV epidemic is old, fixed at a constant 1 million Americans since 1985. 2. Despite an increase in venereal diseases, AIDS remains restricted to long-term drug users and small groups with clinical deficiencies. 3. Over 72% of AIDS occurs in 20 to 44-year-old males, because they make up over 80% of hard psychoactive drug use. 4. Distinct AIDS diseases correlate with the use of distinct drugs, e.g. Kaposi's sarcoma with nitrite inhalants, tuberculosis with intravenous drugs, and leukopenia, anemia, and nausea with AZT. AIDS diseases are only acquired after long-term drug consumption, rather than after single contacts as the virus hypothesis predicts. The drug hypothesis can be tested epidemiologically and experimentally in animals. It predicts that most AIDS can be prevented by stopping the consumption of drugs, and provides a rational basis for therapy. Was ist das Schwersaint von allem? Was dir das leicht Estatuant? Mit den Augen ziehen. Was vor den Augen dir liegt? What is the most difficult of all? That which seems easiest to you. To see with your eyes. What is in front of your eyes? Zenian, Goethe. Introduction. AIDS is a new syndrome of 25 previously known diseases, 19,62,63, in American 63% are microbial diseases such as pneumonia, candidiasis, tuberculosis, cytomegalovirus, and herpes virus disease, 18,19, that result from immunodeficiency due to a severe depletion of T cells, 62, 63. The remaining 37% of AIDS diseases are dementia, wasting disease, Kaposi's sarcoma, and lymphoma, which are not caused by, and not consistently associated with immunodeficiency and microbes, 18, 33, 36, 115. In the US, 32% of AIDS patients are intravenous drug users, 18, 86, about 60% are male homosexuals, 18, and most of the remainder have severe clinical or congenital deficiencies, including hemophilia, 1833, 100. Over 80% of the American AIDS patients are 20 to 44-year-olds, of which about 90% are males, 18. Different AIDS risk groups have different AIDS diseases. For example, homosexuals have 20 times more Kaposi's sarcoma than other AIDS patients, 7. Intravenous drug users have a proclivity for tuberculosis, 12,114, crack, cocaine, smokers exhibit pneumonia, 41, and users of the cytotoxic DNA chain terminator AZT, prescribed to inhibit human immunodeficiency virus, HIV, develop anemia, leukopenia, and nausea, 94, 99, 113. Currently, most medical scientists believe that AIDS is caused by HIV, 62, 63. This hypothesis assumes, 1. that HIV is new and therefore AIDS is new in all countries with HIV. 9. 2. That AIDS is acquired by sexual and parenteral transmission of HIV. 62, 63, and 3. That AIDS occurs in the presence of antibodies to HIV, a positive AIDS virus. 6, 33, 48. However, each of these assumptions has been invalidated. 1. HIV has long been established in the US, fixed to an extremely constant reservoir of about 1 million carriers, ever since 1985 when it became possible to detect antibody against it with the AIDS test 33, 122. This indicates that the HIV epidemic is old in America, because new microbes spread exponentially in a susceptible population. 46. Thus HIV is not a plausible cause for a new epidemic. 2. HIV is naturally transmitted from mother to child at an efficiency of about 50%. 33. The real efficiency may be higher than serological tests indicate because some latent proviruses only become activated with advanced age. 33, 95. By contrast, sexual transmission of HIV is highly inefficient, depending on an average of about 1,000 sexual contacts. 33, 71 because there is no HIV in semen from 1 out 25 antibody-positive men. 121. It follows that HIV depends on perinatal transmission for its survival. 33. Since retroviruses 37 and other viruses 42, 80 that survive from perinatal transmission cannot be fatally pathogenic, HIV is probably harmless. 3. Within weeks after horizontal infection HIV activity is rapidly ineffective to less than 1 in 10,000 T cells. 36. This immunity does not protect against AIDS, eliminating a role for HIV in AIDS. Indeed, there are numerous inconsistencies between AIDS and infectious disease. 1. There is not even one confirmed case of a healthcare worker who contracted AIDS from a patient, although there were over 160,000 AIDS patients in the US in the last 10 years. 1836, and there is no antiviral vaccine or drug. Likewise, not a single scientist has contracted AIDS from the AIDS virus or from other microbes from AIDS patients, which are propagated in hundreds of research laboratories and companies. 33, 36. 2. All new infectious diseases spread exponentially in susceptible populations. 46. However, despite widespread alarm, AIDS has since 1987 claimed only about 30,000 or 0.03% per year from a reservoir of over 100 million susceptible, sexually active Americans, although conventional venereal diseases are increasing in the US. 5. And there is no anti-HIV vaccine and no anti-HIV drug. 3. The distribution of all infectious venereal diseases is almost even between the sexes. 65. By contrast, 90% of American AIDS is restricted to males since 1981. 18. 4. Almost all, 94%, of the Americans who develop AIDS have been subject to abnormal health risks. 18. These risks include either long-term consumption of recreational, psychoactive, and aphrodisiac drugs and anti-HIV drugs like the cytosidyl DNA chain terminator AZT, see below, or congenital or acquired deficiencies like hemophilia. 18, 33. This indicates that specific health risks are necessary for AIDS. 5. The observations that distinct AIDS risk groups have distinct AIDS diseases, e.g. homosexuals having 20 times more Kaposi's sarcoma than HIV carriers from other risk groups. 7. Intravenous drug users having a proclivity for tuberculosis. 12, 114, crack, cocaine, smokers for pneumonia. 41, and AZT users for leukopenia, anemia, lymphoma, and nausea. 50, 94, 99, 113, 130 are also hard to reconcile with a common infectious cause. 6. All AIDS diseases occur in all AIDS risk groups in the absence of HIV. 33. Ironically, the monthly HIV/AIDS surveillance reports of the Centers for Disease Control, CDC, never survey HIV in AIDS patients. 18. In view of these inconsistencies between AIDS and infectious disease and the total lack of a common, active microbe in AIDS, several investigators have concluded that AIDS may not be infectious. 3, 17, 33, 34, 56, 59, 61, 70, 77, 92, 96, 100. Here the hypothesis is investigated that American and European AIDS diseases, above their normal background, are the result of the long-term consumption of recreational and anti-HIV drugs. Chronological coincidences between the AIDS and the drug epidemics. The appearance of AIDS in America in 1981-62, 63, coincided with a massive escalation in the consumption of psychoactive drugs 16, 44, 57, 73, 87, 89, 100, and 11. The Bureau of Justice Statistics reports that the number of drug arrests in the U.S. has increased from about 450,000 in 1980 to 1.4 million in 1989-16, 100, and 11. About 500 kilograms of cocaine were confiscated by the Drug Enforcement Administration in 1980, about 9,000 kilograms in 1983, 80,000 kilograms in 1989, and 100,000 in 1990-16, 44, 100, and 31. The agency estimates that at most 20% of the cocaine smuggled into the U.S. is confiscated for. Cocaine-related hospital emergencies increased five-fold from 1984 to 1988-89. The number of dosage units of domestic stimulants, such as amphetamines, confiscated increased from 2 million in 1981 to 97 million in 1989-44. Further, the recreational use of psychoactive and aphrodisiac nitrite inhalants began in the 1960s and reached epidemic proportions in the mid-1970s, a few years before AIDS appeared. 88. The National Institute on Drug Abuse reports that in 1979 to 80 over 5 million people used nitrite inhalants in the U.S. at least once a week. 88. A total of 250 million doses per year. 128. In 1976 the sales of nitrite inhalants in one American city alone amounted to $50 million annually. 88. At $5 per 12 ml dose. 106. Since 1987 the cytocidal DNA chain terminator AZT is prescribed as an anti-HIV drug. 67, 100, and 30. Currently about 80,000 HIV antibody-positive Americans and 120,000 worldwide, with and without AIDS, take this drug. 33. Epidemiological overlaps among drug and AIDS-related health statistics. Drugs and AIDS appear to claim their victims from the same risk groups. For instance, the CDC reports that the annual mortality of 25-44-year-old American males increased from 0.21% in 1983 to 0.23% in 1987, corresponding to about 10,000 deaths among about 50 million in this group. 15. Since the annual AIDS deaths had also reached 10,000 by 1987, HIV was assumed to be the cause. 18, 19, 62. However, mortality in 25-44-year-old males from septicemia, considered an indicator of intravenous drug use, rose almost fourfold from 0.46 per 100,000 in 1980 to 1.65 in 1987, and direct mortality from drug use doubled. 15, 85, suggesting that drugs played a significant role in the increased mortality of this group. 15. Moreover, deaths from AIDS diseases and non-AIDS pneumonia and septicemia per 1,000 intravenous drug users in New York increased at exactly the same rates, from 3.6 in 1984 to 14.7 and 13.6 respectively in 1987. 110. In view of this, the CDC acknowledges, we cannot discern, however, to what extent the upward trend in death rates for drug abuse reflects trends in illicit drug use independent of the HIV epidemic. 15. Further, maternal drug consumption was blamed by some 119 and HIV infection by others. 18, 63 for a new epidemic of physiological and neurological deficiencies, including mental retardation, in American children. Another striking coincidence is that over 72% of all American AIDS patients 18 and over 80% of all Americans who consume hard psychoactive drugs 51, 87 or get arrested for possession of drugs 16 are 20-44-year-old males. Thus there is substantial epidemiological overlap between the two epidemics. 73 reported as the twin epidemics of substance use and HIV by the National AIDS Commission. 86. Drug use in AIDS risk groups. Intravenous drug users generate a third of all AIDS patients. Currently 32% of the American AIDS patients come from groups that use intravenous drugs such as heroin, cocaine, and others 1886. This group includes about 75% of the heterosexual AIDS cases, 71% of the females with AIDS, and over 10% of the male homosexuals and hemophiliacs with AIDS 1886. In addition, about 50% of American children with AIDS were born to mothers who are confirmed intravenous drug users and another 20% to mothers who had sex with intravenous drug users and are thus likely users themselves 1886. Likewise, over 30% of European AIDS patients are intravenous drug users 26. Homosexuals who use oral drugs generate about 60% of American AIDS patients. Approximately 60% of the American AIDS patients are 20-44-year-old male homosexuals 18. The following evidence indicates that they come from groups who use oral psychoactive and aphrodisiac drugs. A survey of 3,916 self-identified American homosexual men, the largest of its kind, reports in 1990 that 83% had used one, and about 60% two or more drugs with sex during the previous 6 months 91. These drugs include nitrite and ethyl chloride inhalants, cocaine, amphetamines, methaqualone, lysergic acid, phenylcyclidine, and more too. 10, 27, 56, 70, 91, 96, 97, 100, 101, 112, 120, 126. A study of a group of 359 homosexual men from San Francisco reported in 1987 that 84% had used cocaine, 82% alkyl nitrites, 64% amphetamines, 51% quaaludes, 41% barbiturates, 20% injected drugs and 13% shared needles 27. About 74% had past or current infection by gonorrhea, 73% by hepatitis B virus, 67% by HIV, 30% by amoebae and 20% by syphilis 27. This group had been randomly selected from a list of homosexuals who had volunteered to be investigated for hepatitis B virus infection and to donate antisera to hepatitis B virus between 1978 and 1980. It is the same group for which the 50% progression rate from HIV to AIDS was calculated to be about 10 years 72, 83, and reported to be relevant for the population as a whole 83. Nitrite inhalants and possibly other drugs are preferred by male homosexuals as aphrodisiacs because they facilitate anal intercourse 81, 88, 91, 100, and 1. For example, an early CDC study that included 420 homosexual men found nitrite use far more frequent among homosexuals than among heterosexuals and correlated directly with the number of different homosexual partners 17. Surveys studying the use of nitrite inhalants in San Francisco found that among homosexual men 58% were users in 1984 and 27% in 1991 compared to less than 1% among heterosexuals and lesbians of the same age group 102. An unknown percentage of AIDS patients comes from groups of AZT users. About 80,000 Americans and 120,000 persons worldwide, with and without AIDS, currently take the cytosidal DNA chain terminator AZT33 and an unknown number take other DNA chain terminators like DDI and DDC113. AZT has been prescribed since 1987 to symptomatic 43, 63, 67, 99, and since 1990 to asymptomatic carriers of HIV including babies and hemophiliacs 38, 123, in an effort to inhibit HIV DNA synthesis 130. Thus an unknown, but possibly a high percentage of the 30,000 Americans that currently develop AIDS per year 18 have used AZT prior to or after the onset of AIDS. For instance, 249 out of 462 HIV-positive, AIDS-free homosexual men from Los Angeles, included in the above survey 91, are on AZT or DDI 84. Drug use necessary in HIV positives and sufficient in HIV negatives for AIDS diseases. To distinguish between HIV and drugs as causes of AIDS, it is necessary to identify either HIV carriers that develop AIDS only when they use drugs or to identify HIV-free drug users that develop AIDS indicator diseases. Drug use necessary for AIDS in presumed or confirmed carriers of HIV. 1. Epidemiological correlations indicate that nitrites are necessary for Kaposi's sarcoma. A 27 to 58-fold higher consumption of nitrites 102 correlates with a 20-fold higher incidence of Kaposi's sarcoma in male homosexuals compared to all other AIDS patients of the same age group. 7. B. Among male homosexuals, those with Kaposi's sarcoma have used nitrite inhalants twice as often as those with other AIDS diseases. 56. C. During the last 6 to 8 years the use of nitrite inhalants among male homosexuals decreased, e.g. from 58% in 1984 to 27% in 1991 in San Francisco. 102. In parallel, the incidence of Kaposi's sarcoma among American AIDS patients decreased from a high of 35% in 1983-20 to a low of 10% in 1990-18. In fact, nitrites may be sufficient causes for these diseases, because there was no evidence of HIV infection in any of these studies. 2. Specific correlations also indicate that nitrites are necessary for AIDS. The first 5 cases diagnosed as AIDS in 1981, before HIV was known, were male homosexuals who had all consumed nitrite inhalants and presented with pneumocystis pneumonia and cytomegalovirus infection. 53. Early CDC data indicate that in 1981 and 1982, 75% of male homosexuals with AIDS had used oral drugs at least once a week and 97% occasionally. 57, and that every one of 20 Kaposi's sarcoma patients had used nitrites. 77. The National Institute on Drug Abuse reports correlations from 69% 68 to virally 100% 56, 88 between nitrite inhalants and Kaposi's sarcoma and pneumonia. Again, drugs may have been sufficient to cause these diseases, because HIV was not diagnosed. 3. The incidence of AIDS diseases among 297 HIV-positive, asymptomatic intravenous drug users over 16 months was 3 times higher in those who persisted than in those who stopped injecting drugs. 124. 4. The T-cell count of 65 HIV-infected drug users from New York dropped over 9 months in proportion with drug injection, on average 35%, compared to controls who had stopped. 28. 5. A placebo-controlled study investigating AZT as AIDS prophylaxis in HIV-positive, AIDS-free 25-45-year-old male homosexuals and intravenous drug users indicates that AZT induces various diseases, including some in the AIDS definition. 123. During 1 year of taking 500 mg AZT per day, a group of 453 developed 11 AIDS cases, and a group of 457 taking 1,500 mg AZT per day developed 14 cases. The placebo group of 428 developed 33 cases. However, the price for the presumed savings of 22 and 19 AIDS cases with AZT was high, because 19 more cases of anemia, neutropenia, and severe nausea appeared in the 500 mg AZT group and 72 more such cases appeared in the 1,500 mg AZT group than in the placebo group. This indicates cytocidal effects of AZT on hematopoiesis and on the intestines. Although these AZT-specific diseases were not diagnosed as AIDS, neutropenia generates immunodeficiency and thus AIDS. A loss of T-cells was not reported in this study. This is surprising in view of previous reports describing T-cell and general bone marrow toxicity of AZT 32, 99, 100, and 30. Moreover, 10 of the placebo group-specific AIDS diseases were cancers and dementia, of which only 3 and 2 were observed in the 500 and 1,500 mg AZT groups, respectively. This suggests a selection bias in favor of more healthy subjects for the AZT groups. The study is further compromised by the failure to report and to consider the recreational drug use histories and the many compensatory treatments of the subjects analyzed. 6. Within 48 weeks on AZT, 172, 56%, out of 308 Australian AIDS patients developed one or more new AIDS diseases, including pneumonia and candidiasis 116. This indicates that AZT induces AIDS diseases with less than 1 year and thus much faster than the 10 years HIV is said to need to cause AIDS 72. Likewise, no therapeutic benefits were observed in a study of 365 AIDS patients from France after 6 months on AZT, but new AIDS diseases and approximately 50% leukopenias and 20% deaths occurred within 9 months on AZT. Further, no therapeutic benefits were observed in 4 Norwegian AIDS patients after 6 months on AZT 98. 7. The annual lymphoma incidence of AZT-treated AIDS patients was reported to be 9% by the National Cancer Institute and was calculated to be 50% over 3 years 94. The lymphoma incidence of untreated HIV-positive AIDS risk groups is 0.3% per year and 0.9% per 3 years, derived from the putative average progression rate of 10 years from HIV to AIDS 33, 72, 83, and the 3% incidence of lymphoma in AIDS patients 18. Thus the lymphoma incidence is 30 to 50 times higher in AZT-treated than in untreated HIV-positive counterparts. In addition, during the past 3 years of AZT therapy, a progressive increase in the number of AIDS patients dying from lymphoma, to a current total of 16%, was noted in 1991 in a group of 346 AIDS patients in London, most of whom were on AZT 93. It is likely that the chronic levels of the mutagenic AZT, see below, at 10 to 33 pm, 500-1500 mg/person per day, were responsible for the lymphomas. The alternative proposal that HIV-induced immunodeficiency was responsible for the lymphomas 94 is unlikely, since cancers do not reflect a defective immune system 36, 115. 8. 10 out of 11 HIV-positive AZT-treated AIDS patients recovered cellular immunity after discontinuing AZT in favor or an experimental HIV vaccine 107, suggesting that AZT was a sufficient cause of immunodeficiency. 9. 4 out of 5 AZT-treated patients recovered from myopathy 2 weeks after discontinuing AZT, 2 redeveloped myopathy on renewed AZT treatment 118, indicating that AZT was sufficient for myopathy. 10. 4 patients with pneumonia developed severe pancytopenia and bone marrow aplasia 12 weeks after the initiation of AZT therapy. 3 out of 4 recovered within 4 to 5 weeks after AZT was discontinued 50, indicating that AZT was sufficient for pancytopenia. Drug use sufficient for AIDS indicator diseases in the absence of HIV. 1. Among intravenous drug users in New York representing a spectrum of HIV-related diseases, HIV was only observed in 22 out of 50 pneumonia deaths, 7 out of 22 endocarditis deaths, and 11 out of 16 tuberculosis deaths 114. 2. Pneumonia was diagnosed in 6 out of 289 HIV-free and in 14 out of 144 HIV-positive intravenous drug users from New York 109. 3. Among 54 prisoners with tuberculosis in New York State, 47 were street drug users but only 24 were infected with HIV. 12. 4. In a group of 21 heroin addicts, the ratio of helper to suppressor T cells declined within 13 years from a normal of 2 to less than 1, which is typical of AIDS 19, 63, but only 2 were infected by HIV 31. 5. Thrombocytopenia and immunodeficiency were diagnosed in 15 intravenous drug users on average 10 years after they became addicted, but 2 were not infected with HIV 104. 6. Lymphocyte reactivity and abundance was depressed by long-term injection of drugs not only in 111 HIV-positive but also in 210 HIV-free intravenous drug users from Holland 79. 7. The same lymphadenopathy, weight loss, fever, night sweats, diarrhea, and mouth infections were observed in 49 out of 82 HIV-free, and in about 40% of 113 intravenous drug users from France, of which 69 were HIV positive and 44 were negative 40. The French group had used drugs for an average of 5 years. 8. Among 6 HIV-free male homosexuals with Kaposi's sarcoma, 5 have reported the use of nitrite inhalants. 47. 9. Similar neurological deficiencies were observed among 12 HIV-infected and 16 uninfected infants of drug-addicted mothers. 66. Thus, the long-term use of recreational and anti-HIV drugs appears necessary in HIV positives and sufficient in HIV negatives to induce AIDS indicator and other diseases. Toxic effects of drugs used by AIDS patients. Toxicity of recreational drugs. From as early as 1991, evidence has accumulated that addiction to psychoactive drugs leads to immune suppression and clinical abnormalities similar to AIDS, including lymphopenia, lymphadenopathy, fever, weight loss, septicemia, and increased susceptibility to infections and neurological disorders. 13, 14, 30, 49, 55, 75, 78, 103, 110, 117, 120. Intravenous drugs can be toxic directly, or indirectly via malnutrition, both because of their enormous expense and via septicemia, since most illicit drugs are not sterile. 15, 24, 73, 114. Oral consumption of cocaine and other psychoactive drugs has been reported to cause pneumonitis, bronchitis, edema, and other diseases. 41. In children born to mothers addicted to cocaine and other drugs, physiological and neurological deficiencies including mental retardation are observed. 100, 119. According to the National Institute on Drug Abuse, cocaine is, from a public health perspective, the drug of the greatest national health concern. 54. Alkyl nitrites are directly toxic as they are rapidly hydrolyzed in vivo to yield nitrite ions, which react with all biological macromolecules. 70, 90. Addicts with 0.5 mm nitrite derivatives and 70% methemoglobin in the blood have been recorded. 90. Toxicity for the immune system, the central nervous system, the hematologic system, and pulmonary organs has been observed after short exposure to nitrites in humans and in animals. 88, 128. Further, alkyl nitrites were shown to be both mutagenic and carcinogenic in animals. 60, 64, 82. Several investigators have proposed that nitrite inhalants cause pulmonary 33 and skin Kaposi's sarcoma and possibly pneumonia by direct toxicity on the skin and oral mucosa. 17, 59, 77. In view of this, a causal link between nitrite inhalants and Kaposi's sarcoma and pneumonia in homosexuals was first suggested in 1982 by the CDC. 17 and other investigators. 59, 77. As a consequence, the sale of nitrite inhalants was banned by the U.S. Congress in 1988, Public Law 100 to 690, 58. Toxicity of AZT. Although AZT is an inhibitor of HIV DNA synthesis, it is not a rational medication for persons with antibodies to HIV for the following reasons. 1. There is no proof that HIV causes AIDS. 33, 36. 2. Since no detectable RNA-dependent viral DNA synthesis occurs, and since the number of infected cells remains stable once the virus is neutralized by antibodies. 33, 36, only cell DNA, with or without proviruses of HIV, is terminated by AZT treatment. Further, since AZT cannot distinguish infected from uninfected cells and only 1 in 500 T cells is infected in AIDS patients and asymptomatic carriers. 33, 36, 105, it kills 500 uninfected cells for every infected cell. Thus AZT is inevitably toxic, killing 500 times more uninfected than infected cells. 3. Particularly in view of the hypothesis that HIV causes AIDS by killing T cells. 62 to 63, it is irrational to overkill infected cells with AZT. As expected from an inhibitor of DNA synthesis, many studies report AZT-mediated cytotoxicity. Anemia, neutropenia, and leukopenia occur in 20 to 50% with about 30 to 50% requiring transfusions within several weeks. 32, 50, 99, 113, 116. Severe nausea from intestinal intoxication is observed in up to 45%. 99, 113, 123 and severe muscle atrophy in 6 to 8% 8, 52, 99, 118. Acute hepatitis, insomnia, headaches, dementia, seizures, and vomiting are also reported effects of AZT-113. Lymphomas appear in about 9% within 1 year on AZT-94. AZT is also mutagenic and carcinogenic in animals 23, 129 and transforms cells in vitro as effectively as methylcholinthrin 21. AZT toxicity varies a great deal with the subject treated, due to differences in kinases involved in its uptake and in AZT metabolism 21, 39, 113, 130. Nevertheless, AZT is thought to have serendipitous therapeutic benefits based on the only placebo-controlled study of its effects on AIDS patients 43, 99. The study was sponsored by Burroughs Wellcome, the manufacturer of AZT 43, 99. In this study T-cell counts were observed to increase from 4 to 8 weeks and then to decline to pre-treatment levels. Further, AZT was claimed to decrease mortality because only 1 out of 143 in the AZT-treated group died compared to 19 out of 135 in the placebo group. However, 30 out of the 143 in the AZT group depended on multiple transfusions to survive anemia, compared to only 5 out of the 135 in the placebo group. Since the number of subjects in the AZT group who would have died from anemia if untreated was 30, and thus larger than the AIDS deaths and anemias of the control group combined 19 plus 5, the claim of decreased mortality is not realistic 43, 99. Moreover, 66 in the AZT group suffered from severe nausea and 11 from muscle atrophy compared to only 25 and 3 in the control group. The lymphocyte count decreased over 50% in 34% of the subjects in the AZT group and in only 6% of those in the control group. The study is further compromised by concomitant medication 99, the failure to consider the effects of recreational drug use and of patient-initiated randomization of blinded AZT and placebo treatments 69. The brief AZT-induced gain of T cells may reflect compensatory hematopoiesis and random killing of pathogenic parasites 39 and the influence of concomitant medication 99. Surprisingly, long-term studies of AZT in animals compatible with human applications have not been published 113, 130. In view of the inevitable toxicity of AZT, its popularity as an anti-HIV drug can only be explained by the widespread acceptance of the virus AIDS hypothesis and the failure to consider the enormous difference between the viral and cellular DNA targets. Conclusions. It is concluded that all American AIDS exceeding the normal low incidence of indicator diseases in the general population is the result of recreational and anti-HIV drugs. Thus the American AIDS epidemic is a subset of the drug epidemic. For example, only the pneumonias, tuberculosis, and dementias of the 50% of American intravenous drug users with HIV-86 are recorded as AIDS, while those of their counterparts are diagnosed by their old names. Indeed, the drug AIDS hypothesis is epidemiologically and pathologically better grounded than the virus AIDS hypothesis. About 32% American AIDS patients are confirmed intravenous drug users, possibly 60% use recreational drugs orally, and an unknown but large percentage of both behavioral and clinical AIDS risk groups use AZT. Moreover, the consumption of recreational drugs by AIDS patients is probably underreported because the drugs are illicit, and because medical scientists and support for research are currently heavily biased in favor of viral AIDS 35, 41, 73. The toxicity of these drugs is empirically known for all drugs, and mechanistically for some, notably for AZT and nitrites. Nonetheless, evidence for the role of drugs in AIDS is rejected by proponents of the virus AIDS hypothesis 6, 11, 91, 125. This may be the reason that despite the current drug use epidemic there are no studies that investigate the long-term effects of psychoactive drugs and AZT in animals compatible with the time periods and dosages used by AIDS patients 73. Yet, it is a complete mystery how HIV acts as a pathogen, despite enormous research efforts 6, 9, 33, 74, 125, and even antibodies against are confirmed in only about 50% of AIDS patients 33, 63, 108. The drug AIDS hypothesis resolves all scientific paradoxes posed by the prevailing virus AIDS hypothesis. 1. In America HIV is a long-established, endemic virus, but AIDS is new because the drug epidemic is new. 2. AIDS is restricted for over 10 years to 10,018 or 0.01% of the over 100 million sexually active heterosexual Americans per year, and to 20,018 or 0.25% of the 8 million homosexuals, estimated at 10% of the adult male population 120. But conventional venereal diseases are on the rise in the US 5, and there is no vaccine or drug against HIV. This is because AIDS is due to drug consumption rather than sexual activity. 3. Over 72% of American AIDS cases are 20-44-year-old males 18. Although no AIDS disease is male-specific 18, 19, 63 because males of this age group consume over 80% of all hard psychoactive drugs 51, 87. 4. Distinct AIDS diseases occur in distinct risk groups because they use distinct drugs, e.g. users of nitrites get Kaposi's sarcoma, users of intravenous drugs get tuberculosis and users of AZT get leukopenia or anemia. 5. Viral AIDS occurs on average 10 years after HIV infection 33, 36, 63, although infectious agents, as self-replicating toxins, typically strike within weeks or months after infection 46, 80. Indeed, HIV is immunogenic and may be mildly pathogenic in humans within weeks after infection and is then effectively and rapidly limited by antiviral immunity 22, 25. This is because HIV infection and AIDS are unrelated events. The duration and toxicity of drug consumption and individual thresholds for disease determine when AIDS occurs, of when and whether HIV infect. On average, 5 to 10 years elapse between the first use of drugs and the need for treatment 31, 40, 54. 6. HIV, as well as many other parenterally and venereally transmitted microbes and viruses, are mere markers for AIDS and AIDS risks 27, 33, 45 because the higher the consumption of unsterile, injected drugs 28, 40 and sexual contacts mediated by aphrodisiac drugs, the more microbes are accumulated. 7. Some old diseases of hemophiliacs, other recipients of transfusions, and of the general population are called AIDS, because they follow perinatal or parenteral HIV infection 33. 8. Old African diseases like slim disease, fever, diarrhea, and tuberculosis are called AIDS now, although they are clinically and epidemiologically very different from American and European AIDS. They occur in adolescents and adults of both sexes that are subject to protein malnutrition, parasitic infections, and poor sanitary conditions 36. Since HIV is endemic in over 10% of Central Africans, over 10% of their AIDS-defining diseases will be called AIDS 33, 36, 63. The drug AIDS hypothesis predicts that the AIDS diseases of the behavioral AIDS risk groups in the US and Europe can be prevented by stopping the consumption of recreational and anti-HIV drugs, but not by safe sex 63 and clean injection equipment 86 for unsterile street drugs. According to the drug AIDS hypothesis, AZT is AIDS by prescription. Screening of blood for antibodies to HIV is superfluous, if not harmful, in view of the anxiety that a positive test generates among the many believers in the virus AIDS hypothesis and the toxic AZT prophylaxis, prescribed to many who test positive. Eliminating the test would also reduce the cost of the approximately 12 million annual blood donations in the US 127 by $11 each. Personal Communication 1990, Irwin Memorial Blood Bank San Francisco, and would lift travel restrictions for antibody positives to many countries including the US and China. The drug AIDS hypothesis is testable epidemiologically and experimentally by studying AIDS drugs in animals. Note added in proof. In Europe 33% of AIDS patients are intravenous drug users and 47% are male homosexuals and 86% of all patients are male 132. Acknowledgements. I thank B. Ellison, Berkeley, J. Lauritsen, New York, C. Pirac, Minneapolis; P. Rubinow, Berkeley; H. Rubin, Berkeley; F. Rothschild, Berkeley; J. Shenton, London; C. Thomas Jr., San Diego; and M. Verney Elliott, London, for critical information, and T. Gardner, Santa Barbara, for a generous donation and encouragement. I am supported by Outstanding Investigator Grant No. 5R35CA39915-07 from the National Cancer Institute. End of article. Falsification and Progress. By Peter Duesberg. Rethinking AIDS, August 1992. Progress in the science of biology, like progress in biology itself, depends almost entirely on falsification. Evolution has falsified billions of point mutations to produce the readers of this newsletter. Falsification is the critical element for progress. It provides room for alternative ideas. David Horobin, the editor of Medical Hypothesis, writing in The Scientist, November 1990, notes, biomedical science may be more complex than physics and chemistry and therefore a more difficult field in which to theorize. But if we do not try, we shall certainly not succeed. We, more than any other scientists, labor under the misapprehension that our observations are theory-free. Because we are so little conscious of theory, we fail to develop coherent underlying concepts against which our observations are clearly tested. We have no tradition of analytical criticism of existing theoretical concepts. In addition, most established scientists do not actually want to make rapid progress. A state of theoretical confusion is agreeable to them because in that state almost nothing can ever be shown to be wrong. As a result, general beliefs persist within the biomedical community long after the evidence is available to destroy them. One of these general beliefs holds that all viruses are pathogenic, because some of them are. According to Gallo and Montagnier, a new virus must cause a new disease, their new disease, new agent postulate, Scientific American 1988, 259:41. This seems plausible because in this century some of the worst human epidemics, the smallpox, yellow fever, flu, and most recently polio, have all been diagnosed as viral diseases and most have been eliminated or controlled with vaccines developed by scientists whose names are still household words, like Andrews, Burnett, Enders, and Sabin. But in fact, most viral infections, even by potentially pathogenic viruses, are latent or restricted to small numbers of cells and hence asymptomatic. Above all, the modern virus hunters pursue their latent viruses oblivious to the lessons of lysogeny. Lysogenic lambda phage provided the first examples of how even the most deadly viruses can be harmless genetic passengers, as long as their cytocidal genes are inactive. Despite enormous efforts to date, the belief that the latent virus HIV is the cause of AIDS has not been proven and has not yielded clinically useful results, a traditional indication for a poorly grounded hypothesis. Instead, it has generated unprecedented volumes of scientific dinosaurs and a pandemic of public hysteria. According to Horobin, discovery in science almost always begins with an observation that can be seen to be anomalous against the background of clearly defined theoretical construct. It is hoped that AIDS will be the ultimate loser in the present battle for the best hypothesis. Asterisk. End of article. The HIV Gap in National AIDS Statistics. By Peter H. Duesberg. Bio/Technology, August 11, 1993. The HIV/AIDS hypothesis rests on the assertion that all AIDS cases are associated with HIV, Confronting AIDS Update 1988, Nat Academic Sci Press, Washington, D.C., Blattner, W., ETAL, 1988, Science 241, 514-515, Weiss, R., and Jaff, H., 1990, Nature 345, 659-660, Duesberg, P.H., 1992, Pharmacol. The 55, 201-277. Therefore, the Centers for Disease Control, CDC, groups American AIDS cases in its HIV/AIDS surveillance into exposure to HIV categories. However, there are no national AIDS statistics that document the natural coincidence between AIDS diseases and HIV. Contrary to its title, the HIV/AIDS surveillance of the CDC does not report HIV tests. Correlations between HIV and AIDS can only be determined from individual studies and from those CDC AIDS case report forms that include HIV tests. But most HIV tests measure antibodies against HIV rather than the virus itself. And antibodies are not unambiguous evidence for the presence of a virus, nor are they rational predictors for viral disease. Instead, antibodies neutralize HIV and restrict the virus to latency. This is the reason that leading AIDS researchers have had notorious difficulties in isolating HIV, even in people dying from AIDS. Weiss, R. 1991, Nature 349, 374, Cohen, J. 1993, Science 259, 168-170. Moreover, antibody tests generate false positive results if an epitope is shared between different organisms. According to a recent review entitled HIV Testing, State of the Art, depending on the population tested, 20 to 70% of 2 successive positive ELISAs, enzyme-linked immunosorbent assay, are confirmed by Western blot, an alternative antibody assay. Sloan, E.M., ETAL, 1991, JAMA 266, 2861-2866. In a population with a low probability of infection, the false positive rate is high. According to the widely cited study of applicants to the U.S. Army by Burke et.al., 83% of all initially positive ELISAs, 10,000/12,000, were false positives. New ENGJ Med 319, 961-964, 1988. In a population with a high incidence of infection, however, the false positive rate is expected to be low. Therefore, the CDC assumes that the tiny proportion of possibly false positive screening tests in persons with AIDS-indicative diseases is of little consequence, confronting AIDS Update 1988. But this is not observed. For example, one study documented 131 repeatedly ELISA-positive homosexual men with negative Western blots in a cohort of 4,994 homosexuals, of which 37% were HIV positive. Fair, J. ETAL, 1992, J. AIDS 5, 988-992. Another study found HIV-I infection in only 12.5%, of 32 high-risk cases with repeatedly positive ELISAs. SEM, CI, ETAL, 1991, J. Infect. DIS 164, 656-664. HIV infection was negative by Western blot, provirus amplification with the polymerase chain reaction, PCR, and virus isolation tests. Another study identified 33 ELISA-positive and even Western blot-positive subjects who were HIV negative based on the PCR test for HIV DNA. Schechter, M. ETAL, 1991, AIDS 5, 373-379. These subjects were from a group of 316 homosexuals, of which 158, 50%, were PCR positive. The relatively high incidence of false positive HIV antibody tests in these HIV risk groups probably reflects the presence of antibodies to other viruses and microbes that may cross-react with HIV. For example, 7 out of 10 blood donors treated with an influenza virus vaccine in 1991 became HIV ELISA positive. Each of these proved to be false positives upon confirmation with a Western blot. Mackenzie, W.R., ETAL, 1992, JAMA 268, 1015-1017. Since the CDC accepts a reactive screening test for HIV antibody without confirmation by a supplemental test, confronting AIDS Update 1988, and does not request a repeatedly positive antibody test in its AIDS Adult Confidential Case Report forms, it includes false positives in its HIV/AIDS surveillance. In fact, the CDC even includes AIDS cases in its HIV/AIDS surveillance without laboratory evidence regarding HIV infection, Confronting AIDS Update 1988. Upon request, the CDC's director of the HIV/AIDS division, Harold Jaffe, stated that the HIV status of 43,606 out of the 253,448 American AIDS cases recorded by the end of 1992 was not tested, per com. 1993. However, this figure seems to be an understatement. Obviously, all 10,360 American AIDS cases diagnosed before the HIV antibody test, i.e., before 1985, were not tested. HIV/AIDS surveillance, February 1993. In addition, the CDC published that approximately one-third of AIDS patients in the United States have been from New York and San Francisco, where, since 1985, 7% have been reported with HIV antibody test results, compared with 60% in other areas, Confronting AIDS Update 1988. Thus, between 1985 and 1987, 58%, 93% x one-third plus 40% x two-thirds, of the 56,807 AIDS cases recorded in that period, or 32,948, have not been tested. For 1988, the CDC reported that 27% or 9,039 of the 33,480 AIDS cases recorded for that year were not tested for HIV, Celiac, RM, ETAL, 1990, J-AIDS 3, 73-82. According to the CDC's Technical Information Activity, 3,682 AIDS cases without an HIV test were recorded in 1989, 2,888 in 1990, 1960 in 1991, and 1,395 in 1992, per com. 1993. Thus, at least 62,272, or 18,666 more than Jaffe reports, were not tested. Determination of the HIV-AIDS correlation is further obscured because HIV-free AIDS cases are not recorded in the CDC's HIV/AIDS surveillance. By 1993, at least 4,621 HIV-free AIDS cases had been documented in the .S, Europe and Africa with the clinical AIDS definition, Table 1. Even Jaffe, again upon request, reported 89 HIV-free AIDS cases, per com. 1993. The cases recorded in Table 1 suffered from one or more of the over 25 heterogeneous AIDS-defining diseases and from AIDS-defining immunodeficiencies without diseases. Some of these proved to be HIV-free even by PCR amplification of viral RNA and DNA. Table 1. HIV-free AIDS-defining diseases and immunodeficiencies. Risk Group US and Canada Europe Africa, references *. Homosexuals 722-37, 1-22/23-26,74. Intravenous, 4, drug users 251-335, 18-20,2735,75/3639,74. Infants of 4 drug users 5511, 40-43/44,45. Hemophiliacs 256-78, 46-58/57-61. None/unreported 307-14 2555, 16-21,62-67/21,68/26,69-73. Totals 1,591 475 2,555. Sum total 4,521. Table 1 includes some American and European immunodeficiencies that may not exactly fit the current definition of AIDS-defining immunodeficiency without disease, which is 200 T cells per microliter, CDC, 1992, MMWR 41, RR 17, 1-19, as for example, HIV-free male homosexuals on various recreations drugs with X cells per cubic millimeter, Table 1, Ref 14, or HIV-negative hemophiliacs with T4/T8 cell ratios of about 1 or 1, Table 1, Refs 46-61. But even if not all of these cases fit the current definition of AIDS defining immunodeficiency exactly, they do so prospectively. This is because their T cells typically continue to decline either because of risk behavior, such as the consumption of recreational drugs, or because of clinical AIDS risks, such as chronic transfusion of foreign proteins as prophylaxis against hemophilia. Duesberg, Ph. 1992, OPCIT. Since a clinical definition is used in Africa, statistics from this continent are not biased against HIV-free AIDS. For example, 2,215 out of 4,383, 50.0%. African AIDS patients from Abidjan, Ivory Coast, Lusaka, Zambia, and Kinshasa, Zaire, were HIV antibody negative, Table 1, Ref 70, 71. Another study using antibody tests and supplementary PCR tests for HIV reports 135, 59%. HIV-free AIDS patients from Ghana out of 227 suffering from weight loss, diarrhea, chronic fever, tuberculosis, and neurological diseases, Table 1, Ref 72. Only 30%, of a group of 122 African tuberculosis patients were HIV positive, according to a study published in 1993, Table 1, Ref 73. An earlier study documents 116 HIV negatives among 424 African patients, and Montagnier et al. Diagnosed HIV in 4 out of 8, Table 1, Ref 26, 69. It follows that about 50% of the African AIDS cases, or 65,000 of the 129,000 diagnosed by 1992, Duesberg, Ph. 1992, OPCIT, may be HIV-free and thus not caused by HIV. Instead of considering the potential usefulness of HIV-free AIDS cases in the search for the cause of AIDS, the CDC and the NIH's Director for AIDS hid in 1992 the then rapidly growing numbers of HIV-free AIDS cases. Duesberg, Ph. 1992, OPCIT, under a new name, idiopathic CD4 lymphocytopenia or ICL. Indeed, the new name has sent HIV-free AIDS cases into obscurity. But efforts to set apart HIV-free from HIV-positive AIDS cases by the new term are not based on clinical or scientific arguments. According to an editorial by Anthony Fossey, HIV-free AIDS or ICL cases are unlike the HIV-positive cases because, 1. Given the heterogeneity of the ICL syndrome, it is highly likely that there is no common cause, and because, 2. Approximately one-third of the patients are women, as compared with 11% among those with HIV. In America, Fossey, A. 1993, New ENGJ Med 328, 429-431. Yet proponents of the HIV hypothesis, including Fossey, insist that HIV is the common cause of the more than 25 heterogeneous AIDS diseases and that HIV causes African AIDS, although about 50% of the African patients are women. Duesberg, Ph. 1992, OPCIT. In view of the above, I submit that the natural coincidence between HIV and AIDS in America and Europe remains unknown, and is certainly less than perfect. Thus arguments for the etiological role of HIV in AIDS, which assume a perfect correlation, are fundamentally flawed. End of article. The Enigma of Slow Viruses. Book Review. By Peter Duesberg. The Lancet, 18 September 1993. P. Libersky, The Enigma of Slow Viruses, Facts and Artifacts, Archives of Virology 6, Springer Verlag 1993, DM 250. ISBN 32211822428. Virology and the law have much in common, a few basic rules like the Ten Commandments and Koch's postulates, and many practitioners who can obscure the basic rules with specialized interpretations. A highly specialized attorney may secure acquittal of a drug dealer caught in the act, despite the Ten Commandments, and a scientist can blame phantom viruses for rare genetic diseases, despite the rules of virology. The later case is the subject of this book written for the 70th anniversary of Carleton Gajdusek. Gajdusek won the 1977 Nobel Prize in Medicine for the hypothesis that human neurodegenerative diseases such as Kuru, Creutzfeldt-Jakob disease, Alzheimer's disease, and scrapie in sheep are caused by unconventional slow viruses that may induce disease months or even decades after infection. Science 1977, 197, 943-60. This hypothesis has attracted attention because it suggests that Kuru is naturally transmitted by cannibalism practiced by the Fore tribe in New Guinea. Gajdusek showed that the disease can be experimentally transmitted to chimpanzees by inoculating their brains with homogenates of the brains of patients with Kuru who died. But Gajdusek and his followers have not shed much light on this viral hypothesis over the past 20 years. Despite numerous efforts, neither the Kuru virus nor any other slow virus has been isolated. Therefore, the claim for an infectious origin of these neurological diseases is open to several questions. Why was intracerebral inoculation, instead of feeding, chosen as a model for transmission by cannibalism? Were toxins excluded? Clearly, brain homogenates of death Kuru patients must be toxic. Is human Kuru indistinguishable from diseases observed in monkeys intracerebrally inoculated with brain homogenates? Moreover, Gajdusek's claim about cannibalism has been questioned because the original photographic documentation that claimed to depict a human was in fact a pig. Science 1986, 232:1497. Challenged to provide further evidence, Gajdusek cited arrests for cannibalism of New Guineans by Australian authorities. Science 1986, 233:926. Despite his admiration for Gajdusek and his fascination with slow viruses, Libersky retains an element of skepticism, as the title of his book suggests. It is probably for this reason that he dedicates a lot of space to an hypothesis that offers a self-replicating protein, a prion, as a solution to the undetectable enigmatic viruses. The paradox of a self-replicating protein seemed to be solved when the prion proved to be a cellular protein. But that created a new enigma, a pathogenic cellular protein. Prion researchers quickly proposed that the mutated prions were to be blamed for neurological disease, until some patients proved to have normal prions and some normal people proved to have mutated prions. According to this book, conventional and slow viral diseases are about as compatible as night and day. For example, in conventional viral diseases, all viruses conate a nucleic acid and can be seen under the electron microscope. Conventional viral diseases elicit a febrile response, whereas slow viral diseases do not. Conventional viral diseases, like viruses, spread horizontally, but neurodegenerative diseases such as Kuru are familial, tribal, or genetically determined, just like conventional genetic diseases. For conventional viral diseases, the latent period between infection and disease is determined by the generation time of viruses, 8 to 48H, and the number of cells that need to be infected for pathogenicity. For slow viral diseases, the latent period is determined by incubation period genes of the host. In the author's words, the purpose of his book was to summarize almost all existing data on scrapie and related infections, asking whether they fit one complete pattern. Having written this review, the author is convinced that such a task is not possible. However, if one changes one's point of view just a little, a surprisingly complete pattern emerges. The neurodegenerative diseases are conventional genetic diseases and the slow viruses are phantoms of virology. But can we afford an alternative hypothesis in view of the number of papers, more than 1,000, on slow viruses that Libersky's book cites? Certainly not until the scientific monarchy of the slow virologists is broken, a process that may be slower than the alleged neuropathogenesis by slow viruses. Asterisk. End of article. Results fall short for HIV theory. By Peter H. Duesberg. Insight, February 14, 1994. After more than a decade, the war on AIDS has been a dismal failure. At the 1993 International AIDS Conference in Berlin, even the most dogged researchers were beginning to admit their frustration, and the mood of the conference was generally pessimistic. As the number of AIDS victims continues to rise, so do the bewildering questions. Why do so many people infected with the virus remain healthy? Why do homosexuals manifest radically different diseases than the hemophiliacs to whom they donated infected blood? Why can we not, as the most technologically sophisticated biomedical establishment in history, produce a vaccine, as was previously accomplished for polio, measles, and other viruses? Where is a safe, effective treatment for AIDS? Such questions are asked not only by the so-called HIV/AIDS dissidents, such as myself, but also by an increasing number of mainstream AIDS researchers and concerned scientists, including this year's Nobel laureate in chemistry, Kary Mullis. The government currently is spending $6 billion annually for AIDS research and treatment, according to the U.S. Public Health Service. Yet despite spending more than $22 billion since 1982, and despite a staggering 75,000 scientific papers written during the last decade, a cure for AIDS remains as elusive as ever. It now appears likely that AIDS researchers have made a terrible mistake in blaming HIV, the so-called AIDS virus, for causing AIDS. This fatal assumption mostly was the result of a rush to judgment in 1984 when virologist Robert Gallo from the National Institutes of Health, along with the Department of Health and Human Services, announced at an international press conference that acquired immune deficiency syndrome was caused by a retrovirus, now known as human immunodeficiency virus. The announcement was made in the New York Times before even one American study on HIV had appeared in scientific literature. It was welcomed by the Reagan administration as a quick answer to gay pressures for a solution to the growing AIDS epidemic. Gallo and his collaborators cited antibodies against the virus in about 85% of patients with AIDS as the evidence for their hypothesis. Yet that was their only evidence. In the scientific papers that followed, HIV was said to cause AIDS by depleting the white blood cells known as T cells. The hypothesis proposed that HIV would cause 30 previously known diseases, including a number of diseases that are not consequences of immunodeficiency, such as cancer, weight loss, and dementia. It now appears that the HIV/AIDS hypothesis is the only link that holds together the 30 heterogeneous AIDS diseases. AIDS is defined as a syndrome that occurs only in the presence of HIV. For example, if tuberculosis occurs in the presence of antibodies to HIV, it is AIDS. In the absence of those antibodies, it is tuberculosis. Given this definition, the link between HIV and AIDS is unfalsifiable. However, to date, the HIV/AIDS hypothesis remains just that, an unproven hypothesis. It is supported only by circumstantial evidence, primarily by the claim that all AIDS patients carry antibodies against HIV. But this correlation is biased by the practice of excluding from AIDS statistics those patients with AIDS-defining diseases in whom no trace of HIV can be found. The disease of such a patient will be diagnosed either by its old name, for example, pneumonia or Kaposi's sarcoma, or will be called idiopathic CD4 lymphocytopenia. This explains why some researchers see the perfect correlations between HIV and AIDS. To date, the virus AIDS hypothesis has failed to yield any public health benefits. No vaccine has been developed and AIDS continues to spread despite efforts to stop the spread of HIV. However, the acid test of a hypothesis is not to produce useful results, but to make accurate predictions. The HIV/AIDS hypothesis makes the following testable predictions, none of which has been proved. AIDS in America would explode from the original risk groups via sexual transmission into the general population. Like all other sexually transmitted diseases, AIDS would tend to strike an equal share of both genders. In America, AIDS has remained in the original risk groups: male homosexuals, male and female intravenous drug users and recipients of transfusions. Since 1981, 90% of all American AIDS patients have been males. The spread of AIDS would follow the dissemination of HIV. Although AIDS increased in America from a few hundred to about 50,000 cases annually in the last 10 years, HIV did not spread at all. Ever since HIV became detectable in 1985, an unchanging 1 million Americans have been HIV positive. To account for this discrepancy, defenders of the hypothesis claim the virus has a latency period of 10 years or more. Healthcare workers would contract AIDS from their patients, scientists from propagating virus, and prostitutes from their clients, particularly in the absence of an anti-HIV vaccine or drug. Not a single confirmed case exists in scientific literature of a healthcare worker who contracted AIDS from one of the more than 250,000 American AIDS patients. None of the tens of thousands of HIV researchers has developed AIDS from propagating HIV. And prostitutes have not picked up AIDS from their clients, despite the absence of antiviral vaccines or effective drugs. Chimpanzees inoculated with HIV would develop AIDS, and the 15,000 American hemophiliacs who were infected iatrogenically before 1984 would die from AIDS. Not one of the 150 chimpanzees inoculated with HIV since 1983 has developed AIDS. Contrary to the prediction, the median life of American hemophiliacs has doubled during the last 10 to 15 years after 75%, 15,000, already had been infected by transfusions. Natural or vaccine-induced anti-HIV immunity would cure AIDS or protect against future AIDS. Natural antiviral immunity that is observed in many AIDS patients does not protect against AIDS. It would be demonstrated that HIV causes AIDS by killing the white blood cells known as T cells. The hallmark of all retroviruses is that they do not kill cells that they infect. HIV is the only retrovirus that is asserted to kill its host cell. Several researchers, including HIV discoverer Luc Montagnier, have found that HIV does not kill its host cell in laboratory tests. All AIDS diseases are the consequence of HIV-mediated T cell deficiency. About 61% of all American AIDS diseases, opportunistic infections such as Pneumocystis carinii, Candida, tuberculosis etc., are related to breakdown of the immune system. But 39%, including Kaposi's sarcoma, lymphoma, 10% weight loss and dementia, are neither caused by nor consistently associated with immunodeficiency. 2 studies of homosexuals with Kaposi's sarcoma report that the immune systems of 20 and 19 of the subjects were normal when their disease was first diagnosed. AIDS would be restricted by controlling the spread of HIV via safe sex and through programs adopting the use of clean needles for the injection of street drugs. AIDS continues to increase despite the safe sex and clean needle programs. These points, among many others, give me reason to believe that HIV is in fact a harmless virus. AIDS may be a non-infectious condition acquired by recreational drugs and other non-contagious risk factors. Even proponents of the HIV/AIDS hypothesis, such as Jaap Goudsmit of the University of Amsterdam, have been forced to admit that AIDS does not have the characteristics of an ordinary infectious disease. Indeed, AIDS does not meet even one of the common criteria of known infectious diseases. First, all infectious diseases are equally distributed between the genders and never remain tightly segregated in special risk groups. This is especially true of the venereal diseases, including herpes, syphilis, gonorrhea, and chlamydia. This also is true of hepatitis, the blood-borne virus long considered the model for predicting the spread of HIV. Despite loud publicity to the contrary by vested interests, AIDS has not been spreading to females as predicted nor to the heterosexual population at large. Second, all infectious diseases caused by viruses, the virus infects every individual with the disease and the virus is abundant and very active in target tissues during the course of the illness. However, at least 4,621 AIDS cases have been documented since 1984 in which there is no HIV. About one-third of these, 1,691, were recorded in the US, 475 were recorded in Europe and 2,555 in Africa. Since Africa uses only the clinical, rather than the HIV-based AIDS definition, most of these cases were observed there. Several scientific teams have also documented that, even in those patients who are HIV infected, the virus is usually totally dormant once immune deficiency is acquired and AIDS appears. Using standard laboratory techniques now available for decades, the active, infectious form of the virus cannot be isolated from the blood or other tissues of most HIV-positive AIDS patients. Even the dormant form of HIV, resting quietly inside infected cells, can be found in only 1 out of every 1,000 T cells in the patient. Nor is there a correlation between AIDS and the number of HIV-infected cells. There are, for example, healthy infected individuals with up to 43 times the rate of HIV-infected cells in AIDS patients. Third, infectious diseases typically follow within days or weeks after infection by viruses, before the immune system has had time to make antibodies against the invader. This is because all viruses begin replicating in the body within hours after infection and multiply into armies of viruses within days or weeks unless stopped by an antiviral immunity. As a rule, viruses strike quickly or not at all. Although it has become fashionable among many scientists to believe in special slow viruses, or lentiviruses, which are said to take months or years to multiply, such viruses have never actually been found. In those HIV-positive people who eventually develop AIDS, the syndrome appears only after unpredictable latent periods averaging about 10 years. When HIV first infects a person, it can reach moderately high concentrations in the blood, yet AIDS never shows up at that time and T cell levels remain normal. Within days or weeks, the immune system makes antibodies against HIV, and the virus quickly disappears from the blood. Years later, if AIDS shows up at all, the virus rarely comes back to life and multiplies again. In other words, AIDS never strikes a patient until years after the active virus has been permanently eliminated from the body. This strongly suggests that AIDS is caused by something else. As an alternative to the HIV explanation, I suggest that AIDS in America and Europe is caused by the long-term use of recreational drugs and by the toxic effects of anti-HIV treatments, and that African AIDS is an unrelated epidemic caused by malnutrition, parasitic infections, and poor sanitation. Indeed, AIDS in America and Europe fits all classical criteria of a drug-induced disease syndrome. Part of the evidence for this hypothesis is that about 30% of all American and European AIDS patients are intravenous drug users. This group includes nearly all heterosexuals with AIDS. It also includes 80% of America and European babies with AIDS who were drug users before birth because their mothers injected drugs during pregnancy. Since 1982, virtually 10% of homosexual males with AIDS or at risk for AIDS have been long-term users of oral, aphrodisiac drugs, particularly nitrite inhalants that confer euphoria and facilitate anal intercourse. Epidemiological studies from San Francisco and Vancouver, British Columbia, have just confirmed, in 1993, that 100% of several hundred male homosexuals with AIDS had used multiple recreational drugs. The immunotoxicity of recreational drugs has been documented in the literature since 1909. In addition, some had also used the drug azithromycin, or AZT, as an antiviral agent. AZT was originally developed 30 years ago to kill human cells in chemotherapy. About 200,000 HIV-positive healthy people and AIDS patients are currently treated 4 times daily with AZT and other drugs that attack the DNA chains within the cell. But these drugs kill all growing cells, particularly those of the highly proliferative immune system. Thus AZT could, by itself, trigger AIDS. Indeed, Sigma, an American chemical company, accords AZT the highest warning against toxicity, a skull with crossbones. In the United States, recreational drug use has increased during the last decade at about the same rate as AIDS. For example, cocaine consumption increased 200-fold from 1980 to 1990, based on the fact that the amount of cocaine seized by authorities increased from 500 kilograms in 1980 to 100,000 kilograms in 1990. During the same period, cocaine-related hospital emergencies increased 24-fold from 3,296 cases in 1981 to 835.5 cases in 1990. Note the increase in AIDS and the rise in cocaine and cocaine-related hospital emergencies run parallel since 1981, whereas HIV has failed to spread since at least 1984. This would also explain why 90% of American AIDS patients are males, since, according to the Bureau of Justice Statistics, males consume about 75% of all illicit injected drugs. In addition, homosexual males are virtually the only consistent users of aphrodisiac drugs such as alkyl nitrites. Furthermore, we could then explain why AIDS occurs, on average, 10 years after initiation of risk behavior. The great variations in latent periods from HIV to AIDS may in fact be the time periods required by individuals to accumulate sufficient drug toxicity to generate AIDS diseases. It takes years of recreational drug consumption to cause disease, just as it frequently takes 20 years of smoking to get lung cancer or emphysema. In view of the multiple failures of the HIV hypothesis, it is high time to look at alternative hypothesis of causation. But since 1984, virtually every dollar of funding from the federal government for AIDS research has been devoted to work premised on the causal role of HIV and awarded only to grant recipients who do not question that causal role. Such a disproportionate research focus on a single avenue of investigation could conceivably be justified if it had resulted in meaningful advance. Yet, sadly, this massive, narrowly targeted expenditure of public money has not resulted in saving or prolonging a single life. Asterisk. End of article. Infectious AIDS. Stretching the germ theory beyond its limits. Peter Duesberg. Department of Molecular and Cell Biology, University of California, Berkeley, California, USA. Abstract. The hypothesis that human immunodeficiency virus, HIV, causes AIDS was advanced in 1984, based only on circumstantial evidence. To this date, the primary evidence are correlations between the presence of antibody against HIV and AIDS. But these correlations are biased by proponents of the HIV hypothesis in favor of HIV. They ignore HIV-free AIDS and they base correlations on selected studies because there are no national HIV/AIDS statistics. The HIV/AIDS hypothesis has made the following predictions. 1. AIDS would explode from the original risk groups into the general population via sexual transmission of HIV. 2. Healthcare workers would contract AIDS from their patients, scientists from propagating HIV, and prostitutes from their clients. 3. The 150 chimpanzees that have been experimentally inoculated with HIV, and the 15,000 American hemophiliacs who have been iatrogenically inoculated before 1984, would develop AIDS. 4. Immunity and vaccines would protect against AIDS. 5. HIV would cause AIDS by killing T cells. 6. AIDS would occur only in people infected by HIV. But none of these predictions proved to be correct. Recent studies show that HIV is a passenger virus instead of the cause of AIDS. 1. AIDS occurs at unpredictable intervals after infection. 2. HIV may be active, passive, or totally absent from otherwise identical AIDS cases. Indeed, AIDS does not meet one of the classical criteria of infectious disease. 1. Equal distribution between the sexes. 2. Disease following infection within days or weeks, the time microbes take to become either immunogenic or pathogenic or both. 3. The presence of a common active microbe. Therefore it is proposed that American and European AIDS is caused by the long-term consumption of recreational drugs and the anti-HIV drug AZT. This hypothesis is testable and provides a rational basis for AIDS control. Introduction. In April 1984, the retrovirologist Robert Gallo from the National Institutes of Health in Bethesda and the American Secretary of Health and Human Services announced, at an international press conference in Washington, that the acquired immunodeficiency syndrome, AIDS, was caused by a retrovirus, now termed human immunodeficiency virus, HIV. 1. The announcement was made before even one American study on HIV had appeared in the scientific literature. Gallo and his collaborators cited antibodies against the virus in about 85% of patients with AIDS as the only evidence for their hypothesis. 1. Although AIDS occurred despite antiviral antibodies, the researchers expressed hope that a vaccine would be ready in about 2 years. 1. In the scientific papers that followed the next month, HIV was said to cause AIDS by depleting T cells. 2. 3. The hypothesis proposed that HIV would cause all of the 30 heterogeneous AIDS diseases including those that are not consequences of immunodeficiency, such as cancer, weight loss, and dementia. Table 1, 5. Infectious AIDS, from hypothesis to dogma. In 1986, the American Academy of Sciences and the Institute of Medicine assembled a Blue Ribbon Committee of medical scientists to confront the growing AIDS epidemic. The committee, chaired by David Baltimore, concluded that the isolation of HIV by Montagnure et al. 6 and Gallo et al. 2 led to its definitive identification as the cause of AIDS. 7. Without mention of dissent. 8, 9, a derivative committee declared in 1988, the committee believes that the evidence that HIV causes AIDS is scientifically conclusive. Boldface in original and proposed to rename AIDS HIV disease. 10. The committees had sealed the hypothesis into national dogma. However, the committee's conclusion was based only on circumstantial evidence including 5 questionable assumptions. 1. The primary assumption was that, close to 100% of the affected individuals can be found to harbor the virus, and the probability that this distribution might have occurred by chance is less than 1 in a million. 10. However, since no national HIV/AIDS statistics exist. 11. The committee had to rely on selected individual studies and unpublished observations. For example, the committee selected a Science News and Comment article entitled "A Rebel Without a Cause of AIDS." 9. An unpublished speech of the epidemiologist Winkelstein and the papers of Montagnure and Gallo as the source for the close to 100% correlation. 10. But the original paper by Montagnure et al. 6 only reported a single isolation of HIV from the lymph node of a person who did not have AIDS, and Gallo's isolate proved to be Montagnure's virus. 12. And the authenticity of the HIV/AIDS correlations from Gallo's group has since been questioned on several accounts. 12. 2. The committee also believed that the virus is not found in persons who are not at risk for infection, assuming that infection was restricted to AIDS risk groups, e.g. male homosexuals, intravenous drug users and recipients of transfusions. 7. However, HIV has since been found in 1 million healthy Americans, 0.5 million healthy Europeans, 1.5 million healthy South Americans, 1.5 million healthy Asians and 8 million healthy Africans. 13. 3. The committee further believed that AIDS is unknown in populations that are free of HIV antibodies, i.e. that there is no HIV-free AIDS. 10. However, many HIV-free AIDS cases had already been reported by 1986 and 1988, when the committees confronted AIDS. 11. 4. The committee accepted without questioning the unique practice of the HIV researchers to present antibodies against HIV as pathogenic powers of HIV. Proponents of the HIV/AIDS hypothesis interpret these antibodies as indicators of current and future HIV disease. However, antibodies against all other microbes are signs of rejection of the microbe and protection against future disease. 5. The committee accepted uncontrolled statistics as evidence for AIDS from transfusion of HIV. 10. For example, AIDS researchers blame HIV for pneumonia and other opportunistic infections that occur in about 2% of HIV-positive hemophiliacs per year. 5, 10. However, controlled studies have since shown that the incidence of immunodeficiency in matched groups of HIV-positive and negative hemophiliacs is the same. 5. Thus the committee had adopted the virus AIDS hypothesis on the basis of questionable assumptions, primarily the assumption that all AIDS correlates with HIV. How good is the correlation between HIV and AIDS? The natural coincidence between HIV and AIDS can only be determined by first diagnosing AIDS clinically and then testing for HIV. However, since the HIV/AIDS hypothesis has been accepted in 1986, the definition of AIDS by clinical criteria alone has been abandoned in America and Europe in favor of an HIV-based definition. 10. Moreover, all HIV/AIDS correlations are based on selected individual studies, because to date no national and international AIDS statistics reporting HIV tests exist. 11. As a result, proponents of the HIV/AIDS hypothesis bias HIV/AIDS correlations in several ways. 1. They cite HIV/AIDS correlations from selected, individual studies which are frequently based on non-standardized and unconfirmed HIV antibody tests 11, 14. 2. They present antibodies against HIV, instead of activities and titers of HIV, as a rational cause of AIDS. 3. They exclude clinically diagnosed, HIV-free AIDS-defining diseases from their statistics, e.g., the 4,621 cases cited below 11, because the HIV-AIDS hypothesis postulates that HIV causes AIDS. Therefore HIV-free AIDS cases are either diagnosed by their old names, e.g. Kaposi sarcoma, pneumonia, etc., or renamed idiopathic CD4 lymphocytopenia, or ICL-15. But the effort to set apart HIV-positive from HIV-negative AIDS cases is not based on any clinical or convincing epidemiological criteria 11, 16. According to an editorial by Fossey, given the heterogeneity of the ICL syndrome, it is highly likely that there is no common cause 15. Yet at the same time the proponents of the HIV hypothesis, including Fossey, insist that HIV must be the common cause of the 30 heterogeneous AIDS diseases. The editorial also argues that the HIV-free AIDS or ICL cases are unlike the HIV-positive cases because approximately one-third of the patients are women, as compared with 11% among those with HIV in America. But proponents of the HIV-AIDS hypothesis, including Fossey, insist that HIV also causes African AIDS, despite about 50% of the African patients being women 10. Indeed, other retroviruses have been proposed as causes of HIV-free AIDS, particularly at the 8th International AIDS Conference in 1992 in Amsterdam 17, 18, because these cases were clinically indistinguishable from HIV-positive AIDS. Following the HIV precedent, these retroviruses were considered new AIDS causes simply because of their presence in these cases. It follows that the primary argument for the HIV-AIDS hypothesis, the HIV-AIDS correlation, is a circular argument. It is in reality an artifact of the HIV-based AIDS definition, which is a restatement of the HIV hypothesis. To date, the virus AIDS hypothesis has been a complete failure in terms of public health benefits. No vaccine has been developed, AIDS continues to spread despite efforts to stop the spread of HIV, and nobody has ever been cured from AIDS. However, the acid test of a hypothesis is not to produce useful results, but to make accurate predictions. Predictions of the HIV-AIDS hypothesis. The HIV-AIDS hypothesis makes the following testable predictions, none of which proved to be correct. 5, 19. The failure to make valid predictions is the hallmark of a flawed hypothesis. It raises two fundamental questions. 1. Is HIV a passenger virus rather than the cause of AIDS? 2. Is AIDS infectious? Is HIV a passenger virus rather than the cause of AIDS? The correlation argument assumes that the presence of a virus in a disease is sufficient proof of causation. But the presence of a virus in a disease is by no means proof of causation. Particularly since HIV does not cause AIDS if inoculated into chimpanzees or if iatrogenically introduced into hemophiliacs, it could be just a harmless passenger virus. Passenger viruses are widely distributed, in mammals, causing no obvious disease. 37. In the absence of functional proof, the distinction between a causative and a passenger virus can be made by the temporal relations between infection and disease, by the consistency of its presence, and by the biochemical activity of the virus during the course of the disease as follows. HIV meets all criteria of a harmless passenger virus in AIDS. 1. HIV infection precedes AIDS by unpredictable intervals that average about 10 years. This time is referred to as latent period of HIV by proponents of the HIV-AIDS hypothesis, although HIV typically remains latent even when AIDS occurs. 5. Several groups report that HIV may reach high titers during the primary infection 40 to 42. According to Piatok et al., these titers range from 10 to 104 infectious units per milliliter 42. Despite these relatively high HIV titers in some people, and despite the absence of antiviral immunity in all, there is no AIDS during the primary infection 40 to 42. In addition, the T-cell counts are normal 42, 43. When the primary infection is terminated by antiviral immunity, no infectious HIV remains, the T-cell are normal and there is also no AIDS-42. In the face of antiviral immunity, the virus persists as a latent provirus in healthy hosts, figure 2. 2. HIV also meets one of the most telling criteria of a passenger virus in relation to a disease, HIV-free AIDS, see above, figure 2. At least 4,621 AIDS cases have been documented in the literature since 1984 in whom there is no HIV-11. About a third of these, 1,691, were recorded in the US, 475 in Europe and 2,555 in Africa. 11. Since Africa uses the clinical, rather than the HIV-based AIDS definition, most of these cases were observed in Africa. The US and Europe bias AIDS statistics against HIV-free AIDS, because they use the HIV-based AIDS definition, see above. 3. Several groups have documented that HIV may be either active or passive once immunodeficiency is acquired and AIDS appears. 42, 44, 45, Piatok et al. Observe either no infectious HIV, e.g. 0 infectious units per milliliter plasma in 5 out of 27 HIV antibody-positive AIDS cases, or fewer than 25 infectious HIVs per milliliter in 6 out of 27 cases, or 102 to 105 in 16 out of 27 otherwise identical AIDS cases. 42, 43. Others have reported similar non-correlations between virus titers and AIDS. 44 to 46. Likewise, there is no correlation between AIDS and the number of HIV-infected cells. Simmons et al. Report that there are from 1 to 700 to 1 in 83,000 HIV-infected leukocytes in healthy HIV carriers and from 1 in 900 to 1 in 30,000 in AIDS patients. 47. Bagasra et al. Report that there are from 1 in 30 to 1 in 1,000 infected leukocytes in healthy carriers and from 1 in 10 to 1 in 1,000 in patients with fatal AIDS. 48. Thus there are healthy persons with 43 times 30,000:700, and 33 times 1,000:30, more HIV-infected cells than in AIDS patients. It follows that there is neither a correlation between HIV titers, nor between the number of HIV-infected cells and AIDS, the hallmark of a passenger virus. 4. Even as an active passenger, HIV does not aggravate the course of AIDS by any HIV-specific symptom, as some other passenger viruses or microbes do. For example, cytomegalovirus, herpes virus, pneumocystis carinii, and candida each cause corresponding opportunistic infections if they are activated by acquired immunodeficiency, Table 1. By contrast, the AIDS cases with active or passive HIV appear to be identical, according to several groups of investigators. 42, 44, 45, 48. Indeed, no HIV-specific AIDS symptom has ever been described, as all AIDS-defining diseases have been known previously. 10, 49, and occur in HIV-free AIDS patients. 11. Thus HIV is not even a cofactor for AIDS. It follows that HIV is a harmless passenger virus that does neither cause AIDS nor even contribute an HIV-specific symptom to AIDS. Since AIDS is not caused by HIV nor consistently associated with another active infectious agent. 5. It may not be infectious. AIDS fails all criteria of infectious disease. Proponents of the HIV/AIDS hypothesis acknowledge that AIDS does not have the characteristics of an ordinary infectious disease. This view is incontrovertible. 50. More specifically, the epidemiologists Eggers and Weyer 51 state that the spread of AIDS does not behave like the spread of a disease that is caused by a single sexually transmitted agent. To reconcile AIDS with infectious disease, they simulated a cofactor that cannot be identified with any known infectious agent. 52. The epidemiologists Anderson and May 53 had to invent assortative scenarios for different AIDS risk groups to match AIDS with infectious disease. Indeed, AIDS does not meet even one of the common criteria of all known infectious diseases. Thus AIDS does not fit even one of the classical criteria of an infectious disease. The drug AIDS hypothesis. The paradoxa of the virus AIDS hypothesis are all readily resolved by postulating non-infectious AIDS. In view of this, I have proposed that AIDS in America and Europe is caused by the long-term consumption of recreational drugs and AZT 559. African AIDS has been proposed to be an unrelated epidemic caused by malnutrition, parasitic infections, and poor sanitation 5. Indeed, AIDS in America and Europe fits all classical criteria of a drug-induced disease syndrome. AIDS correlates epidemiologically and chronologically with the drug epidemics that started in America and Europe after the Vietnam War. 1. About 30% of all American and European AIDS patients are intravenous drug users. 4. 5. This group includes nearly all heterosexuals with AIDS. 4. 5. It also includes 80% of all American and European babies with AIDS who were intrauterine drug users, because their mothers injected drugs during pregnancy. 5. It is known since 1982 that virtually 100% of homosexual males with AIDS or at risk for AIDS have been long-term users of oral, aphrodisiac drugs, particularly nitrite inhalants, that confer euphoria and facilitate anal intercourse. 60 to 69. Epidemiological studies from San Francisco and Vancouver have just confirmed, in 1993, that 100% of several hundred male homosexuals with AIDS had used multiple recreational drugs. 70, 71, 81. In addition, some had also used the cytotoxic DNA chain terminator AZT as antiviral drug. 72 to 75, 81. The immunotoxicity of these recreational drugs has been documented in the literature since 1909. 5, 76. About 200,000 HIV-positive healthy people and AIDS patients are currently treated 4 times daily with AZT and other DNA chain terminators as anti-HIV drugs. These drugs kill all growing cells, particularly those of the highly proliferative immune system. 5. Thus AZT is AIDS by prescription. 2. In the US, recreational drug use increased over the last years at about the same rate as AIDS. 5. For example, cocaine consumption increased 200-fold from 1980 to 1990 based on cocaine seizures that increased from 500 kilograms in 1980 to 100,000 kilograms in 1995. During the same time, cocaine-related hospital emergencies increased 24-fold from 3,296 cases in 1981 to 80,355 cases in 1995, Figure 1b. Note the parallelisms between the spreads of AIDS, Figure 1a, and the spreads of cocaine and cocaine-related hospital emergencies since 1981, and the contrast with the non-spread of HIV since 1984, Figure Law. 3. 90% of the American AIDS patients are male, because according to the U.S. Bureau of Justice Statistics, males consume about 75% of all illicit injected drugs, and because homosexual males are virtually the only consistent users of aphrodisiac drugs like alkyl nitrites. 5, 59. 4. AIDS occurs on average 10 years after initiation of risk behavior, because it takes years of recreational drug consumption to cause disease. 5, 63, 77, e.g., 20 years of smoking to get lung cancer. 78. Or emphysema, or years of alcoholism to develop liver cirrhosis. The great variations in latent periods from HIV to AIDS that currently average 10 years. 10 are euphemisms for the time required by individuals to accumulate sufficient drug toxicity to generate AIDS diseases. 5. 5. Different risk groups have risk group specific AIDS diseases, e.g., Kaposi sarcoma is observed almost exclusively in homosexuals. 79. Because homosexuals are the almost exclusive users of aphrodisiac nitrite inhalants. 5. 65. Tuberculosis and weight loss is observed in intravenous drug users, because intravenous drugs cause those symptoms. 5. Anemia and lymphocytopenia is observed in recipients of AZT, which kills proliferating bone marrow cells. 5. 80. And hemophiliacs get pneumonias and candidiasis almost exclusively, because long-term transfusion of foreign proteins is immunosuppressive. 5. The drug AIDS hypothesis is experimentally and epidemiologically testable and provides a rational basis for AIDS prevention and control. Asterisk. This article is the first of 2 in Controversy, HIV and AIDS, sponsored by George Wick, Editor-in-Chief of International Archives of Allergy and Immunology. Acknowledgements. I thank Jody Schwartz for a critical review and Brian Ellison for discussions. Supported in part by the Council for Tobacco Research, USA, and private donations from Glenn Braswell, Los Angeles, California, USA, Dr. Richard Fisher, Annandale, Virginia, USA, Dr. Fabio Franchi, Trieste, Italy, and Dr. Friedrich Luft, Berlin, FRG. End of article. The culprit is non-contagious risk factors. By Peter Duesberg. The Scientist, March 20, 1995. Would you have believed in 1984 that AIDS was infectious if you had known that not even one of the healthcare workers who have treated more than 401,000 American AIDS patients over the last 10 years contracted AIDS from a patient despite the absence of an antiviral vaccine or effective drug? No infectious agent that is so difficult to transmit could survive as a microbe. Moreover, the median age of the 15,000 total American HIV-positive hemophiliacs increased from 11 in 1972 to 27 in 1987, although they were all infected in the decade prior to 1984. From 1988 to 1993 their annual risk of AIDS diseases, not corrected for the normal incidence of HIV-free AIDS diseases, was constant at 2%. Centers for Disease Control and Prevention, HIV/AIDS Surveillance, 5:133, 1994, Ph. Duesberg, Jean Tika, in Press. The hypothesis that HIV causes AIDS after 10 years predicts that more than half of them would have died by now. Furthermore, if HIV is a blood-borne virus, one would expect that recipients of HIV from transfusions would develop all AIDS-defining diseases, yet nobody ever contracted AIDS Kaposi's sarcoma from a transfusion. In contrast to AIDS, all known infectious diseases are characterized by equal distribution between the sexes, the appearance of primary symptoms within days or weeks after infection, and prior to antimicrobial immunity, a common, active microbe in all acute cases, and patients who are generally under 20, with underdeveloped immune systems, or over 60, with failing immune systems. While sexually transmitted diseases share AIDS's age distribution, between 20 and 50, they are all equally distributed between the sexes. For references on this and other points, see Ph. Duesberg, Proceedings of the National Academy of Sciences, 88:1575-9, 1991, Ph. Duesberg, Pharmacology and Therapeutics, 55:201-77, 1992, Ph. Duesberg, International Archives of Allergy and Applied Immunology, 103:131-42, 1994. A passenger virus. Owing to the relentless campaigns against viruses by epidemiologists and virologists, even scientists have forgotten that the vast majority of viruses never cause disease: retroviruses, adenoviruses, reoviruses, echoviruses, and so forth. Just like humans, viruses are here to continue their species, and they succeed best if they do this without harming their host, certainly without causing a fatal disease. Such viruses are called passenger viruses. Lionel Crawford and H. Frankel Conrad, ed., Molecular Basis of Virology, New York, Van Nostrand Reinhold, 1968, pages 393 to 434. A passenger virus infects at totally unpredictable times, even 10 years, prior to a disease, just like HIV, while a causative microbe is pathogenic within days or weeks. Given a generation time of 2 days and a yield of 100 HIVs per cell, AIDS should occur 14 days after infection if it were a causative microbe. By that time, HIV would have infected all 1014 cells of the body. Moreover, a passenger virus may be absent during a disease, as in the 4,621 HIV-free cases of AIDS reported in the literature. And, if present, a passenger virus can be active or passive during disease. In the majority of AIDS patients, active HIV cannot be found, only antibodies against the virus. Because of antiviral immunity, HIV infects only about 1 in 1,000 T cells of AIDS patients. There are only a minority of cases in the literature in which HIV is found in moderate or high titers. Thus, I propose that HIV is merely a passenger virus, which does not cause any disease. A hypothesis. Diseases can be acquired by microbes but are now acquired in the US and Europe much more often by drugs. There are 50 million smokers and many millions of alcoholics in the US acquiring emphysema, lung cancer, and liver cirrhosis. And millions use illicit recreational drugs. In view of this, I propose that all AIDS diseases in America and Europe that exceed their long-established, normal backgrounds are caused by the long-term consumption of illicit recreational drugs and by AZT and its analogues. The normal low background of AIDS-defining diseases like tuberculosis, diarrhea, and pneumonia would be a consequence of their long-established causes. Hemophilia AIDS, transfusion AIDS, and the extremely rare AIDS cases of the general population reflect the normal incidence of the AIDS-defining diseases, plus the AZT-induced incidence of these diseases under a new name. African AIDS is likewise a new name for old diseases caused by malnutrition, parasitic infections, and poor sanitation. The drug hypothesis predicts American/European AIDS precisely. 1. American AIDS will be restricted to intravenous and oral users of recreational drugs and of AZT. Since 1981, 94% of all American AIDS cases have been from risk groups who had used such drugs. About one-third of these were intravenous drug users. CDC, HIV/AIDS Surveillance Report, 6:1-27, 1994, and two-thirds were male homosexuals who had used oral recreational drugs and AZT. The drug correlation is even better than 94% if those HIV-positive hemophiliacs and transfusion recipients receiving AZT are included. 2. American AIDS predominantly affects adult males, because they are the predominant users of recreational drugs and AZT. CDC reports that 87% of all American AIDS patients are males. This number is the sum of the following constituents. First, the National Institute on Drug Abuse, NIDA, and the Bureau of Justice Statistics report that more than 75% of hard, recreational drugs are consumed intravenously by males. Second, CDC and independent investigators report that nearly all male homosexuals with AIDS and at risk for AIDS are long-term users of oral drugs such as nitrite inhalants, ethyl chloride inhalants, amphetamines, cocaine, and others to facilitate sexual contacts, particularly anal intercourse. A.R. Lifson et al., American Journal of Epidemiology, 131:221-31, 1990, M.S. Asher et al., Nature, 362:103-4, 1993. And third, many HIV-positive homosexuals are prescribed AZT. 3. American AIDS is new because the American drug use epidemic is new. In the U.S., recreational drug use is epidemiologically new, as it has increased over the last 2 decades from statistically undetectable levels to epidemic levels at about the same rate as AIDS. Cocaine consumption increased 200-fold from 1980 to 1990, based on cocaine seizures that increased from 500 kilograms in 1980 to 100,000 kilograms in 1990. During the same time, cocaine-related hospital emergencies increased from 3,296 cases in 1981 to 80,355 cases in 1990 and 119,843 in 1992. Amphetamine consumption has increased 100-fold from 1980 to 1990, Bureau of Justice Statistics, Catalog of Federal Publications on Illegal Drug and Alcohol Abuse, Washington, D.C., U.S. Department of Justice, 1991. According to a recent report from NIDA and CDC, nitrite use has increased in the 1990s in gay men in Chicago and San Francisco after a decline in the 1980s. And in 1992 alone, 200,000 HIV positives were given AZT. For only a small fraction of drug users will get AIDS. The cumulative total of 401,749 American AIDS cases since 1981 that were reported in June 1994 have been recruited from a much larger reservoir of drug users. There were 8 million cocaine addicts in the U.S. in 1992. In 1980, 5 million Americans had used nitrite inhalants. In 1989, 100 million doses of amphetamines were consumed in the U.S. In addition, about 150,000 HIV-positive Americans were on AZT in 1992. The small percentage of AIDS patients among the many American drug users represents the heaviest drug abusers, just as lung cancer and emphysema generally occur among the heaviest smokers. The long latent period of HIV is a euphemism for the time that its human host needs to accumulate sufficient drug dosage for AIDS. 5 specific drugs cause specific AIDS-defining diseases. Indeed, group-specific drug use explains risk group-specific AIDS diseases. For example, Kaposi's sarcoma as an AIDS diagnosis is 20 times more common among homosexuals who use nitrite inhalants than among AIDS patients who are intravenous drug users or hemophiliacs. Because of their carcinogenic potential, nitrites were originally proposed as causing Kaposi's sarcoma. H. W. Haverkos, J. A. Doherty, eds. Health Hazards of Nitrite Inhalants, NIDA Research Monograph 83, Washington, D.C., U.S. Department of Health and Human Services, 1988. Aggressive and life-threatening Kaposi's sarcoma is observed exclusively in male homosexuals, E. Sloan et al., Southern Medical Journal, 86:1219-24, 1993, 32% of which occur in the lungs, the primary site of exposure to nitrite inhalants. D. H. Irwin, L. D. Kaplan, Seminars in Respiratory Infections, 8:139-48, 1993. 80% of pediatric AIDS cases in America and Europe are also predicted by the drug hypothesis. These children were born to mothers who were intravenous drug users during pregnancy. The remainder reflects the normal incidence of AIDS-defining diseases among newborns. Case studies. The abundant AIDS literature in fact includes conclusive evidence that cessation of drug use prevents, stabilizes, or cures AIDS-defining diseases. For example, 10 out of 11 HIV-positive, AZT-treated AIDS patients recovered cellular immunity after discontinuing AZT in favor of an experimental vaccine. M. Scolaro et al., Lancet, 337:731-2, 1991. Since AZT is now licensed for treatment or prevention of AIDS, no large-scale studies have been done in which patients are taken off AZT. Another investigation found that the T-cell counts of 65 HIV-positive intravenous drug users from New York dropped 35% over 9 months, compared with stable T-cell counts for HIV-positive controls who had stopped injecting. D. Desjarlais et al., AIDS, 1:105-11, 1987. In yet another report, the T-cells of 29% of 1,020 HIV-positive male homosexuals and intravenous drug users even increased over 2 years. M. D. Hughes et al., Journal of Infectious Diseases, 169:2836, 1994. These HIV positives belonged to the placebo arm of an AZT trial for AIDS prevention and thus were not intoxicated by AZT. There was no mortality reported among a group of 918 British HIV-positive homosexuals who had avoided the experimental medications on offer and chose to abstain from or significantly reduce their use of recreational drugs, including alcohol, during the course of a study lasting 1.25 years. J. Wells, Capital Gay, August 20, 1993, pages 14 to 15. Assuming a 10-year latent period from HIV to AIDS, the virus AIDS hypothesis would have predicted at least 115 AIDS cases among 918 HIV positives over 1.25 years, 918/10 x 1.25. Furthermore, as of July 1, 1994, there was still not a single AIDS case in this group. J. Wells, London, Personal Communication. In sum, the drug AIDS hypothesis correctly predicts all aspects of American/European AIDS, while the HIV hypothesis predicts none. The solution of AIDS could be as close as a very testable, and very affordable, alternative hypothesis. Asterisk. End of article. Foreign protein-mediated immunodeficiency in hemophiliacs with and without HIV. Peter H. Duesberg. Department of Molecular and Cell Biology, Stanley Hall, University of California at Berkeley, Berkeley, California 94720 USA. Abstract. Hemophilia AIDS has been interpreted in terms of two hypotheses, the foreign protein AIDS hypothesis and the human immunodeficiency virus, HIV, AIDS hypothesis. The foreign protein AIDS hypothesis holds that proteins contaminating commercial clotting factor VIII cause immunosuppression. The foreign protein hypothesis, but not the HIV hypothesis, correctly predicts 7 characteristics of hemophilia AIDS. 1. The increased lifespan of American hemophiliacs in the 2 decades before 1987, although 75% became infected by HIV because factor VIII treatment, begun in the 1960s, extended their lives and simultaneously disseminated harmless HIV. After 1987 the lifespan of hemophiliacs appears to have decreased again, probably because of widespread treatment with the cytotoxic anti-HIV drug AZT. 2. The distinctly low, 1.3 to 2%, annual AIDS risk of hemophiliacs, compared to the higher 5 to 6% annual risk of intravenous drug users and male homosexual aphrodisiac drug users because transfusion of foreign proteins is less immunosuppressive than recreational drug use. 3. The age bias of hemophilia AIDS, i.e. that the annual AIDS risk increases twofold for each 10-year increase in age because immunosuppression is a function of the lifetime dose of foreign proteins received from transfusions. 4. The restriction of hemophilia AIDS to immunodeficiency diseases because foreign proteins cannot cause non-immunodeficiency AIDS diseases, like Kaposi's sarcoma. 5. The absence of AIDS diseases above their normal background in sexual partners of hemophiliacs because transfusion-mediated immunotoxicity is not contagious. 6. The occurrence of immunodeficiency in HIV-free hemophiliacs because foreign proteins, not HIV, suppress their immune system. 7. Stabilization, even regeneration, of immunity of HIV-positive hemophiliacs by long-term treatment with pure factor VIII. This shows that neither HIV nor factor VIII plus HIV are immunosuppressive by themselves. Therefore, AIDS cannot be prevented by elimination of HIV from the blood supply and cannot be rationally treated with genotoxic antiviral drugs, like AZT. Instead, hemophilia AIDS can be prevented and has even been reverted by treatment with pure factor VIII. 1. The drug and hemophilia AIDS epidemics in America and Europe. About 30 previously known diseases are now called AIDS if they occur in the presence of antibody against human immunodeficiency virus, HIV, Institute of Medicine, 1988, Centers for Disease Control and Prevention, 1992. These diseases are thought to be consequences of an acquired immunodeficiency syndrome and hence are grouped together as AIDS, Institute of Medicine, 1988. From its beginning in 1981, AIDS has been restricted in America and Europe to specific risk groups, Centers for Disease Control, 1986, World Health Organization, 1992b. Currently, over 96% of all American AIDS cases come from AIDS risk groups, rather than from the general population, Centers for Disease Control, 1993. These include over 60% male homosexuals who have been long-term oral users of psychoactive and aphrodisiac drugs, 33% mostly heterosexual, intravenous drug users and their children, 2% transfusion recipients, and about 1% hemophiliacs. Duesberg, 1992a, Centers for Disease Control, 1993. Altogether, about 90% of all American and European AIDS patients are males. World Health Organization, 1992a, Centers for Disease Control, 1993. Each risk group has specific AIDS diseases. For example, Kaposi's sarcoma is almost exclusively seen in male homosexuals, tuberculosis is common in intravenous drug users, and pneumonia and candidiasis are virtually the only AIDS diseases seen in hemophiliacs. Duesberg, 1992a. In view of these epidemiological and clinical criteria, American and European AIDS has been interpreted alternatively as an infectious and a non-infectious epidemic by the following hypotheses. 1. The virus AIDS hypothesis. This hypothesis postulates that all AIDS is caused by the retrovirus HIV, and thus an infectious epidemic. The inherent danger of a transmissionable disease quickly promoted the HIV hypothesis to the favorite of responsible healthcare workers, scientists, and journalists. Booth, 1988. For example, a columnist of the New York Times wrote in July 1994 that all non-HIV/AIDS science is cruelly irresponsible anti-science. Lewis, 1994. And the retrovirologist David Baltimore warned in Nature there is no question at all that HIV is the cause of AIDS. Anyone who gets up publicly and says the opposite is encouraging people to risk their lives. Massel-Wayne, 1994. Moreover, the U.S. Centers for Disease Control, CDC, have favored the HIV/AIDS hypothesis from the beginning. Centers for Disease Control, 1982, Centers for Disease Control, 1986, Schiltz, 1987, Booth, 1988, Oppenheimer, 1992, because according to Red Cross official Paul Cumming in 1983 the CDC increasingly needs a major epidemic to justify its existence. Associated Press, 1994. Indeed, there has been no viral or microbial epidemic in the US and Europe since polio in the 1950s. All infectious diseases combined now account for less than 1% of morbidity and mortality in the Western world. Cairns, 1978. The control of infectious diseases is the primary mission of the CDC. 2. The drug AIDS hypothesis. This hypothesis holds that AIDS in the major risk groups is caused by group-specific, recreational drugs and by anti-HIV therapy with cytocidal DNA chain terminators like AZT and is thus not infectious. Lauritsen and Wilson, 1986, Haverkas and Doherty, 1988, Duesberg, 1991, 1992a, Oppenheimer, 1992. The drug AIDS hypothesis was favored by many scientists, including some from the CDC, before the introduction of the HIV/AIDS hypothesis in 1984, Marmer et al., 1982, Mather-Wack et al., 1984, Haverkas et al., 1985, Mather-Wack, Mildvan and Sini, 1985, Newell et al., 1985, Haverkas and Doherty, 1988, Duesberg, 1992a, Oppenheimer, 1992. 3. The foreign protein hemophilia AIDS hypothesis. This hypothesis holds that hemophilia AIDS is caused by the long-term transfusion of foreign proteins contaminating factor VIII and other clotting factors and thus not infectious. This hypothesis also preceded the virus hypothesis and has coexisted with it, despite the rising popularity of the HIV hypothesis. See Section 3. The infectious and non-infectious AIDS hypotheses indicate entirely different strategies of AIDS prevention and therapy. Here we analyze the cause of hemophilia AIDS in the lights of the HIV/AIDS hypothesis and the foreign protein AIDS hypothesis. The hemophiliacs provide the most accessible group to test AIDS hypotheses of infectious versus non-infectious causation. This is because the time of infection via transfusion can be estimated more accurately than HIV infection from sexual contacts, and because the role of treatment-related AIDS risks can be controlled and quantitated much more readily than AIDS risks due to the consumption of illicit, recreational drugs. 2. The HIV/AIDS hypothesis. The HIV hypothesis claims that AIDS began to appear in hemophiliacs in 1981, Centers for Disease Control, 1982, because hemophiliacs were accidentally infected via transfusions of factor VIII contaminated with HIV since the 1960s, when widespread prophylactic factor VIII treatment began, but no longer after 1984 when HIV was eliminated from the blood supply, and because, 2. AIDS is currently assumed to follow HIV infection on average only after 10 years, Centers for Disease Control, 1986. Institute of Medicine, 1988, Corba et al., 1994. Indeed, about 15,000 of the 20,000 American hemophiliacs, or 75%, are HIV antibody positive from transfusions of HIV-contaminated clotting factors received before HIV was detectable. Sukas et al., 1984, Institute of Medicine and National Academy of Sciences, 1986, Sullivan et al., 1986, McGrady, Jason and Evett, 1987, Institute of Medicine, 1988, Koerper, 1989. Contamination of factor VIII with HIV reflects the practice, developed in the 1960s and 1970s, of preparing factor VIII and other clotting factors from blood pools collected from large numbers of donors. Aronson, 1983, Koerper, 1989, Korba et al., 1994. The HIV hypothesis claims that 2,214 American hemophiliacs developed AIDS-defining diseases between 1982 and the end of 1992 because of HIV. Centers for Disease Control, 1993. However, this corresponds only to a 1.3% annual AIDS risk, i.e. 201 cases per 15,000 HIV-positive hemophiliacs per year. Note that the non-age-adjusted annual mortality of an American with a life expectancy of 80 years is 1.2%. Further, the HIV/AIDS hypothesis claims that the mortality of hemophiliacs has increased over twofold in the 3-year period from 1987 to 1989 compared to periods from 1968 to 1986, although infection with HIV via transfusions had already been halted with the HIV antibody test in 1984. Korba et al., 1994. HIV is thought to cause immunodeficiency by killing T cells, but paradoxically only after the virus has been neutralized by antiviral immunity, and only on average 10 years after infection. Institute of Medicine, 1988, Duesberg, 1992a, Weiss, 1993. However, HIV, like all other retroviruses, does not kill T cells or any other cells in vitro. In fact, it is mass-produced for the HIV antibody test in immortal T cell lines. Duesberg, 1992a. Moreover, the basis for the 10-year latent period of the virus, which has a generation time of only 24 to 48 H, is entirely unknown. Duesberg, 1992a, Weiss, 1993, Fields, 1994. It is particularly paradoxical that the loss of T cells in hemophiliacs over time does not correspond to viral activity in abundance. No T cells are lost prior to antiviral immunity, when the virus is most active. Duesberg, 1993a, Piatok et al., 1993. Instead, most T cells are lost when the virus is least active or latent in hemophiliacs. Phillips et al., 1994a. And other risk groups. Duesberg, 1992a, 1993a, 1994, Piatok et al., 1993, Shepard, Asher and Kroka, 1993, namely after it is neutralized by antiviral immunity, a positive HIV antibody test. Indeed, there are healthy, HIV antibody-positive persons in which 33 to 43 times more cells are infected by latent HIV than in AIDS patients. Simmons et al., 1990, Bagasra et al., 1992, Duesberg, 1994. Even Gallo, who claims credit for the HIV/AIDS hypothesis, Gallo et al., 1984, has recently acknowledged, "I think that if HIV is not being expressed and not reforming virus and replicating, the virus is a dud, and won't be causing the disease. Nobody is saying that indirect control of the virus is not important." Jones, 1994. There is also no explanation for the profound paradoxes that AIDS occurs only after HIV is neutralized and that antiviral immunity does not protect against AIDS, although this immunity is so effective that free virus is very rarely detectable in AIDS patients. Duesberg, 1990, 1992a, 1993a, Piatok et al., 1993. The high efficiency of this antiviral immunity is the reason that leading AIDS researchers had notorious difficulties in isolating HIV from AIDS patients. Weiss, 1991, Cohen, 1993. All of the above associations between HIV and AIDS support the hypothesis that HIV is a passenger virus, instead of the cause of AIDS. Duesberg, 1994. A passenger virus differs from one that causes a disease in 3 criteria. 1. The time of infection by the passenger virus is unrelated to the initiation of the disease. For example, the passenger may infect 10 years prior to, or just immediately before, initiation of the disease, just as HIV does in AIDS. 2. The passenger virus may be active or passive during the disease, i.e. the disease is not influenced by the activity of the passenger virus or the number of virus-infected cells, as is the case for HIV and AIDS. 3. The disease may occur in the absence of the passenger virus. In the case of AIDS, over 4,621 HIV-free AIDS cases have been clinically diagnosed. Duesberg, 1993b, see also section 4.6. Therefore, HIV meets each of the classical criteria of a passenger virus exactly. Duesberg, 1994. Moreover, since HIV is not active in most AIDS patients, and often more active in healthy carriers than in AIDS patients, Duesberg, 1993a, 1994, Piatok et al., 1993, and since AIDS patients with and without HIV are clinically identical, Duesberg, 1993b, HIV is in fact only a harmless passenger virus. It is harmless, because it does not contribute secondary diseases to AIDS pathogenicity, as for example pneumocystis pneumonia, candida, or herpes virus do. These microbes each cause typical AIDS-defining opportunistic infections. But HIV does not appreciably affect the pathogenicity of AIDS, as HIV-free and HIV-positive AIDS cases are clinically indistinguishable. Duesberg, 1993b, 1994. Likewise, there is no clinical distinction between AIDS cases in which HIV is active and those in which it is totally latent and restricted to very few cells. Duesberg, 1993a, Piatok et al., 1993. Thus, despite enormous efforts in the last 10 years, there is no rational explanation for viral pathogenesis, and the virus AIDS hypothesis stands unproved. Weiss and Jaffe, 1990, Duesberg, 1992a, Weiss, 1993, Fields, 1994. Above all, the hypothesis has failed to make any verifiable predictions, the acid test of a scientific hypothesis. For example, the predicted explosion of AIDS into the general population, or among female prostitutes via sexual transmission of HIV, or among healthcare workers treating AIDS patients via parenteral transmission did not occur. Duesberg, 1992a, 1994. As yet, the hypothesis is supported only by circumstantial evidence, i.e. correlations between the occurrence of AIDS and antibodies against HIV in AIDS patients. Blattner, Gallo and Temin, 1988, Institute of Medicine, 1988, Weiss and Jaffe, Weiss, 1993. However, because AIDS is defined by correlation between diseases and antibodies against HIV, Institute of Medicine, 1988, the relevance of the correlation argument for AIDS etiology has been challenged. Duesberg, 1992a, 1993b, 1994, Thomas Jr., Mullis and Johnson, 1994. States Mullis, at a London Sunday Times Nobel laureate lecture in 1994, any postgraduate student who had written a convincing paper demonstrating that HIV causes AIDS would have published the paper of the century. Dixon, 1994. In view of the circularity of the correlation argument, the apparent transmission of AIDS to hemophiliacs via transfusion of HIV-infected blood or factor VIII has been cited as the most direct support for the virus AIDS hypothesis. Blattner, Gallo and Temin, 1988, Institute of Medicine, 1988, Weiss and Jaffe, 1990, Weiss, 1993. However, the HIV hemophilia AIDS hypothesis is weakened by the extremely long intervals between infection and AIDS, averaging between 10 years, Institute of Medicine, 1988, and 35 years, Duesberg, 1992a, Phillips et al., 1994b, compared to the short generation time of HIV, which is only 24 to 48 h. See section 4.2. During such long intervals, other risk factors could have caused AIDS diseases, particularly in hemophiliacs who depend on regular transfusions of clotting factors for survival. The fact that HIV is typically not more active, and often even less active, in those who develop AIDS than in those who are healthy, further weakens the HIV hemophilia AIDS hypothesis, see above. 3. The foreign protein hemophilia AIDS hypothesis. Before the introduction of the HIV AIDS hypothesis, but after the introduction of prophylactic long-term treatment of hemophilia with blood-derived clotting factors had begun, numerous hematologists had noticed immunodeficiency and corresponding opportunistic infections in hemophiliacs. Several of these had advanced the foreign protein hemophilia AIDS hypothesis, which holds that the long-term transfusion of foreign proteins contaminating commercial factor VIII, and possibly factor VIII itself, is the cause of immunosuppression in hemophiliacs. Indeed, until recently most commercial preparations of factor VIII contained from 99% to 99.9% foreign, non-factor VIII proteins. Bredler and Levine, 1989, Manuxi et al., 1992, Sirmatis et al., 1993, Jerset et al., 1994. According to the foreign protein hypothesis, immunodeficiency in hemophilia patients is proportional to the lifetime dose of foreign proteins received. Manitov et al., 1983, Madhawk et al., 1986, Schulman, 1991. Long before HIV had been discovered, it was known empirically that transfusion of patients undergoing renal transplantation is associated with improved graft survival, and it has been suggested that transfusion is immunosuppressive in an as yet unidentified way. Jones et al., 1983. The authors had cited this empirical knowledge to explain immunosuppression in 8, and pneumocystis pneumonia in 6 British hemophiliacs. Jones et al., 1983. A multi-center study investigating the immune systems of 1,551 hemophiliacs, treated with factor VIII from 1975 to 1979, documented lymphocytopenia in 9.3% and thrombocytopenia in 5%, Aister et al., 1985. Further, the CDC reported AIDS-defining opportunistic infections in hemophiliacs between 1968 and 1979, including 60% pneumonias and 20% tuberculosis. Johnson et al., 1985. An American hematologist commented on such opportunistic infections in hemophiliacs, including 2 candidiasis and 66 pneumonia deaths that had occurred between 1968 and 1979. It seems possible that many of the unspecified pneumonias in hemophiliacs in the past would be classified today as AIDS. Aronson, 1983. In 1983, Gordon from the National Institutes of Health noted that all hemophiliacs with immunodeficiency identified by the CDC had received factor VIII concentrate. While acknowledging the possibility of a transmissible agent, Gordon argued that repeated administration of factor VIII concentrate from many varied donors induces a mild disorder of immune dysregulation by purely immunological means, without the intervention of infection. Gordon, 1983. Froebel et al. Also argued against the hypothesis that immunodeficiency in American hemophiliacs was due to a virus, and suggested that it was due to treatments with factor VIII because Scottish patients with hemophilia, most of whom had received no American factor VIII concentrate for over 2 years, were found to have immunological abnormalities similar to those in their American counterparts. Froebel et al., 1983. Already in 1983, Manitov et al. Described a correlation between immunosuppression of hemophiliacs and the amount of factor VIII received over a lifetime. The more factor a hemophiliac had received, the lower was his T4/T8 cell ratio. Their data were found to be consistent with the possibility that commercially prepared lyophilized factor VIII concentrates can induce an AIDS-like picture. Manitov et al., 1983. Also in 1983, Kessler et al. Proposed that repeated exposure to many blood products can be associated with development of T4/T8 abnormalities and significantly reduced mean T4/T8 ratios compared with age and sex-matched controls. Kessler et al., 1983. After the introduction of the HIV/AIDS hypothesis in 1984, Carr et al. Studied immunodeficiency in HIV-positive and HIV-negative hemophiliacs and proposed that the abnormalities low T4 to T8 cell ratios result from transfusion of foreign proteins. Carr et al., 1984. Likewise, Tsukas et al. Concluded these data suggest that another factor, or factors, instead of, or in addition to, exposure to HTLV3, old term for HIV, is required for the development of immune dysfunction in hemophiliacs. Tsukas et al., 1984. In 1985, even the retrovirologist Weiss reported the abnormal T lymphocyte subsets are a result of the intravenous infusion of factor VIII concentrates per SE, not HTLV3 infection. Ludlam et al., 1985. Likewise, the hematologists Pollock et al. Deduced that derangement of immune function in hemophiliacs results from transfusion of foreign proteins or a ubiquitous virus rather than contracting AIDS infectious agent. Pollock et al., 1985. The AIDS infectious agent was a reference to HIV, because in 1985 HIV was extremely rare in blood concentrates outside the US, but immunodeficiency was observed in Israeli, Scottish, and American hemophiliacs. Pollock et al., 1985. A French AIDS hemophilia group also observed, allogenic or altered proteins present in factor VIII, seem to play a role of immunocompromising agents. They stated that a correlation between treatment intensity and immunologic disturbances was found in patients infused with factor VIII preparations, irrespective of their positive or negative LAV HIV antibody status. AIDS Hemophilia French Study Group, 1985. Likewise, Holland et al. Reported in 1985 an immunodeficiency independent of HTLV3 infection in Hungarian hemophiliacs. Holland et al., 1985. In 1986, Madhawk et al. Arrived at the conclusion that clotting factor concentrate impairs the cell-mediated immune response to a new antigen in the absence of infection with HIV. Madhawk et al., 1986. Moreover, Jason et al. From the CDC observed that hemophiliacs with immune abnormalities may not necessarily be infected with HTLV3/LAV, since factor concentrate itself may be immune suppressive even when produced from a population of donors not at risk for AIDS. Jason et al., 1986. Sullivan et al. Deduced from a comprehensive study of hemophiliacs that hemophiliacs receiving commercial factor VIII concentrate experience several stepwise incremental insults to the immune system, alloantigens in factor VIII concentrate etc. Sullivan et al., 1986. In 1987, Sharp et al. Commented that 5 out of 12 such patients had a mild T4 lymphocytopenia, and this may have been related to parenteral administration of large quantities of protein. Sharp et al., 1987. And Aildert observed that chronic recipients of factor VIII, factor IX, and pooled products demonstrated significant T-cell abnormalities regardless of the presence of HIV antibody. Aildert, 1988. Bredler and Levine proposed in 1989 that factor concentrate itself, perhaps secondary to the large amount of foreign protein present, may cause alterations in the immune systems of hemophiliac patients. Bredler and Levine, 1989. And even Stergreen et al. From the CDC conceded that foreign proteins were at least a cofactor of HIV in immunosuppression. Repeated exposure to factor concentrate could also account for more rapid progression of HIV infection with age. Stergreen et al., 1989. Although Beckerer et al. Claimed in 1990 that clotting factor does not cause immunodeficiency, they showed that immunodeficiency in hemophiliacs increases with both the age and the cumulative dose of clotting factor received during a lifetime. Beckerer et al., 1990. Likewise, Simmons et al. Observed in 1991 that even among HIV-positive hemophiliacs the rate of disease progression, as assessed by the appearance or not of AIDS symptoms or signs within 5 years of seroconversion, was related to the concentration of total plasma IgM before exposure to infection. Simmons et al., 1991. The hematologist Prince noted in a review from 1992 that when serum samples from these immunodeficient hemophilia patients were tested for antibodies to HIV-1, it was found that a sizable group of hemophilia patients, usually 25% to 40%, were seronegative for HIV-1, and all found marked anergy, lack of response, in HIV seronegative concentrate recipients. Taken together, these findings were interpreted as evidence that clotting factor concentrates suppressed the immunocompetence of recipients. Prince, 1992. In 1991, Schulman concluded that immunosuppressive components in F8 concentrates cause immunodeficiency not only in HIV-positive but also in HIV-negative hemophiliacs. Schulman, 1991. Schulman had observed reversal of immunodeficiency and thrombocytopenia in HIV-positive hemophiliacs treated with purified factor VIII, and that immunity was inversely correlated with the annual amount of factor VIII infused. Schulman, 1991. At the same time, several groups have reported that T-cell counts are stabilized, or even increased in HIV-positive hemophiliacs treated with factor VIII free of foreign proteins. Debiase et al., 1991, Hilgertner et al., 1993, Sirmatis et al., 1993, Godard et al., 1994, see also section 4.7. And in 1994, the editor of AIDS News, published by the Hemophilia Council of California, granted foreign proteins the role of a cofactor of HIV in hemophilia AIDS with an editorial factor concentrate as a CO factor. Maynard, 1994. According to the foreign protein hypothesis, antibodies against HIV and against other microbes would merely be markers of the multiplicity of transfusions received. Evett et al., 1984, Pollock et al., 1985, Bredler et al., 1986, Sullivan et al., 1986, Koerper, 1989. Since HIV has been a rare contaminant of blood products, even before 1984, only those who have received many transfusions would become infected. The more immunosuppressive transfusions a person has received, the more likely that person is to become infected by HIV and other microbes contaminating factor VIII. See section 4.6. For example, only 30% of hemophiliacs who had received less than 400 units factor VIII per kg per year were HIV positive, but 80% of those who had received about 1,000 units, and 93% of those who had received over 2,100 units per kg per year were HIV positive. Sullivan et al., 1986. 4 predictions of the foreign protein and HIV/AIDS hypotheses. Here we compare the HIV and the foreign protein AIDS hypotheses in terms of how well their predictions can be reconciled with hemophilia AIDS. 4.1 Mortality of hemophiliacs with and without HIV. The virus AIDS hypothesis predicts that the mortality of HIV-positive hemophiliacs will be higher than that of matched HIV-free counterparts. Considering the high 75% rate of infection of American hemophiliacs by HIV since 1984, one would expect that the median age of all American hemophiliacs would have significantly decreased and that their mortality increased. The HIV/AIDS hypothesis predicts that in 1994, at least one 10-year latent period after most American hemophiliacs were infected, over 50% of the 15,000 HIV-positive American hemophiliacs would have developed AIDS or died from AIDS. Institute of Medicine, 1988, Duesberg, 1992a. But despite the many claims that HIV causes AIDS in hemophiliacs, Centers for Disease Control, 1986, Institute of Medicine, 1988, Weiss and Jaff, 1990, Korba et al., 1994, there is not a single controlled study showing that the morbidity or mortality of HIV-positive hemophiliacs is higher than that of HIV-negative controls matched for the lifetime consumption of factor VIII. Instead, the mortality of American hemophiliacs has decreased and their median age has increased since 75% were infected by HIV. The median age of American hemophiliacs has increased from 11 years in 1972, to 20 years in 1982, to 25 years in 1986, and to 27 years in 1987, although 75% had become HIV antibody positive prior to 1984, Institute of Medicine and National Academy of Sciences, 1986, Coerper, 1989, Stergreen et al., 1989. Likewise, their median age at death has increased from about 40 to 55 years in the period from 1968 to 1986, Corba et al., 1994. Contrary to the HIV/AIDS hypothesis, one could make a logical argument that HIV, instead of decreasing the lifespan of hemophiliacs, has in fact increased it. A more plausible argument suggests that the lifespan of American hemophiliacs has increased as a consequence of the widespread use of factor VIII that started in the late 1960s, see above. As predicted by the foreign protein hypothesis, the price for the extended lifespan of hemophiliacs by treatment with commercial factor VIII was immunosuppression due to the long-term parenteral administration of large quantities of foreign protein, see section 4.2. Prior to factor VIII therapy, most hemophiliacs died as adolescents from internal bleeding, Coerper, 1989. However, a recent CDC study reports that the mortality of American hemophiliacs suddenly increased 2.5-fold in the period from 1987 to 1989, after it had remained almost constant in the period from 1968 to 1986, Corba et al., 1994. Since American hemophiliacs became gradually infected via the introduction in the 1960s of pooled factor VIII treatments until 1984, when HIV was eliminated from the blood supply, see above, one would have expected first a gradual increase in hemophilia mortality and then a rather steep decrease. The increase in mortality would have followed the increase of infections with a lag defined by the time that HIV is thought to require to cause AIDS. The presumed lag between HIV and AIDS has been estimated at 10 months by the CDC in 1984, Auerbach et al., 1984, and at 10 years by a committee of HIV researchers, including some from the CDC, in 1988, Institute of Medicine, 1988. Therefore the sudden increase in hemophilia deaths in 1987 is not compatible with HIV-mediated mortality. Hemophilia mortality should have gradually decreased after 1984, when HIV was eliminated from the blood supply, depending on the lag period assumed between infection and AIDS. Even if the lag period from HIV to AIDS were 10 years, the mortality of hemophiliacs should have significantly decreased by 1989, 5 years after new infections had been stopped. An obvious explanation for the chronological inconsistency between infection of hemophiliacs with HIV since the 1960s and the sudden increase in their mortality 20 years later is the introduction of the cytotoxic DNA chain terminator AZT as an anti-HIV drug in 1987. AZT has been recommended and prescribed to symptomatic HIV carriers since 1987, Fiscal et al., 1987, Richmond et al., 1987, and to healthy HIV carriers with lower than 500 T cells since 1988, Valberding et al., 1990, Goldsmith et al., 1991, Phillips et al., 1994b. Approximately 200,000 HIV antibody positives with and without AIDS diseases are currently prescribed AZT worldwide, Duesberg, 1992a. According to a preliminary survey of hemophiliacs from a national group, Concerned Hemophiliacs Acting for Peer Strength, CHAPS, 35 out of 35 HIV-positive hemophiliacs asked had taken AZT, and 20 out of 35 who had taken AZT at some time were currently on AZT. Personal Communication, Brent Runyon, Executive Director of CHAPS, Wilmington, NC. The DNA chain terminator AZT was developed 30 years ago to kill growing human cells for cancer chemotherapy. Because of its intended toxicity, chemotherapy is typically applied for very limited periods of time, i.e. weeks or months, but AZT is now prescribed to healthy HIV positives indefinitely, despite its known toxicity. Nussbaum, 1990, Valberding et al., 1990. Indeed, AZT has been shown to be toxic in HIV positives and proposed as a possible cause of AIDS diseases since 1991, Duesberg, 1991, 1992c, 1992a, 1992b. Recently, the European Concord trial, Seligman et al., 1994, and several other studies have shown that, contrary to earlier claims, AZT does not prevent AIDS. Audun et al., 1993, Talkers et al., 1993, Lenderking et al., 1994, Lundgren et al., 1994. The Concord trial even showed that the mortality of healthy, AZT-treated HIV carriers was 25% higher than that of placebo-treated controls, Seligman et al., 1994. Likewise, an American multi-center study showed that the death risk of hemophiliacs treated with AZT was 2.4 times higher, and that their AIDS risk was even 4.5 times higher than that of untreated HIV-positive hemophiliacs, Godard et al., 1994. Thus, the widespread use of AZT in HIV positives could be the reason for the sudden increase in hemophilia mortality since 1987. The AZT hemophilia AIDS hypothesis and the foreign protein AIDS hypothesis both predict that hemophilia AIDS would stay constant or increase as long as unpurified factor VIII is used and AZT is prescribed to HIV-positive hemophiliacs. By contrast, the HIV-AIDS hypothesis predicts that hemophilia AIDS should have decreased with time since 1984 when HIV was eliminated from the blood supply. The HIV hypothesis further predicts that AIDS should have decreased precipitously since 1989 when AZT was prescribed as AIDS prevention to inhibit HIV. But the decrease in hemophilia AIDS predicted by the HIV-AIDS hypothesis was not observed. Instead, the data confirm the AZT/foreign protein AIDS hypotheses. The CDC reports 300 hemophilia AIDS cases in 1988, 295 in 1989, 320 in 1990, 316 in 1991, 316 in 1992 and, after broadening the AIDS definition as of January 1993, Centers for Disease Control and Prevention, 1992, 1096 in 1993, Centers for Disease Control, 1993, Centers for Disease Control and Prevention, 1994, and prior HIV/AIDS surveillance reports. 4.2 Annual AIDS risk of HIV-positive hemophiliacs compared to other HIV-positive AIDS risk groups. The HIV-AIDS hypothesis predicts that the annual risk of HIV-positive hemophiliacs would be the same as that of other HIV-infected risk groups. One could in fact argue that it should be higher, because the health of hemophiliacs is compromised compared to AIDS risk groups without congenital health deficiencies. By contrast, the foreign protein AIDS hypothesis makes no clear prediction about the annual AIDS risk of hemophiliacs compared to drug AIDS risk groups, because the relative risks have not been studied and are hard to quantitate. By the end of 1992, 2,214 American hemophiliacs with AIDS were reported to the CDC, Centers for Disease Control, 1993, Corba et al., 1994. Since there are about 15,000 HIV-positive American hemophiliacs, an average of only 1.3%, 201 out of 15,000, have developed AIDS annually between 1981 and 1992, Tsukas et al., 1984, Hardy et al., 1985, Institute of Medicine and National Academy of Sciences, 1986, Sullivan et al., 1986, Stergreen et al., 1988, Godard et al., 1989, Koerper, 1989, Morgan, Curran and Burke-Ellman, 1990, Gompertz, de Biasi and de Vreeker, 1992. But after the inclusion of further diseases into the AIDS Syndrome, Institute of Medicine, 1988, and the introduction of AZT as an anti-HIV drug, both in 1987, the annual AIDS risk of American hemophiliacs appears to have stabilized at 2%, e.g. about 300 out of 15,000 per year until 1993 when the AIDS definition was changed again. Centers for Disease Control, 1993, see section 4.1. Hemophilia AIDS statistics from Germany are compatible with American counterparts. About 50% of the 6,000 German hemophiliacs are HIV positive, Koerper, 1989. Only 37 or 1% of these developed AIDS-defining diseases during 1991, Leonhardt, 1992, and 186 or 1.5% annually during the 4 years from 1988 to 1991, Schwartlander et al., 1992. The 1.3% to 2% annual AIDS risk indicates that the average HIV-positive hemophiliac would have to wait for 25 to 35 years to develop AIDS diseases from HIV. Indeed, latent periods of over 20 years have just been calculated for HIV-positive hemophiliacs based on the loss of T cells over time, Phillips et al., 1994b. By contrast, the annual AIDS risk of the average HIV-positive American is currently 6%, because there are now about 60,000 annual AIDS cases, Centers for Disease Control, 1993, per 1 million HIV-positive Americans, Curran et al., 1985, Centers for Disease Control, 1992b, Duesberg, 1992a. This reflects the annual AIDS risks of the major risk groups, the male homosexuals and intravenous drug users who make up about 93% of all American AIDS patients, Centers for Disease Control, 1993. The annual AIDS risks of intravenous drug users, Lemp et al., 1990, and male homosexuals appear to be the same, as both were estimated at about 5 to 6%, Anderson and May, 1988, Louis et al., 1988, Lemp et al., 1990, Table 1. In view of the compromised health of hemophiliacs, it is surprising that the annual AIDS risk of HIV-infected hemophiliacs is only 1.3% to 2% and thus 3 to 5 times lower than that of the average HIV-infected, non-hemophiliac American or European, Table 1. Commenting on the relatively low annual AIDS risk of hemophiliacs compared to that of homosexuals, the hematologist Sullivan et al. Noted that the reasons for this difference remain unclear, Sullivan et al., 1986. Hardy et al. From the CDC also noted the discrepancy in the latent periods of different risk groups. The magnitude of some of the differences in rates is so great that even gross errors in denomination estimates can be overcome, Hardy et al. 1985. And Christine Lee, senior author of the study that had estimated latent periods of over 20 years from infection to hemophilia AIDS, Phillips et al., 1994b, commented on the paradox it may be that hemophiliacs have got that cofactor of foreign blood contaminants, homosexuals have got another cofactor, drug users have got another cofactor, and they all have the same effect, so that at the end of the day you get approximately the same progression rate. Jones, 1994. Thus, the 3-5-fold difference between the annual AIDS risks of HIV-positive hemophiliacs and the other major risk groups is not compatible with the HIV hypothesis. However, it can be reconciled with the foreign protein and drug AIDS hypothesis, Duesberg, 1992a, 1994, because different causes, i.e. drugs and foreign proteins, generate AIDS diseases at different rates. 4.3 The age bias of hemophilia AIDS. The HIV/AIDS hypothesis predicts that the annual AIDS risks of HIV-positive hemophiliacs is independent of their age, because virus replication is independent of the age of the host. Predictions would have to be adjusted, however, by the hypothetical lag period between infection and AIDS. If the average latent period from HIV to AIDS is 10 months, as was postulated in 1984, Auerbach et al., 1984, less than 10-month-old HIV-positive hemophiliacs would have a lower probability of having AIDS. If the average latent period from HIV to AIDS is 10 years, Institute of Medicine, 1988, LWE-ETAL, 1988, LEMP-ETAL, 1990, Weiss, 1993, HIV-positive hemophiliacs under 10 years of age would have a lower probability of having AIDS. In other words, if the time of infection is unknown, the annual AIDS risks of HIV-positive hemophiliacs over 10 months or 10 years, respectively, would be independent of the age of the HIV-positive hemophiliac. By contrast, the foreign protein hypothesis predicts that the annual AIDS risk of HIV-positive and negative hemophiliacs increases with age because immunosuppression is the result of the lifetime dose of proteins transfused. Pollock et.al., 1985, Bredler et.al., 1986, Sullivan et.al., 1986, Koerper, 1989, see above. The more years a hemophiliac has been treated with unpurified blood products, the more likely he is to develop immunodeficiency. Thus, the foreign protein hypothesis predicts that the annual AIDS risk of a hemophiliac would increase with age. Table 1. Annual AIDS risks of HIV-infected groups. American/European annual AIDS risk group in % references. Hemophiliacs 1.3 to 2 see text. Male homosexuals 5 to 6 LWE-ETAL 1988. Anderson and May, 1988, Lemp. ETAL 1990. Intravenous drug 5 to 6 LWE-ETAL 1988. Users Anderson and May, 1988, Lemp. ETAL, 1990. Statistics show that the median age of hemophiliacs with AIDS in the U.S., Evatt et.al., 1984, Koerper, 1989, Stergreen et.al., 1989, and other countries, Darby et.al., 1989, Bigger and the International Registry of Seroconverters, 1990, Blattner, 1991, is about 5 to 15 years higher than the average age of hemophiliacs. In the U.S., the average age of hemophiliacs was 20 to 27 years from 1980 to 1986, while that of hemophiliacs with AIDS was 32 to 35 years. Evatt et.al., 1984, Koerper, 1989, Stergreen et.al., 1989. Likewise, the annual AIDS risk of HIV-positive hemophiliacs shows a strong age bias. An international study estimated the annual AIDS risk of children at 1% and that of adult hemophiliacs at 3% over a 5-year period of HIV infection, Bigger and the International Registry of Seroconverters, 1990. In the U.S., Goddard et al. Reported that the annual AIDS risk of 1 to 17-year-old hemophiliacs was 1.5%, that of 18 and 34-year-old hemophiliacs was 3%, and that of 64-year-old hemophiliacs was 5%, Goddard et.al., 1989. Goldsmith et.al. Reported that the annual T-cell loss of hemophiliacs under 25 years was 9.5% and for hemophiliacs over 25 years 17.5%, Goldsmith et.al., 1991. Lee et.al. Reported that the annual AIDS risk of hemophiliacs 11 years after HIV seroconversion was 31% under 25 years and 56% over 25 years, Lee et.al., 1991. They estimated that the relative risk of AIDS increased 5-fold over 25 years. The same group confirmed in 1994 that the annual AIDS risk of HIV-positive hemophiliacs over 30 years is 2 times higher than in those under 15 years of age, Phillips et.al., 1994b. Stergreen et.al. estimated that the risk of AIDS increased 2-fold for each 10-year increase in age after controlling for year of seroconversion. Stergreen et.al., 1989. Likewise, Fletcher et.al. reported a 4-fold higher incidence of AIDS in hemophiliacs over 25 years of age than in those aged 5 to 13 years, Fletcher et.al., 1992. Thus, the annual AIDS risk of hemophiliacs increases about twofold for each 10-year increase in age. This confirms the foreign protein hypothesis, which holds that the cumulative dose of transfusions received is the cause of AIDS-defining diseases among hemophiliacs. According to the hematologist Koerper, this may reflect lifetime exposure to a greater number of units of concentrate. And to Evett et al., this age bias may be due to differences in duration of exposure to blood products. Evett et al., 1984, Koerper, 1989. A recent study of HIV-free hemophiliacs is directly compatible with the foreign protein hypothesis. The study showed that despite the absence of HIV with increasing age, numbers of CD4 plus CD45 RAN cells decreased and continued to do so throughout life. Fletcher et al., 1992. By contrast, AIDS caused by an autonomous infectious pathogen would be independent of the age of the recipient because the replication cycle of viruses, including HIV, is independent of the age of the host. Thus the foreign protein AIDS hypothesis, rather than the HIV/AIDS hypothesis, correctly predicts the age bias of hemophilia AIDS. 4.4 Hemophilia-specific AIDS diseases. The 30 AIDS diseases fall into two categories, the microbial immunodeficiency diseases and the non-immunodeficiency diseases, i.e. diseases that are neither caused by, nor consistently associated with, immunodeficiency. Duesberg, 1992a, 1994. Based on their annual incidence in America in 1992, 61% of the AIDS diseases were microbial immunodeficiency diseases, including pneumocystis pneumonia, candidiasis, tuberculosis, etc., and 39% were non-immunodeficiency diseases, including Kaposi's sarcoma, lymphoma, dementia, and wasting disease. Table 2, Centers for Disease Control, 1993. The virus AIDS hypothesis predicts that the probability of all HIV-infected persons to develop a given immunodeficiency or non-immunodeficiency AIDS disease is the same and independent of the AIDS risk group. By contrast, the hypothesis that AIDS is caused by drugs or by foreign proteins predicts specific diseases for specific causes. Duesberg, 1992a. In America, 99% of the hemophiliacs with AIDS have immunodeficiency diseases, of which 70% are fungal and viral pneumonias. Evett et al., 1984, Koerper, 1989, Papadopoulos Eliopoulos et al., 1994. Only one study reports that 1% of hemophiliacs with AIDS had Kaposi's sarcoma, Selleck, Starcher and Curran, 1987. The small percentage of Kaposi's sarcoma may be due to aphrodisiac nitrite inhalants used by male homosexual hemophiliacs as sexual stimulants. Haverkos and Doherty, 1988, Duesberg, 1992a. There are no reports of wasting disease or dementia in American hemophiliacs. An English study also reported predominantly pneumonias and other immunodeficiency diseases among hemophiliacs, and also 3 cases of wasting syndrome. Lee et al., 1991. It appears that the AIDS diseases of hemophiliacs are virtually all immunodeficiency diseases, whereas 39% of the AIDS diseases of intravenous drug users and male homosexuals are non-immunodeficiency diseases. Table 2. Since AIDS diseases in hemophiliacs and non-hemophiliacs are not the same, their causes can also not be the same. The almost exclusive occurrence of immunodeficiency AIDS diseases among hemophiliacs is correctly predicted by the foreign protein AIDS hypothesis, but not by the HIV/AIDS hypothesis. The prediction of the HIV hypothesis, that the distribution of immunodeficiency and non-immunodeficiency diseases among hemophiliacs is the same as in the rest of the American AIDS population, is not confirmed. 4.5 Is hemophilia AIDS contagious? The virus AIDS hypothesis predicts that AIDS is contagious, because HIV is a parenterally and sexually transmitted virus. It predicts that hemophilia AIDS is sexually transmissible. Indeed, AIDS researchers claim that the wives of hemophiliacs develop AIDS from sexual transmission of HIV. Booth, 1988, Lawrence et al., 1990, Weiss and Jaff, 1990, Centers for Disease Control, 1992a, 1993. Further, the HIV/AIDS hypothesis predicts that wives of hemophiliacs will develop the same AIDS diseases as other risk groups. The foreign protein hypothesis predicts that AIDS is not contagious and that the wives and sexual partners of hemophiliacs do not contract AIDS from their mates. To test the hypothesis that immunodeficiency of hemophiliacs is sexually transmissible, the T4 to T8 cell ratios of 41 spouses and female sexual partners of immunodeficient hemophiliacs were analyzed, Kreis et al., 1984. 22 of the females had relationships with hemophiliacs with T-cell ratios below 1, and 19 with hemophiliacs with ratios of 1 and greater. The mean duration of relationships was 10 years, the mean number of sexual contacts was 111 during the previous year, and only 12% had used condoms, Kreis et al., 1984. Since the T-cell ratios of all spouses were normal, averaging 1.68 exactly like those of 57 normal controls, the authors concluded that there is no evidence to date for heterosexual or household contact transmission of T-cell subset abnormalities from hemophiliacs to their spouses. Kreis et al., 1984. The CDC reports that between 1985 and 1992, 131 wives of American hemophiliacs were diagnosed with unnamed AIDS diseases, Centers for Disease Control, 1993. If one considers that there have been 15,000 HIV-positive hemophiliacs in the U.S. since 1984 and that one-third are married, then there are 5,000 wives of HLV-positive hemophiliacs. About 16 of these women have developed AIDS annually during the 8 years, 131, 8, from 1985 to 1992. But these 16 annual AIDS cases would have to be distinguished from the at least 80 wives of hemophiliacs that are expected to die per year based on natural mortality. Considering the human lifespan of about 80 years and that on average at least 1.6% of all those over 20 years of age die annually, about 80 out of 5,000 wives over 20 would die naturally per year. Thus, until controls show that among 5,000 HIV-positive wives of hemophiliacs 16 more than 80, i.e. 96, die annually, the claim that wives of hemophiliacs die from sexual or other transmission of HIV is unfounded speculation. Moreover, it has been pointed out that all AIDS-defining diseases of the wives of hemophiliacs are typically age-related opportunistic infections, including 81% pneumonia, Lawrence et al., 1990. Kaposi's sarcoma, dementia, lymphoma, and wasting syndrome are not observed in wives of hemophiliacs, Lawrence et al., 1990. Again, the foreign protein, but not the HIV hypothesis, correctly predicts the non-contagiousness of hemophilia ALDS. It also predicts the specific spectrum of AIDS diseases in wives of hemophiliacs. By contrast, the virus AIDS hypothesis predicts the same spectrum of AIDS diseases among wives of hemophiliacs as among the major risk groups, see Table 2. It appears that the virus AIDS hypothesis is claiming normal morbidity and mortality of the wives of hemophiliacs for HIV. 4.6 Immunodeficiency in HIV-positive and negative hemophiliacs. The HIV hypothesis predicts that immunodeficiency is observed only in HIV-positive hemophiliacs. By contrast, the foreign protein hypothesis predicts that immunodeficiency is a function of the lifetime dose of transfusions received, and not dependent on HIV or antibodies against HIV. The foreign protein hypothesis also predicts that HIV-positive hemophiliacs are more likely to be immunosuppressed than HIV negatives because HIV is a rare contaminant of blood transfusion and thus is a marker for the number of transfusions received, see Section 3, and below. Tsukas et al., 1984, Ludlam et al., 1985, Kreis et al., 1986, Sullivan et al., 1986, Koerper, 1989, Fletcher et al., 1992. 21 studies, summarized in Table 3, have observed 1,186 immunodeficient hemophiliacs, 416 of whom were HIV-free. Immunodeficiency in these studies was either defined by a T4 to T8 cell ratio of about 1 or less than 1, compared to a normal ratio of 2, or by other tests such as immunological anergy. Since immunodeficiency was observed in the absence of HIV, most of the studies listed in Table 3 have concluded that immunodeficiency in hemophiliacs was caused by transfusion of factor VIII and contaminating proteins. According to the first of Koch's postulates, Merriam-Webster, 1965, the absence of a microbe, i.e. HIV, from a disease excludes it as a possible cause of that disease. Thus, transfusion of foreign protein, not the presence of HIV, emerges as the common denominator of all hemophiliacs with immunodeficiency. Nevertheless, several of the controlled studies listed in Table 3, which compare HIV-negative to HIV-positive hemophiliacs, have shown that immunodeficiency is more often associated with HIV positives than with negatives. Although some studies did not report immunodeficiency in HIV positives, Table 3 lists 770 HIV positives and 416 HIV negatives per 1,186 immunodeficient hemophiliacs. In view of this, one could argue that HIV is one of several possible causes of immunodeficiency. However, some of the investigators listed in Table 3, Psukas et al., 1984, Ludlam et al., 1985, Kreis et al., 1986, Madhawk et al., 1986, Sullivan et al., 1986, and others who have not performed controlled studies, Koerper, 1989, have proposed that HIV is just a marker for the number of transfusions received. Section 3. As a rare contaminant of factor VIII, HIV has in fact been a marker for the number of transfusions received before it was eliminated from the blood supply in 1984, just like hepatitis virus infection was a marker of the number of transfusions received until it was eliminated from the blood supply earlier. Anonymous, 1984, Koerper, 1989. According to Kreis et al., seropositive hemophiliac subjects, on average, had been exposed to twice as much concentrate as seronegative S. Kreis et al., 1986. Sullivan et al. Also reported that seropositivity to LAV/HTLV3, HIV, was 70% for the hemophiliac population and varied directly with the amount of factor VIII received. See Section 3. Sullivan et al., 1986. More recently, Schulman reported that a high annual consumption of factor VIII concentrate predisposed to HIV seroconversion. Schulman, 1991, and Fletcher et al. Described a positive relationship between the amount of concentrate administered and anti-HIV prevalence rate. Fletcher et al., 1992. The chronology of studies investigating immunodeficiency in HIV-free hemophiliacs faithfully reflects the popularity of the HIV hypothesis. The more popular the HIV hypothesis became over time, the fewer studies investigated immunodeficiency in HIV-free hemophiliacs. Indeed, most of the controlled studies investigating the role of HIV in immunodeficiency of HIV-positive and matched HIV-negative hemophiliacs were conducted before the virus hypothesis became totally dominant in 1988, Institute of Medicine, 1988, namely between 1984 and 1988, Table 3. The studies by Jin, Cleveland, and Kaufman, and Lang et al., both dated 1989, and the studies by Beckerer et al. And by Jason et al., both dated 1990, all described data collected before 1988, Table 3. After 1988, the question whether HIV-free hemophiliacs developed immunodeficiency became increasingly unpopular. As a result, only a few studies have described immunodeficiency in HIV-free hemophiliacs. For example, Schulman reported worrisome evidence of similar immunological disturbances has been observed, albeit to a lesser degree, in anti-HIV negative hemophiliacs, and that immunodeficiency in hemophiliacs correlates more strongly with annual consumption of factor concentrates than with HIV status. Schulman, 1991. Fletcher et al. Published a median T4/T8 cell ratio of 1.4, with a low 10 percentile of 0.8, in a group of 154 HIV-free hemophiliacs, and also showed a steady decline of T-cell counts with treatment years, Fletcher et al., 1992. Likewise, Hassett et al. Reported that patients with hemophilia A without human immunodeficiency virus type 1, HIV-1, infection have lower CD4+ counts and CD4+/CD8+ ratios than controls, Hassett et al., 1993. The study observed an average T4/T8 cell ratio of 1.47 in a group of 307 HIV-free hemophiliacs, differing over 50 years in age, compared to an average of 1.85 in normal controls. Unlike others, Hassett et al. Attributed the lowered CD4+ counts to a hemophilia-related disorder rather than to foreign proteins, but like others they attributed increased CD8+ counts to treatment with commercial factor VIII. However, Fletcher et al.'s and Hassett et al.'s practice of averaging immunodeficiency markers of large numbers of people, differing over 50 years in age, obscures how far the immunity of the longest, and thus most treated cases had declined compared to cases which have received minimal treatments. Since the authors of these studies did not report the lifetime dosage of factor VIII treatments of HIV-free hemophiliacs, a correlation between foreign protein dosage and immunosuppression cannot be determined. On the contrary, averaging immunodeficiency parameters of newcomers and long-term treatment recipients obscures the relationship between the lifetime dosage of factor VIII and immunosuppression. Moreover, the CDC reported 7 HIV-free hemophiliacs with AIDS, Smith et al., 1993. This study was one of a package that proposed to set apart HIV-free AIDS from HIV-positive AIDS with the new term idiopathic CD4 lymphocytopenia. The goal of these studies was to save the virus AIDS hypothesis, despite the presence of HIV-free AIDS, Duesberg, 1993b, 1994, Fossey, 1993. Nevertheless, all of the 7 HIV-free hemophiliacs met one or more criteria of the CDC's clinical AIDS definition from 1993, Centers for Disease Control and Prevention, 1992, e.g. they all had less than 300 T cells per microliter, range from 88 to 296, and 3 also had AIDS-defining diseases such as herpes and thrombocytopenia, Smith et al., 1993. The occurrence of immunodeficiency in HIV-free hemophiliacs demonstrates most directly that long-term transfusion of foreign proteins contaminating factor VIII is sufficient to cause immunodeficiency in hemophiliacs. To prove the foreign protein hypothesis, it would be necessary to show that treatment of HIV-positive hemophiliacs with pure factor VIII does not cause immunodeficiency. It is shown below that this is actually the case. 4.7 Stabilization, even regeneration of immunity of HIV-positive hemophiliacs by treatment with pure factor VIII. Commercial preparations of factor VIII contain between 99% and 99.9% non-factor VIII proteins, Aster and Now, 1978, Bredler and Levine, 1989, Jerset et al., 1994, Manuxi et al., 1992, Sirmatis et al., 1993. The foreign protein hemophilia AIDS hypothesis predicts that long-term transfusion with commercial factor VIII would be immunosuppressive, because of the presence of contaminating proteins. Further, it predicts that pure factor VIII, containing 100 to 1,000 times less foreign protein per functional unit, may not be immunosuppressive. Several studies have recently tested whether the impurities of factor VIII or factor VIII by itself are immunosuppressive in HIV-positive hemophiliacs. Debiase et al. Showed that over a period of 2 years the average T-cell counts of 10 HIV-positive hemophiliacs treated with non-purified, commercial factor VIII declined twofold, while those of matched HIV-positive controls treated with pure factor VIII remained unchanged. Moreover, 4 out of 6 anergic HIV-positive patients treated with purified factor VIII recovered immunological activity, Debiase et al., 1991. Goldsmith et al. Also found that the T-cell counts of 13 hemophiliacs treated with purified factor VIII remained stable for 1.5 years, Goldsmith et al., 1991. Sirmatis et al. Have confirmed and extended Duesberg et al.'s conclusion by establishing that the T cells of HIV-positive hemophiliacs were not depleted after treatment with pure factor VIII for 3 years, Sirmatis et al., 1993. Indeed, the T-cell counts of 14 out of 31 HIV-positive hemophiliacs increased up to 25% over the 3-year period of treatment with purified factor VIII despite infection by HIV. By contrast, in the group treated with unpurified factor VIII, the percentage of those with less than 200 T cells per ml increased from 7% at the beginning of the study to 47% at the end. Likewise, Hilgertner et al. Reported individual increases of T-cell counts of up to 50% in a group of 36 HIV-positive hemophiliacs treated with purified factor VIII whose average T-cell count had declined 1% during 6 months, Hilgertner et al., 1993. Godard et al. Have also reported that T-cell counts fell less rapidly with high-purity products, Godard et al., 1994. Moreover, Schulman observed that 4 HIV-positive hemophiliacs recovered from thrombocytopenia upon treatment with pure factor VIII for 2 to 3 years, and others from CD8-related immunodeficiency upon treatment for 6 months, Schulman, 1991. However, despite the evidence that purified factor VIII is beneficial in maintaining or even increasing T-cell counts, several studies testing purified factor VIII are ambiguous about its effectiveness in preventing or treating AIDS, Goldsmith et al., 1991, Hilgertner et al., 1993, Jerset et al., 1994, Godard et al., 1994, Phillips et al., 1994a. Some of these studies have only tested partially purified, i.e. 2 to 10 units/mg, instead of highly purified, i.e. 2000 to 3000 units/mg, factor VIII, Jerset et al., 1994. But each of the studies that are ambiguous about the benefits have also treated their patients with toxic antiviral DNA chain terminators like AZT. Indeed, the study by Duesberg et al. Was the only one that has tested purified factor VIII in the absence of AZT. The study by Sirmatis et al. Initially called for no AZT, but later allowed it anyway. Thus, in all but one study, the potential benefits of highly purified factor VIII have been obscured by the toxicity of AZT, see section 5.4. It is concluded that treatment of HIV-positive hemophiliacs with pure factor VIII provides lasting stabilization of immunity, and even allows regeneration of lost immunity. It follows that foreign proteins, rather than factor VIII or HIV, cause immunosuppression in HIV-positive hemophiliacs. 5 Conclusions and discussion. 4 criteria of proof have been applied to distinguish between the virus and the foreign protein hypothesis of hemophilia AIDS. I. Correlation. 2. Function, Koch's third postulate. 3. Predictions. 4. Therapy and prevention. Each of these criteria proved the foreign protein hypothesis valid and the HIV hypothesis invalid. 5.1 Correlations between hemophilia AIDS and the long-term administration of foreign proteins or HIV. Although correlation is not sufficient, it is necessary to prove causation in terms of Koch's postulates, Merriam-Webster, 1965. The first of Koch's postulates calls for the presence of the suspected cause in all cases of the disease, i.e. a perfect correlation, the second calls for the isolation of the cause, and the third for causation of the disease with the isolated causative agent. All hemophiliacs with immunodeficiency described here have been subject to long-term treatment with foreign proteins contaminating factor VIII. This establishes a perfect correlation between foreign protein transfusion and hemophilia AIDS, and fulfills Koch's first postulate. By contrast, a summary of 21 separate studies showed that 416 of 1,186 immunodeficient hemophiliacs were HIV-free, Table 3. Since HIV does not correlate well with hemophilia AIDS, it fails Koch's first postulate and is thus not even a plausible cause of AIDS. 5.2 Foreign protein hypothesis, but not HIV hypothesis, meets Koch's third postulate as cause of immunodeficiency. The fact that all hemophiliacs with immunodeficiency had been subject to long-term treatment with foreign proteins, and that factor VIII treatment in the absence of foreign proteins does not cause immune suppression, and may even revert it, provides functional proof for the foreign protein hypothesis. Thus, the foreign protein hypothesis meets Koch's third postulate of causation. Regeneration of immunity of HIV positives by treatment with pure factor VIII further indicates that HIV by itself or in combination with factor VIII is not sufficient for hemophilia AIDS. Therefore, HIV fails Koch's third postulate as a cause of AIDS. 5.3 Foreign protein hypothesis correctly predicts hemophilia AIDS and resolves paradoxa of HIV hypothesis. The ability to make verifiable predictions is the hallmark of a correct scientific hypothesis. Application of the two competing hypotheses to hemophilia AIDS proved that the foreign protein hypothesis, but not the HIV hypothesis, correctly predicts 7 characteristics of hemophilia AIDS, see sections 4.1 to 4.7. 1. The increased lifespan of American hemophiliacs, despite infection of 75% by HIV, due to factor VIII treatment, that extended their lives and disseminated harmless HIV. 2. The 3 to 5 times lower annual AIDS risk of hemophiliacs, compared to other AIDS risk groups. 3. The age bias of the annual AIDS risk of hemophiliacs, increasing twofold for each 10-year increase in age. 4. The restriction of hemophilia AIDS to immunodeficiency-related AIDS diseases, setting it apart from the spectrum of AIDS diseases in other risk groups. 5. The non-contagiousness of hemophilia AIDS, i.e. the absence of AIDS diseases above their normal background in sexual partners of hemophiliacs. 6. The occurrence of immunodeficiency in HIV-free, factor VIII-treated hemophiliacs. 7. The stabilization, even regeneration, of immunity of HIV-positive hemophiliacs upon long-term treatment with pure factor VIII. It follows that the foreign protein hypothesis, but not the HIV hypothesis, correctly predicts hemophilia AIDS. In addition, the foreign protein hypothesis resolves all remaining paradoxa of the HIV hypothesis, see section 2. 1. The failure of HIV neutralizing antibody to protect against AIDS because HIV is not the cause of AIDS. 2. The non-correlation between the loss of T cells and HIV activity because foreign proteins rather than HIV are immunotoxic. 3. The failure of HIV to kill T cells because T cell synthesis is suppressed by immunotoxic foreign proteins. 4. The latent periods of 10 to 35 years between HIV and hemophilia AIDS because the lifetime dosage of foreign proteins, not HIV, causes AIDS. 5.4 Treatment and prevention of AIDS. The prevention or cure of a disease, by eliminating or blocking the suspected cause, provides empirical proof of causation. Drug treatment based on HIV hypothesis. On the basis of the HIV hypothesis, AIDS has been treated since 1987 with anti-HIV drugs, such as the DNA chain terminators AZT, DDI, etc., Duesberg, 1992a. The rationale of the AZT treatment is to prevent HIV DNA synthesis at the high cost of inhibiting cellular DNA synthesis, the original target of AZT cancer chemotherapy, see above. However, not a single AIDS patient has ever been cured with AZT. Since 1989, healthy HIV-positive hemophiliacs have also been treated with DNA chain terminators in efforts to prevent AIDS. But the alleged ability of AZT to prevent AIDS has recently been discredited by several large clinical trials: Audun et al., 1993, Talkers et al., 1993, Godard et al., 1994, Lenderking et al., 1994, Lundgren et al., 1994, Seligman et al., 1994. Moreover, all studies of AZT treatments have confirmed the unavoidable cytotoxicity of DNA chain terminators. Duesberg, 1992, Audun et al., 1993, Talkers et al., 1993, Lenderking et al., 1994, Lundgren et al., 1994, Seligman et al., 1994. One study observed a 25% increased mortality, Seligman et al., 1994, and another a 4.5-fold higher annual AIDS risk and a 2.4-fold higher annual death risk in AZT-treated HIV-positive hemophiliacs compared to untreated controls, Godard et al., 1994. The failure of AZT therapy to cure or prevent AIDS indicates either that the drug is not sufficient to inhibit HIV or that HIV is not the cause of AIDS. The lower mortality and much lower incidence of AIDS-defining diseases among hemophiliacs not treated with AZT compared to those treated indicates that AZT causes AIDS-defining diseases and mortality. Thus, there is currently no rational or empirical justification for AZT treatment of HIV positives with or without AIDS. The apparent ability of AZT to cause AIDS-defining and other diseases in hemophiliacs is just one aspect of the many roles that drugs play in the origin of AIDS, see footnote. 2. Treatment based on foreign protein hypothesis. In the light of the foreign protein hypothesis, hemophiliacs have been treated with factor VIII freed of foreign proteins. This treatment has provided lasting stabilization of immunity in HIV-positive hemophiliacs. Moreover, the long-term treatment of immunodeficient, HIV-positive hemophiliacs with purified factor VIII has even regenerated lost immunity. Immunological energy has disappeared and the T cells in HIV-positive hemophiliacs have increased up to 25% in the presence of pure factor VIII, see section 4.7, Duesberg et al., 1991, Sirmatis et al., 1993. Thus, therapeutic benefits including AIDS prevention and even recovery of lost immunity by omission of foreign proteins from factor VIII lend credence to the foreign protein AIDS hypothesis. 3. Two treatment hypotheses and one treatment dilemma. The failure to distinguish between two alternative hypothetical AIDS causes, HIV and foreign proteins, has created a dilemma for contemporary hemophilia treatment. For example, Godard et al. Acknowledge that CD4 count fell less rapidly with high-purity products, Godard et al., 1994. But since they are also treating their patients with toxic AZT, see section 4.1, they observe that F8-related changes in CD4 concentration may have little relevance to clinical disease, Godard et al., 1994. Indeed, the group had published a rare comparison between the annual AIDS and death risks of hemophiliacs treated and not treated with AZT, which indicated that the AIDS risk of AZT-treated hemophiliacs is 4.5 times and the death risk 2.4 times higher than in untreated controls. In order to reconcile the apparent benefits of purified factor VIII on T-cell counts with the apparent toxicity of simultaneous AZT treatment, they try to separate T-cell loss from AIDS diseases. However, despite non-immunodeficiency AIDS diseases, see Table 2, Section 4.4, AIDS is defined as a T-cell deficiency, Institute of Medicine and National Academy of Sciences, 1986, Institute of Medicine, 1988, and dozens of AIDS researchers have observed that AIDS tends to develop only after patients' CD4 lymphocyte counts have reached low levels. Phillips et al., 1994b. Indeed, as of January 1993, the CDC defined less than 200 T cells per ml as an AIDS disease, Centers for Disease Control and Prevention, 1992, and sequential T cell counts of hemophiliacs are used as a basis to calculate their long-term survival, Phillips et al., 1994b. Because of their exclusive faith in the HIV/AIDS hypothesis, readers of the study by Sirmatis et al., Sirmatis et al., 1993, which had demonstrated that foreign proteins associated with factor VIII suppress T-cell counts, have even proposed to consider the use of high-purity factor VIII concentrates in non-hemophiliac HIV-positive patients as a treatment for other AIDS patients, i.e. intravenous drug users and homosexuals. Since hemophiliacs treated with pure factor VIII did either not develop immunodeficiency or even recovered lost immunity, they assumed, in view of the HIV hypothesis, that pure factor VIII must inhibit HIV and thus would help all AIDS patients, Swartz et al., 1994. The solution to the treatment dilemma can only come from treatments that are each based only on one hemophilia AIDS hypothesis. To test the foreign protein hypothesis, two groups of hemophiliacs must be compared that are matched for their lifetime dosage of factor VIII, for their percentage of HIV positives, for their percentage and dosage of prior AZT treatment, if applicable, and for their age. All AIDS-defining diseases must be diagnosed in each group clinically for the duration of the test. No anti-HIV treatments must be performed. One group would be treated with purified factor VIII, the other with commercial factor VIII contaminated with foreign proteins. To test the HIV/AIDS hypothesis, two groups of hemophiliacs must be compared that are matched for their lifetime dosage of factor VIII treatment and their age. The two groups must differ only in the presence of antibody against HIV. Both groups would be treated with the same factor VIII preparation. Only the HIV-positive group would receive AZT. All compensatory treatments of AZT recipients, e.g. blood transfusions to treat for AZT-induced anemia, neutropenia, or pancytopenia, Richmond et al., 1987, Valberding et al., 1990, Duesberg, 1992, would have to be recorded. During the duration of the test, all AIDS-defining diseases would each be recorded clinically in both groups. The outcome of each treatment strategy, purified factor VIII or AZT, would be determined based on morbidity and mortality, including AZT morbidity and mortality, and corrected for treatments compensating for AZT toxicity. As yet, no controlled treatment studies based on a single AIDS hypothesis have been performed. Nevertheless, the study by Debiassi et al., 1992, and with reservations that by Sirmatis et al., 1993, come close to the stated criteria for a test of the foreign protein hypothesis, section 4.7. Sirmatis et al. Initially excluded, but later allowed AZT treatment. Both studies showed that purified factor VIII improved immunodeficiency, C2. However, since all subjects in these studies were HIV positive, one could indeed argue that the improvement of those treated with purified factor VIII was due to a cooperation between HIV and purified factor VIII. The definitive treatment of immunodeficiency in hemophiliacs, or of hemophilia AIDS, could be only as far away as the duration of one carefully controlled treatment test. End of article. The toxicity of azidothymidine, AZT, on human and animal cells in culture at concentrations used for antiviral therapy. David T. Chu and Peter H. Duesberg. Department of Molecular and Cell Biology, Stanley Hall, University of California at Berkeley, Berkeley, California, 94720 USA. Abstract. AZT, a chain terminator of DNA synthesis originally developed for chemotherapy, is now prescribed as an anti-human immunodeficiency virus, HIV, drug at 500 to 1500 mg/person/day, which corresponds to 20 to 60 mAZT. The human dosage is based on a study by the manufacturer of the drug and their collaborators, which reported in 1986 that the inhibitory dose for HIV replication was 0.05 to 0.5 mAZT and that for human T cells was 2000 to 20.000 times higher, i.e. 1000 mAZT. This suggested that HIV could be safely inhibited in humans at 20 to 60 mAZT. However, after the licensing of AZT as an anti-HIV drug, several independent studies reported 20 to 1000-fold lower inhibitory doses of AZT for human and animal cells than did the manufacturer's study, ranging from 1 to 50 m. In accord with this, life-threatening toxic effects were reported in humans treated with AZT at 20 to 60 m. Therefore, we have re-examined the growth inhibitory doses of AZT for the human SEM T cell line and several other human and animal cells. It was found that at 10 m and 25 m AZT, all cells are inhibited at least 50% after 6 to 12 days, and between 20 to 100% after 38 to 48 days. Unexpectedly, variants of all cell types emerged over time that were partially resistant to AZT. It is concluded that AZT, at the dosage prescribed as an anti-HIV drug, is highly toxic to human cells. Introduction. AZT, 3-azido-3-deoxythymidine, is an analog of thymidine in which the 3-hydroxyl group is replaced by an azido group. This prevents the extension of a growing DNA strand ending with AZT to the 5' end of another nucleotide triphosphate. Thus AZT functions as a chain terminator of DNA synthesis. AZT was originally designed in the 1960s to be used as chemotherapy for leukemia. Horwitz, Chua and Noel, 1964. The rationale for cancer chemotherapy is to kill cancer cells during mitosis with cytotoxic chemicals like AZT. Because chemicals cannot distinguish cancer cells from normal cells, the price for chemotherapy is the death of normal cells that are in mitosis. Therefore, chemotherapy must be restricted to days or weeks. Successful chemotherapy kills the cancer before it kills the host. Since 1987, chronic administration of AZT and similar nucleoside analogues, like DDC and DDI, have been prescribed to AIDS patients to inhibit human immunodeficiency virus, HIV, the presumed cause of AIDS. Fiscal et al., 1987, Richmond et al., 1987, Yarchone et al., 1991. Since 1990, AZT has also been prescribed to healthy HIV antibody-positive persons to prevent AIDS. Valberding et al., 1990, Talkers et al., 1993, Seligman et al., 1994. The rationale is to inhibit HIV DNA synthesis at doses that do not inhibit cell DNA synthesis. Yarchone et al., 1991. It is claimed by Burroughs Wellcome, the manufacturer of AZT, and its collaborators that this can be achieved, because AZT would inhibit DNA synthesis with HIV DNA polymerase in vitro 100 times more effectively than DNA synthesis with cellular DNA polymerase. Furman et al., 1986. Moreover, this study claimed that in vivo AZT was 2,000 to 20,000 times more inhibitory to HIV replication, i.e. at 0.05 to 0.5/1 m, than to cell division, i.e. at 1000 m. Furman et al., 1986. Accordingly, anti-HIV doses of AZT were chosen to fall into this therapeutic window, e.g. to be 500 to 1500 mg per person per day, or about 20 to 60 m per kg per day. Furman et al., 1986, Fiscal et al., 1987, Valberding et al., 1990, Physician's Desk Reference, 1994. However, in view of its inherent cytotoxicity, AZT has been questioned as an acceptable anti-HIV drug on 3 theoretical grounds. Duesberg, 1992. Even if AZT were to inhibit HIV DNA synthesis 100 times more than cell DNA synthesis, it could not selectively inhibit HIV, as is claimed by the manufacturer. Furman et al., 1986. Since HIV DNA measures only 10 kb and cell DNA measures 106 kb, and since both DNAs are made in vivo simultaneously inside the same cell, cell DNA provides a 105-fold bigger DNA target for AZT toxicity than does HIV DNA. Therefore, the 100-fold higher selectivity of AZT claimed for HIV DNA synthesis is immaterial. 2. Inhibition of HIV DNA synthesis in HIV antibody-positive persons is completely unnecessary, because HIV does not spread in TTE presence of antiviral antibody. Duesberg, 1992. It is for this reason that only about 1 in 1,000 T cells is ever infected in HIV antibody-positive persons. Duesberg, 1992. The fact that only about 0.1% of all susceptible T cells are ever infected by HIV in HIV-positive persons with and without AIDS proves that HIV is very effectively neutralized by antiviral immunity. Moreover, there is no correlation between the number of HIV-infected cells and AIDS. Duesberg, 1993, Piatok et al., 1993. For example, there are healthy HIV-positive persons who have 30 to 40 times more HIV-infected cells than AIDS patients. Simmons et al., 1990, Bagasra et al., 1992, Duesberg, 1992. 3. Since only about 1 in LOOO T cells are ever infected by HIV in persons with or without AIDS, Duesberg, 1992, 1992, AZT must kill 999 uninfected cells in order to kill just one HIV-infected cell, a very poor pharmacological index. Thus theory predicts that AZT cannot selectively restrict HIV replication in vivo. AZT can only inhibit HIV by killing infected and uninfected target cells. Theory further predicts that AZT is unacceptable as anti-HIV therapy in HIV antibody-positive persons, because it will kill 999 uninfected cells for every infected cell. In response to these theoretical considerations, it is argued by the manufacturer of AZT and its collaborators that, contrary to expectations, AZT is an effective anti-HIV drug, because cell division was observed to be 2,000 to 20,000 times more drug-resistant than HIV replication. Furman et al., 1986. However, after AZT had been licensed for human use, several independent studies reported that the drug is about 20 to 1,000 times more toxic to human cells in culture than the manufacturer had claimed, i.e. that the half-inhibitory doses, ID 50, ranged between 1 and 50 m, Table 1. In accordance with these results, life-threatening toxicity including anemia, leukopenia, nausea, muscle atrophy, dementia, hepatitis, and mortality, has been documented in humans treated with 20 to 60 m AZT, Mir and Costello, 1988, Duesberg, 1992, Fryman et al., 1993, Talkers et al., 1993, Beischeler et al., 1994, Goodard et al., 1994, Seligman et al., 1994. If these results were correct, both the dosage of AZT prescribed to humans and the advisability of AZT as an anti-HIV drug need to be reconsidered. In view of up to a 1,000-fold discrepancy between the cytotoxicity of AZT reported by the manufacturer and his collaborators, Furman et al., 1986, and the cytotoxicity as reported by other investigators, Table 1, we set out to redetermine the cytotoxicity of AZT. We have investigated the effects of AZT on the human SEM T cell line and on several other human and animal cells in culture. In contrast to the previous studies, that measured toxicity over 1 to 3 rounds of mitoses, we decided to measure long-term toxicity over several weeks, representing up to 24 consecutive cell divisions. We reasoned that this experimental design would more closely mimic human exposure, which is indefinite, extending over numerous mitoses. Fiscal et al., 1987, Valberding et al., 1990, Physician's Desk Reference, 1994. Under the conditions AZT is prescribed as an anti-HIV drug, i.e. chronic application, it could indeed be more toxic than it is after only one or a few mitoses studied earlier, because non-lethal mutations would accumulate in surviving cells. Our experimental design would detect cumulative mutational toxicity acquired over several mitoses, in addition to the complete cytotoxicity observed in one or a few mitoses. Materials and methods. Materials. RPMI 1640 medium, Delbecco's modified eaglets medium, and fetal bovine serum were purchased from Gibco Laboratories, Grand Island, New York. Serum Plus was purchased from JRH Biosciences, Lenexa, KS. AZT was purchased from Sigma Chemical Co., St. Louis, MO. Study. Cell type. ID 50 at MAZT. Furman et al., 1986. Human T cell, line H9. 1000. Balzerini, Herduigen and de Klerk, 1989. Human T cell, line SEM. 1000. Mansurai et al., 1990. Human T cell, line SEM. 54. Lemaitre et al., 1990. Human Cell, Line Sim. 36. Avramis et al., 1989. Human T Cell, Line Sim. 4. Samadasi et al., 1990. Human Bone Marrow. 1. Human Bone Marrow. 5. Inu et al., 1989. Human Bone Marrow. 5. Human Bone Marrow. 25. Mansurai et al., 1990. Mouse Bone Marrow. 1.5. Gagu, Beckman and Agrawal, 1989. Mouse Bone Marrow. 2. Mouse Fetal Liver. 1. Table 1. 50% inhibitory dose of AZT for human and animal cells as reported by various laboratories. Culture conditions of cells grown in suspension. The SEM human lymphoid T-cell line was provided by Robert F. Gary, Tulane University School of Medicine, New Orleans. LA SEM T-cells were suspended in 5 ml of RPMI 1640 medium enriched with 10% serum plus in tissue culture flasks, 25 square centimeters growth area, Falcon, and were propagated at 37 degrees Celsius in humidified air with 6.5% CO2. The SEM T cells were maintained at a density around 3x105 cells per ml by diluting them 1:2 every other day. The medium was changed every day by spinning down the cells for 5 minutes in a clinical centrifuge at 6000 rpm and then resuspending them in fresh medium. AZT was added twice every day at 10 and 25 m concentrations by micropipettes with sterile tips. AZT additions were made at about 12H intervals. A control flask of SEM T cells was passaged identically without the addition of AZT. A 10/1L aliquot of evenly distributed cells was counted every other day with a hematocytometer. Culture conditions of cells grown attached to Petri dishes. The C3H mouse fibroblast cell line, the HS27 human foreskin cell line and the WI38 human lung cell line were purchased from the American Type Culture Collection. The secondary Chinese hamster lung cells were prepared from animals in our lab. Each of these cell types was cultured while attached to Petri dishes, 100 x 20 mm, Falcon, in 10 ml of Dulbicot's mod Ifeed Eagles medium enriched with 10% fetal bovine serum at 37 degrees Celsius in humidified air with 6.5% CO2. Each of the monolayer cell types was seeded of approximate EIX 105 cells on a 10 cm dish containing 10 ml of medium. The medium in each dish was changed every day. AZT additions were also made twice a day at 10 and 25 m concentrations. The cells were counted with a Coulter counter by placing a 200 pl sample of evenly distributed cells in 10 ml of isotonic buffered saline solution. Each AZT-treated culture was split 1:5 when the control dish had reached 100% confluency. Fig. 1. The effect of AZT, at 10 m and 25 m, on the growth rate of the human SEM T cell line maintained as described in the text. Results. The effect of long-term AZT treatment on the viability of the human SEM T cell line. To determine the cytotoxicity of AZT on the human SEM T cell line in culture, parallel cultures were incubated with 10 m, 25 m AZT and without AZT. See materials and methods. The untreated cells were maintained at saturation density of SEM cells, which is about 3x105 cells per ml in our conditions. Each culture was divided twofold every 48H, by which time the AZT-free control had regained saturation density. As can be seen in Fig. 1, after 4 days the cell count of the culture at 25 m AZT had been reduced to half of the control, and that of the culture at 10 m AZT to 2/3 of the control. After 12 days the cell densities of both AZT-treated cultures had been reduced to 1/3 of the control culture. From then on, the density of the culture at 25 m AZT continued to decline at a decreasing rate, and that of the culture at 10 m AZT stabilized. Fig. 1. One possible explanation of the decreasing sensitivity of surviving stem cells to AZT over time is that the dividing portion of the cells takes up all AZT in a short time, and that the resting portion of cells subsequently enters mitosis in a culture depleted of AZT. Another explanation suggests that variants are selected that do not incorporate AZT into DNA. To distinguish between these possibilities, each AZT-treated culture was further divided into two. One of the two subcultures was maintained with daily medium changes containing 10 and 25 mAZT respectively as before. The other subculture was supplemented, 12 hours after the medium including AZT had been changed, with the equivalent of an extra 10 and 25 mAZT respectively. All cultures were further incubated under these conditions for another 32 to 36 days. It can be seen in Fig I that even at 2 daily applications of AZT at 10 m, a decreasing fraction of T cells retained viability for 14 days, when the culture became contaminated. However, no survivors were observed after 14 days at 2 daily applications of 25 mAZT. It is concluded that T cell variants are selected, on long-term exposure to AZT, that are relatively resistant to AZT compared to the average T cell prior to treatment. The effect of long-term exposure to AZT on the viability of human and animal fibroblasts. To determine whether other human and animal cells are similar to human T cells with regard to AZT sensitivity, the viability of a human lung, WI, and foreskin, HS, cell line, of a mouse cell line, C3H, and of secondary Chinese hamster cells, CH, was studied in AZT. Each of the different cell types was seeded at 1x105 cells per 10 cm dish and exposed to AZT at 10 and 25 m. See materials and methods. AZT was added to each dish twice every day, once in the morning and again at night as described above. The inhibition of cell growth was expressed as the percentage of cells in the AZT culture compared to that of the untreated control. The cells were counted by the time the control had reached confluency, Fig 2. The first count of cells was taken at the end of 2 weeks when all control dishes had become completely confluent. Thereafter control cells were split 1:4 and allowed to reach confluency again. This process was repeated several times as shown in Fig 2. As can be seen in Fig 2, the general pattern of AZT sensitivity observed with T cells was confirmed with other human and animal cells. C3H mouse cells appeared to be most sensitive to the effects of AZT. At day 14, the densities of C3H cells, maintained at both concentrations of AZT, had already declined to below 50% of the control. Possibly due to a counting error, the density of C3H cells at 10 mAZT appeared lower than that of cells at 25 mAZT. After the same time, the concentrations of HS27, WI38, and CH cells ranged from 50 to 60% of the control at 25 mAZT, and from 60 to 70% of the control at 10 mAZT. From 14 to 38 days of AZT treatment, all fibroblast cells remained at about half the density of the controls. However, the densities of C3H and HS27 cell lines gradually increased over time at both concentrations of AZT. By day 38, the density of HS27 cells at both AZT concentrations had reached up to 80% of the control. Fig 2. The effect of AZT, at 10 m and 25 m, on the growth of human lung, WL, and foreskin cells, HS, on mouse fibroblasts, C3H, and on secondary Chinese hamster, CH, fibroblasts. AZT-treated cells were counted whenever the untreated control culture had reached confluency. Discussion. I. AZT toxic to human cells in the micromolar range. Our results indicate that long-term exposure to AZT inhibits the growth of human SEM T cells about 50% at 10 m, and gradually up to 100% at 25 m. Similar results were obtained with human lung and foreskin cells, and also with mouse and Chinese hamster cells, although complete inhibition was not observed with any of these cells under our conditions. Thus our results confirm and extend those of others summarized in Table 1, that AZT is toxic to human cells in the micromolar range. Indeed, AZT, like all other nucleotide analogues of DNA, is expected to be toxic in the micromolar range, because the Michaelis constants of authentic nucleotide triphosphates are also in the micromolar range. Kornberg, 1980. These results are incompatible with the claim of the manufacturer and its collaborators that AZT is only toxic to human cells in the millimolar range. That claim is also hard to reconcile with the manufacturer's own observation that HIV replication is inhibited by AZT at 0.05 to 0.5 m, Furman et al., 1986. Since HIV and cell DNA are both replicated in vivo inside the same cellular vesicle and at the same time, Rubin and Temin, 1958, Weiss et al., 1985, 2. Retroviral and cellular DNA synthesis depend on the same triphosphate pools, and 3. Retroviral DNA is a 105-fold smaller target for AZT than cell DNA. HIV DNA synthesis cannot be more sensitive to AZT than cell DNA. In fact, target theory predicts the opposite. Thus, the preponderance of evidence casts doubts on the claim of the manufacturer of AZT and its collaborators that AZT is only toxic to cells in the millimolar range. Furman et al., 1986. 2. Resistance of human and animal cells to long-term exposure to AZT. Unexpectedly, partially AZT-resistant variants emerged from human T cells and all other cells tested on continued exposure to AZT at 10 to 25 m for 38 to 48 days. These variants did not reach the densities of untreated control cells, but continued to divide in the presence of AZT at various rates. Further work is needed to analyze the basis for the relative AZT resistance acquired by human and animal cells upon long-term exposure to AZT. 3. Toxicity of AZT at micromolar concentrations calls for reappraisal of its use as an anti-HIV drug. The cell culture results described by us and others predict that AZT is toxic to humans at the 20 to 60 micromolar level, the concentrations at which it is prescribed as an anti-HIV drug. Even though our results show that human and animal cells acquire some resistance against AZT upon long-term exposure, no cell has achieved complete resistance to AZT under the conditions tested. This prediction is confirmed by numerous clinical studies that describe life-threatening toxic effects in humans treated with AZT at 20 to 60 m. See Introduction. Thus our data and those of others call into question the merits of AZT as an anti-HIV drug, particularly at the doses currently prescribed to humans. End of article. How much longer can we afford the AIDS virus monopoly? Peter H. Duesberg, Department of Molecular and Cell Biology, Stanley Hall, UC Berkeley, Berkeley, California 94720 USA. Abstract. Until 1984, AIDS science was open. Initially, the new epidemic of pneumonias and Kaposi's sarcomas, since called AIDS, was considered a collection of non-infectious lifestyle diseases. But the Centers for Disease Control in Atlanta published that the pneumonias and Kaposi's sarcomas of male homosexuals, who were addicted to recreational drugs, were caused by a common infectious agent because patients had been linked by sexual contacts. On the basis of the CDC's sexual linkage study, the Secretary of Health and Human Services announced in 1984 the hypothesis that the retrovirus HIV is the cause of AIDS. The HIV/AIDS hypothesis currently holds a monopoly on all AIDS research and treatment. However, the HIV hypothesis is scientifically unproven. It has failed each of 15 testable predictions, as for example that AIDS would explode via sexual transmission of HIV into the general population. Moreover, HIV meets all classical criteria of a harmless passenger virus: unpredictable intervals between infection and any subsequent disease, and unpredictable presence and activity of the virus during a disease. Since HIV is rare in the US, it is a marker of real AIDS risks: frequent injection of intravenous drugs, thousands of drug-mediated sexual contacts, and transfusions. Indeed, AIDS does not meet even one of the classical criteria of an infectious disease, as for example equal distribution between the sexes, disease within days or weeks after infection, and exponential spread of the disease in an unimmunized population, Farr's law. Far from being beneficial, the HIV/AIDS hypothesis has become a threat to public health in the last 10 years. It is the sole basis for 1. The daily treatment of at least 200,000 HIV positives with cytotoxic DNA chain terminators originally designed to kill growing human cells for chemotherapy, like AZT, that are now prescribed as anti-HIV drugs. 2. The clean needle programs that encourage intravenous drug use, and the misinformation that HIV infection is the only health risk of recreational drug use. However, recreational drugs, such as heroin, cocaine, amphetamines, and nitrite inhalants, have long been known to have immunotoxic, cytotoxic, and/or carcinogenic effects, and 3. The anxiety and the many restrictions of human rights associated with a positive HIV test. Here it is proposed that American and European AIDS is caused by the long-term consumption of recreational and of anti-HIV drugs like AZT. The drug AIDS hypothesis correctly predicts American/European AIDS. 1. AIDS is restricted to intravenous and oral users of recreational drugs and AZT. 2. AIDS is 87% male, because males consume this share of recreational drugs. 3. AIDS occurs in newborns, because mothers use recreational drugs during pregnancy. 4. AIDS is new in America, because AIDS is a consequence of the recreational drug use epidemic that started in the 1960s, and of AZT prescriptions that started in 1987. 5. AIDS occurs only in a small fraction of recreational drug users, because only the highest lifetime dose of drugs causes irreversible AIDS-defining diseases. Likewise, only the heaviest smokers get emphysema or lung cancer. 6. AIDS manifests as specific diseases in specific risk groups, because each group has specific drug habits. For example, pulmonary Kaposi's sarcoma is exclusively diagnosed in male homosexuals who inhale carcinogenic alkyl nitrites. 7. AIDS does not occur in millions of HIV-positive non-drug users, and there are thousands of HIV-free AIDS cases, because AIDS is not caused by HIV. 8. AIDS is stabilized, even cured, if patients stop using recreational drugs or AZT, regardless of the presence of HIV. The drug hypothesis predicts that AIDS is an entirely preventable and in part curable disease. The solution to AIDS could be as close as a very testable and affordable alternative to the HIV hypothesis, the drug AIDS hypothesis. The emperor marched in the procession under the beautiful canopy, and all who saw him in the street and out of the windows exclaimed, "Indeed, the emperor's new suit is incomparable. What a long train he has!" "But he has nothing on at all," said a little child at last. "But he has nothing on at all," cried at last the whole people. That made a deep impression upon the emperor, for it seemed to him that they were right, but he thought to himself, "Now I must bear up to the end." And the chamberlains walked with still greater dignity, as if they carried the train which did not exist. Hans Christian Andersen, The Emperor's New Suit. 1. Fabricating the case for infectious AIDS. Hardly anybody remembers that in its first 3 years, from 1981 to 1984, AIDS science was open. The new epidemic of pneumonias and Kaposi's sarcomas, that was called AIDS, was considered infectious by some, but many independent investigators and even scientists from the Centers for Disease Control, CDC, in Atlanta considered AIDS behavioral diseases. Recreational drugs such as nitrite and ethyl chloride inhalants, cocaine, heroin, amphetamines, phenylcyclidine, LSD, and some others were proposed by epidemiologists and toxicologists as the causes of AIDS because in the early 1980s nearly all AIDS patients were either male homosexuals who had used these drugs as aphrodisiacs and psychoactive agents, or were heterosexual intravenous drug users. Goddard et al., 1982, Marmer et al., 1982, Jaff et al., 1983, Matherwack et al., 1984, Haverkas et al., 1985, Newell et al., 1985a, Newell et al., 1985b, Lauritsen and Wilson, 1986, Haverkas and Doherty, 1988. Drugs seem to be the most plausible explanation for the restriction of AIDS to these risk groups, because drug consumption was their only specific common denominator not shared with the general population. This original drug AIDS hypothesis was called the lifestyle hypothesis, Oppenheimer, 1992. But in April 1984 the Secretary of Health and Human Services and AIDS researcher Robert Gallo from the National Institutes of Health, NIH, announced at an international press conference in Washington that a virus is the probable cause of AIDS, Altman, 1984. This AIDS virus, Altman, 1984, had been discovered a year earlier in France, in a male homosexual without AIDS, Barsinasi et al., 1983. Within 2 years after that announcement, an international committee of retrovirologists had named this virus, the human immunodeficiency virus, HIV, to indicate that it was the accepted cause of AIDS, Coffin et al., 1986. The road for the ready acceptance of the AIDS virus by the scientific community had been paved by epidemiologists from the CDC. By tracing sexual contacts of male homosexual AIDS patients, the CDC claimed that it could link patients who had sexual exposure with other AIDS patients within 5 years of the onset of symptoms. Auerbach et al., 1984. On that basis, the CDC proposed that AIDS may be caused by an infectious agent that is transmissible from person to person in a manner analogous to hepatitis B virus infection, through sexual contacts, through parenteral exposure by intravenous drug abusers, through blood products, and, perhaps, through mothers who are intravenous drug users to their infants. Auerbach et al., 1984. However, compared to hepatitis B or any other authentic infectious disease, the CDC's case for infectious AIDS was bizarre with regard to the diversity of the diseases linked. The CDC had linked by sexual contact the Kaposi's sarcomas of some patients to the pneumonias of others and vice versa. The uncritical acceptance by the scientific community of a common infectious cause for such diametrically different diseases as cancer and pneumonia allowed the CDC to fabricate infectious AIDS. Since cancer, pneumonia, and by now about 30 different diseases were all said to have the same cause, they would soon all be called by the same new name, AIDS, Institute of Medicine, 1988, Centers for Disease Control and Prevention, 1992, National Institute of Allergy and Infectious Diseases, 1994. A second bizarre element in the CDC's case for infectious AIDS was the assumption of an average latency period from infection to AIDS of 10 months, Auerbach et al., 1984, now 10 years, see below. The assumption of a microbe that only causes disease after an average latency period of 10 months was without proven precedent. It was necessary, because prospective patients were asymptomatic at the time of sexual exposure, and only developed AIDS up to 5 years after a critical contact, Auerbach et al., 1984. Indeed, the carriers of the assumed infectious agent had to be exceedingly healthy during the latency period, because they had large numbers of approximately 250 different male sexual partners each year. In view of such large numbers of sexual contacts and the long latency periods between infection and AIDS, tracing the one contact that would cause a disease had to be a masterpiece of epidemiological detective work. Therefore, the CDC's case for sexual transmission of AIDS was about as compelling as the claim that one car had a flat tire at an intersection, because another car had blown a head gasket at the same intersection in the previous 5 years. Nevertheless, the sexual contact study was accepted by the scientific community as proof for infectious AIDS without further scrutiny. The fact that linked patients have been frequent users of inhaled amyl and butyl nitrite and of recreational drugs other than nitrite was not considered an AIDS risk by the CDC in 1984, Auerbach et al., 1984, although CDC scientists had originally proposed recreational drugs as the cause of AIDS, Oppenheimer, 1992. In 1984, the CDC also presented typical hemophilia diseases like pneumonia and candidiasis as AIDS from parenteral infection via blood transfusions in support of its claim that AIDS was infectious. Curran et al., 1984, Evatt et al., 1984, Duesberg, 1995b. The paradox that none of the CDC's hemophiliacs with AIDS would have developed Kaposi's sarcoma from an infectious agent that presumably caused Kaposi's sarcoma in homosexuals was effectively hidden because all these entirely unrelated diseases had been named AIDS. Duesberg, 1992, Duesberg, 1994a, Duesberg, 1995b. Indeed, the CDC, originally established to fight infectious diseases, had grown desperate for a new infectious disease, because ever since polio had been eliminated by vaccines over 30 years ago, no new infectious diseases had plagued the Western world. In the words of a Red Cross official, the CDC increasingly needs a major epidemic to justify its existence. Associated Press, 1994. Infectious AIDS, but not the drug AIDS hypothesis, offered such an opportunity. A new infectious epidemic readily generates fear and funding. But research on the toxicity of recreational drugs is trivial, not likely to make headlines in the scientific literature. As a possible investment in its future existence, the CDC has recently launched a new journal, Emerging Infectious Diseases, to raise public awareness of exotic bugs. Kaiser, 1994. For example, in 1994 the CDC promoted the hantavirus after it presumably killed some Indians. Denetclaw and Denetclaw, 1994a, Denetclaw and Denetclaw, 1994b, into a threat to the nation, and in 1995 the Ebola virus, that had apparently killed some Zairens, was promoted into a global killer virus. Associated Press, 1995a. The CDC claimed that 108 people may have been killed by the Ebola outbreak in Zaire in 1995, Centers for Disease Control, 1995. But it failed to mention that 20% of the 55 million Zairens are Ebola virus antibody positive, having survived the virus without apparent disease. Dietrich J., 1995. As AIDS claimed ever more victims and gained ever more media attention, the CDC's message that AIDS was a new infectious disease was enthusiastically picked up by the stars of medical research, particularly the virologists. A new infectious disease is a magnet for virologists, microbiologists, and immunologists because it holds the promise for a new microbial pathogen and new vaccines. Since the discovery of pathogenic microbes by Robert Koch and Louis Pasteur in the 1880s, the identification of a new microbial pathogen has been the key for many brilliant careers like those of Walter Reed, John Enders, and Albert Sabin. Stated the New York Times on the search for an AIDS virus, the greatest thrills for a scientist are in discovering a new microbe, a new disease, cure and prevention. Altman, 1992. After decades of basic research in the war on cancer, an army of highly sophisticated virologists had failed to prove that viruses can cause cancer in humans. Greenberg, 1986, Booth, 1988. Among these were the current leaders of AIDS research, Luc Montagnier from France, Robin Weiss from the UK, David Baltimore, J. Levy, Robert Gallo, and even Peter Duesberg from the US, among others. Searching for other diseases for their viruses, most cancer virologists welcomed AIDS as a new frontier to apply their considerable skills. Duesberg, 1987, Booth, 1988, Duesberg and Schwartz, 1992. With an AIDS virus, the medical virologists could continue their familiar research, and their companies could extend their markets from the narrow confines of conventional virus tests and vaccines to the new multi-billion-dollar markets of HIV antibody tests, HIV vaccines, and anti-HIV drugs. Weiss and Jaff, 1990, Duesberg, 1992. The AIDS virus also proved to be the politically correct cause of AIDS. No AIDS risk group could be blamed for being infected by a God-given egalitarian virus. A virus could reach all of us. Nobody would be ostracized since we are all in this together. Not so with drugs. The consumption of illicit psychoactive drugs implies individual and social responsibilities that nobody wanted to face. Once accepted as the politically correct explanation of AIDS, the HIV hypothesis has become the central investment for a whole generation of AIDS scientists, AIDS companies, AIDS journalists, AIDS politicians, and gay activists. The perceived danger of an AIDS virus decimating the general public also provided the scientific and moral arguments for quick and unreflective action and for the complete dismissal of the competing drug AIDS hypothesis. The fear of nature's presumably uncontrollable microbes created an unscientific war mentality that has since dominated the field. Christie, 1994. Scientists, healthcare workers, and journalists would rather be safe and fast in protecting against HIV, than sorry and slow in reflecting about the clinical and political consequences of drug use. Lang, 1994. The confrontation with man-made drugs, after all, would have to take second place in urgency, as they would not reach the innocent public. Claiming this priority, the virus AIDS orthodoxy justifies intolerance, even censorship, of all those who question infectious AIDS. San Francisco Project Inform, 1992, Maddox, 1993b, Maddox, 1993a, Cohen, 1994a, Lang, 1994, see Chapter 12. Epidemiologists from the CDC warn that to ignore this HIV/AIDS concept would result in an unconscionable tragedy. Garza, Drottman, and Jaff, 1994. Virologists are quick to call those who question the virus AIDS hypothesis irresponsible and pernicious. Booth, 1988, Baltimore and Feinberg, 1989. And the New York Times still calls all non-HIV science cruelly irresponsible anti-science. Lewis, 1994. Therefore, the AIDS virus won unprecedented popularity within a short time after its announcement. But 11 years later, in 1995, the virus AIDS hypothesis has still failed to produce any public health benefits in the war on AIDS. No vaccine, no antiviral drug, no cure, not even an effective AIDS prevention have been developed. It cannot even be predicted whether and when an infected person will get ill. And it cannot predict which of the about 30 AIDS diseases it will be. Duesberg, 1992, Bendit and Jasny, 1993, Cohen, 1994a, Cohen, 1994b, Wade, 1995. Moreover, the very basis of the virus AIDS hypothesis, the assumption that AIDS is infectious, has since become questionable on several grounds. For example, 1. Would you have believed AIDS is infectious 11 years ago, if you had known that until now not even one of the doctors and healthcare workers who have treated the over 400,000 American AIDS patients since 1984, Centers for Disease Control and Prevention, 1994C, is confirmed to have contracted AIDS from a patient? Even if that would not have changed your mind, would you still believe in infectious AIDS if you had considered that the healthcare workers were neither protected by an anti-HIV vaccine nor by an antiviral drug? Duesberg, 1992. 2. Would you have believed that a sexually transmitted virus was causing AIDS if you had known that none of the wives of the 15,000 HIV-positive American hemophiliacs has contracted AIDS from their husbands in the last 10 years? Their risk of developing an AIDS-defining disease is the normal background of these diseases in the U.S. Duesberg, 1992, Duesberg, 1995b. 3. Would you have believed that AIDS was contagious if you had known that after a marriage of 10 years, neither the wife nor the 6-year-old daughter of the late tennis star and AIDS patient Arthur Ashe have developed AIDS or even become HIV positive? Ashe and Rampersad, 1993. Or that the long-term lover of the movie star Rock Hudson has no AIDS symptoms, 10 years after Hudson died from AIDS in 1985? Would you believe that AIDS was sexually transmitted if you had known that, after a 13-year marriage and 2 children, the husband of the late AIDS patient Elizabeth Glazer is healthy and HIV-free? Champkin, 1994. 4. Would you have believed in an AIDS virus if you had known that nobody ever contracted Kaposi's sarcoma in the U.S. from a blood donor with Kaposi's sarcoma? Haverkas, Drottman, and Hansen, 1994. 5. Would you have believed AIDS is a sexually transmitted disease in 1984 if you had known that 11 years later there is still no AIDS in American heterosexuals, not even in prostitutes, unless they are drug addicts? Duesberg, 1992. 6. would you have believed in a sexually transmitted AIDS virus if you had considered that such a virus would be incompatible with life? Because sex is the only known source of human life, a sexually transmitted, fatal virus would have exterminated itself together with its host. Duesberg, 1992. Have we lost the war on AIDS because we have mistaken a harmless virus for its real cause? 2. The HIV-AIDS hypothesis proves to be unprovable. The HIV-AIDS hypothesis, Institute of Medicine, 1988, National Institute of Allergy and Infectious Diseases, 1994, postulates that 1. HIV causes immunodeficiency by killing T cells, lymphocytes. 2. Immunodeficiency occurs on average only 10 years after this virus has been neutralized by antiviral immunity, a condition termed a positive HIV test. 3. Immunodeficiency is the basis for about 30 previously known diseases, including pneumocystis pneumonia, tuberculosis, candidiasis, Kaposi's sarcoma, dementia, diarrhea, 10% weight loss, and many others. Table 1. 4. AIDS is a sexually transmitted disease, because HIV is a sexually transmitted virus. Owing to the immense popularity of this hypothesis, over 100,000 scientific papers have been published on HIV since 1984. But not even one of these has been able to explain how HIV causes AIDS. Worse yet, not one paper exists that proves that HIV causes AIDS. Duesberg, 1992, Dixon, 1994, Fields, 1994, Schock, 1994, Thomas Jr., Mullis, and Johnson, 1994. In view of this proof deficit, the HIV/AIDS establishment cites the perfect correlation between HIV and AIDS as support for the hypothesis that HIV causes AIDS. Blattner, Gallo, and Temin, 1988, Weiss and Jaff, 1990, San Francisco Project Inform, 1992, Maddox, 1993b, Garza, Drottman, and Jaff, 1994, National Institute of Allergy and Infectious Diseases, 1994. However, this argument is inadequate as an element of proof for 3 reasons. 1. According to the HIV/AIDS hypothesis, the 30 AIDS-defining diseases are diagnosed as AIDS only when antibody against HIV is present. In its absence, these diseases are called by their old name and caused by their old causes. In other words, AIDS is defined entirely by its hypothetical cause, HIV. Therefore, the perfect correlation is not a natural coincidence but a perfect artifact of the definition of AIDS by its hypothetical cause, HIV. It is one of the purest examples of circular logic. 2. The HIV antibody test for the detection of HIV is indirect, because it does not assay for the virus. Moreover, it is not reliable. Up to 90% false positives are obtained, depending on the subjects tested and on the tests used. Duesberg, 1993f, Papadopoulos Eliopoulos, Turner, and Papademetrio, 1993. 3. Even a perfect correlation is not sufficient to prove causation. For example, perfect correlations between yellow teeth and lung cancer, or between hospitalization and death do not prove that one causes the other. Duesberg, 1989, Smith and Phillips, 1992, Duesberg, 1993d. In the absence of direct proof, the merit of a scientific hypothesis is determined by the accuracy of its predictions. For example, there is no direct proof for the hypothesis that smoking causes lung cancer and emphysema, but the prediction that long-term smoking causes these diseases has validated this hypothesis. In the following we will analyze the predictions of the HIV/AIDS hypothesis. Duesberg, 1994a. 2.1. HIV-infected persons will get AIDS, and otherwise matched HIV negatives will not. In the face of the relentless propaganda for the HIV hypothesis, it comes as a big surprise to almost everybody that there is not even one study to show that American, heterosexual or homosexual men, who are HIV positive but not drug users or hemophiliacs ever get AIDS. More precisely, there is no study to show that such men would get AIDS-defining diseases that exceed the long-established, low background of these diseases in otherwise matched, HIV-free counterparts, Duesberg, 1995a, see Chapter 10. There is not a single epidemiological study to support the most frightening slogan of the HIV orthodoxy, that HIV positives develop AIDS-defining diseases because of HIV. All studies that claim HIV causes AIDS have instead analyzed the AIDS risks of HIV-positive people who were recreational drug users, were treated with AZT or other antiviral drugs, had received transfusions, suffered from congenital diseases, or were subject to exotic lifestyles as in Africa. The AIDS risks of such groups were then determined by comparisons, either with normal HIV-free people or with HIV-negative people from risk groups who were not matched for drug use or other AIDS risks. Duesberg, 1992, Duesberg, 1993a, Duesberg, 1993c, Duesberg, 1993d, see Chapter 8. In other words, there is no epidemiological evidence properly controlled for confounding factors that HIV is a deadly virus or the virus that causes AIDS. In view of the enormous experimental difficulties and costs in sorting out the possible role HIV plays in AIDS from the roles that recreational drugs, AZT, transfusions, congenital diseases, and exotic lifestyles play, it is surprising that the assumption that HIV causes AIDS has never been studied in people who are free of confounding AIDS risks. There can only be one plausible explanation for the absence of an epidemiological study that shows that HIV causes AIDS in people who are not in risk groups. HIV does not cause AIDS. Indeed, there are 1 million HIV-positive Americans, National Institute of Allergy and Infectious Diseases, 1994, and 17 million HIV-positive humans, Merson, 1993, World Health Organization, 1995, who are healthy, probably because they are not subject to real AIDS risks other than the hypothetical AIDS risk HIV. Moreover, HIV positives who stop practicing risk behavior or stop being subjected to AIDS risks even recover lost immunity despite the presence of HIV, see below Section 8. For example, HIV-positive hemophiliacs treated for 3 years with highly purified blood clotting factor regained lost immunity, while controls treated with unpurified blood products continued to lose immunity. Sirmatis et al., 1993, Duesberg, 1995b and Chapter 11. Thus HIV is only ever deadly for people who are at risk for AIDS from toxic drugs or who depend on long-term blood transfusions to treat underlying deadly diseases. Duesberg, 1992, see Chapter 6. 2.2, American AIDS is new, because HIV is new in America. However, in America HIV is a long-established retrovirus. Duesberg, 1992, see Chapter 6. Ever since the virus could be detected in 1984, an unchanging 1 million Americans are HIV positive. Fig 1a, Curran et al., 1985, National Institute of Allergy and Infectious Diseases, 1994, Farber, 1995b. By contrast, a new microbe/virus spreads exponentially in a susceptible population, CV. Thus the non-spread of HIV establishes it as an old virus in America. Duesberg, 1992. 2.3, HIV is active and abundant in persons with AIDS, and inactive and rare in healthy virus carriers. All microbes cause diseases by killing or alterating a larger number of target cells than the host can spare or regenerate during the course of an infection. Thus HIV would have to infect and kill at least 50% of all human T cells to cause AIDS. However, in AIDS patients HIV is found hibernating in only 0.1% of T cells, and biochemically active in less than 0.01% of T cells. Duesberg, 1992, Duesberg, 1993, Piatok et al., 1993. Indeed, there are healthy HIV-positive people with 30 to 40 times more infected T cells than in AIDS patients. Simmons et al., 1990, Bagasra et al., 1992, Duesberg, 1992. The fact that the vast majority of susceptible T cells remain uninfected, even in people dying from AIDS, is the definitive evidence that there is no active HIV in HIV antibody-positive persons. HIV is neutralized by antiviral immunity, even in AIDS patients. If there were unneutralized HIV, all T cells would be infected. The fundamental problem for the HIV hypothesis is not just how HIV works, but how it causes fatal diseases when it does not work at all. Since there is no rational explanation for how HIV could cause AIDS, HIV researchers now postulate a multiplicity of indirect mechanisms of pathogenesis. Blattner, Gallo, and Temin, 1988, Booth, 1988, Gallo, 1991, Maddox, 1991, Weiss et al., 1992, Maddox, 1993b, Maddox, 1995, Wade, 1995, Wayne Hobson, 1995. 2.4 HIV causes AIDS by killing T cells. However, viruses that integrate their genomes with that of the host, like HIV, cannot kill the host cell. Since the genes of such viruses are part of the host's genes, integrated viruses can only replicate as long as the host survives integration and remains able to express integrated viral genes. All integrated viruses survive from passive, and some retroviruses also from active replication with the host. This strategy only works if the host survives integration. If the virus were to kill the cell as it is integrated, integration would be a useless exercise and it would be undetectable. Indeed, HIV is mass-produced for the HIV test in immortal T cell lines and cell culture at titers of 106 infectious units per mil. Rubenstein, 1990, Karpaz et al., 1992. Luc Montagnier, the discoverer of HIV, and many other researchers have confirmed that HIV does not kill T cells. Lemaitre et al., 1990, Duesberg, 1992. 2.5 Since the generation time of HIV is 2 days, HIV will cause AIDS 2 weeks after infection. The HIV hypothesis predicts AIDS within 2 weeks after infection, because HIV, like all other retroviruses, replicates within 2 days. During that time one infected cell produces at least 100 new viruses. Weiss et al., 1985. In the absence of antiviral immunity, these 100 viruses would in turn infect 100 cells producing 100x100 viruses, or 104 infected cells within 4 days after infection. Within 14 days of such exponential growth, 1014 cells, the equivalent of a human body, would be infected. This is the typical latent period of proven pathogenic retroviruses, like Rous sarcoma virus, and of pathogenic human viruses like flu, measles, mumps, chickenpox, and herpes, which all have generation times like HIV. Fenner et al., 1974, Mims and White, 1984. However, if dated from the time of HIV infection, AIDS occurs at totally unpredictable times. The latent period between infection and AIDS was estimated to average 10 months in 1984, Auerbach et al., 1984, 10 years in 1988, Institute of Medicine, 1988, and over 20 years in HIV-positive hemophiliacs in 1994, Phillips et al., 1994. A Berkeley mathematician recently has determined the most accurate formula for the latent period of HIV, by subtracting 1984, the year when HIV was proposed to cause AIDS, from the current calendar year. But blaming AIDS on a HIV infection that occurred 10 years earlier is the logical equivalent of blaming today's broken leg on stumbling over a crack in the sidewalk 10 years ago. 2.6 Viral AIDS will spread exponentially, explode. The AIDS orthodoxy has predicted that according to Farr's law, Bregman and Langmuir, 1990, AIDS would spread exponentially, explode, into the general, unimmunized population. Duesberg, 1992, just like all other new infectious diseases. However, AIDS in America and Europe remained confined to the original risk groups, i.e. male homosexuals practicing risk behavior and intravenous drug users. National Commission on AIDS, 1991, Centers for Disease Control and Prevention, 1994b. Instead of growing exponentially, AIDS in America and Europe has increased slowly, over 15 years, far from reaching saturation of the susceptible population of over 100 million sexually active adults, Fig 1a. AIDS behaved just like an occupational disease. 2.7 The spread of AIDS will follow the dissemination of HIV. However, there is no correlation between the spreads of AIDS and HIV in America. In the last 10 years, AIDS increased in America from a few hundred to about 100,000 cases annually, Fig 1a, Centers for Disease Control and Prevention, 1994c. The burst of AIDS cases in 1993 is largely an artifact of the most recent redefinition of AIDS, which nearly doubled the AIDS cases in 1 year, Centers for Disease Control and Prevention, 1994c. However, during those same 10 years HIV did not spread at all, Fig 1a. Ever since HIV became detectable in 1985, an unchanging 1 million Americans have been HIV positive up to 1994, Fig 1a, Duesberg, 1992, Duesberg, 1994a, National Institute of Allergy and Infectious Diseases, 1994. To hide this discrepancy, a latency period of 10 years has been postulated between HIV and AIDS. 2.8 Like all other sexually transmitted diseases, AIDS in America will equilibrate between the sexes. However, since 1981, AIDS has remained in the original risk groups in America, i.e. male homosexuals, intravenous drug users of which over 75% are males, Duesberg, 1992, and hemophiliacs which are nearly all males. Since 1981, 347,767 out of 401,749, or 87% of all American AIDS cases, have been males, Centers for Disease Control and Prevention, 1994c. 2.9 HIV is a sexually transmitted virus. However, HIV could never survive an evolution from sexual transmission. Based on studies of discordant couples, e.g. hemophiliacs with HIV and spouses without, conducted by the CDC and others, it takes on average 1,000 unprotected sexual contacts to transmit HIV, Hurst and Hulley, 1988, Peterman et al., 1988, Rosenberg and Wiener, 1988, Lawrence et al., 1990, Blattner, 1991. According to Rosenberg and Wiener, HIV infection in non-drug-using prostitutes tends to be low or absent, implying that sexual activity alone does not place them at high risk. The efficiency of transmission in homosexual contacts is also estimated at 1 in 1,000 contacts, Jacques et al., 1994. Since about 10 to 30 sexual contacts are required to generate a child, but 30 contacts are required to transmit HIV, HIV could never survive natural selection on the basis of sexual transmission, because the host would outgrow the parasite. Conventional venereal microbes, like syphilis and gonorrhea, survive because they are transmitted by about 2 sexual contacts, Freeman, 1979. HIV also could not survive from transmission to newborns if it were fatally pathogenic to babies, as is claimed by the proponents of the HIV hypothesis, Blattner, Gallo, and Temin, 1988, Institute of Medicine, 1988. The extremely low efficiency of sexual transmission of HIV also predicts that the safe sex campaigns conducted by the HIV orthodoxy will be of very limited value. Only those would benefit who either have on average 1,000 sexual contacts with HIV positives or those who have on average 250,000 contacts with average Americans, of which only 1,250,000 is HIV positive, Fig 1a, Duesberg, 1992, National Institute of Allergy and Infectious Diseases, 1994. 2.10 AIDS will be restricted by controlling sexual transmission of HIV via safe sex, and parenteral transmission of HIV via clean needles. But AIDS continues to increase steadily despite the safe sex and clean needle programs, Centers for Disease Control and Prevention, 1994c, see Fig 1a. 2.11 healthcare workers will contract AIDS from their patients, scientists from propagating virus, and prostitutes from their clients. Not a single confirmed case exists in the scientific literature of a healthcare worker who contracted AIDS. Duesberg, 1992, Duesberg, 1994a, from one of the over 400,000 American AIDS patients, Centers for Disease Control and Prevention, 1994c. None of the tens of thousands of HIV researchers have developed AIDS from propagating HIV. And no prostitutes picked up AIDS from their clients despite the absence of antiviral vaccines or effective anti-HIV drugs. Duesberg, 1992, Duesberg, 1994a. A few unpublished cases have been claimed, but each of these seem to have been treated with the cytotoxic drug AZT that is sufficient to cause immunodeficiency. See below. Cohen, 1994a. 2.12 Chimpanzees inoculated with HIV will develop AIDS, and the 15,000 American hemophiliacs who were infected by transfusions before 1984 will die from AIDS. Not one of the 150 chimpanzees inoculated with HIV since 1983 has developed AIDS. Duesberg, 1992. Contrary to prediction, the median life of American hemophiliacs has increased 2.5-fold from 11 to 27 years between 1972 and 1987, Institute of Medicine, 1988, Stergreen et al., 1989, although 75%, 15,000, were infected with HIV by transfusions received before 1984, Duesberg, 1992. However, in 1987 the median life of HIV-positive hemophiliacs started to decrease again, Corba et al., 1994, because since then they have been treated with the cytotoxic AZT. Duesberg, 1995b, see Chapter 11, see below. 2.13 Natural or vaccine-induced anti-HIV immunity will cure AIDS or protect against future AIDS. Natural antiviral immunity, a positive HIV test, is observed in many AIDS patients, but does not protect against AIDS. Paradoxically, anti-HIV immunity is by HIV/AIDS definition the only criterion to predict who gets AIDS. With all other viruses and microbes there is no exception. Immunity is the only criterion to predict who does not get a disease. It is for this reason that antiviral/microbial immunity is artificially induced by vaccination. It is also for this reason that the HIV/AIDS establishment has called for an HIV vaccine since 1984. 2.14 All AIDS diseases are consequences of HIV-mediated T-cell deficiency. Indeed, up to 1992 about 61% of all American AIDS diseases, the microbial diseases such as Pneumocystis carinii, Candida, tuberculosis, etc. were consequences of acquired immunodeficiency. Table 1, Centers for Disease Control, 1993. However, 39% were neither caused by, nor consistently associated with, immunodeficiency. These include Kaposi's sarcoma, lymphoma, 10% weight loss, and dementia. Table 1. Accordingly, Kaposi's sarcoma and dementia have been diagnosed in male homosexuals whose immune systems were normal. Murray et al., 1988, Spornraft et al., 1988, Gill et al., 1989, Friedman-Kien et al., 1990, Duesberg, 1992, Kaldor et al., 1993, Beischeler et al., 1994. In 1993, the CDC introduced, once more, a new AIDS definition, Centers for Disease Control and Prevention, 1992. This has shifted the balance of immunodeficiency to non-immunodeficiency AIDS diseases significantly in favor of immunodeficiency diseases, i.e. from 61% to 80%, Table 1, Centers for Disease Control and Prevention 1994a. The critical innovation of this new definition was that a healthy person with less than 200 T cells, but with no clinical disease, would now be registered as an AIDS patient. The new AIDS definition nearly doubled the new AIDS cases, thus adding new life to the sagging curves of the American AIDS statistics, Fig 1a. However, if one subtracts from the 1993 statistics the new AIDS cases with less than 200 T cells, the ratio of the remaining real immunodeficiency diseases to the non-immunodeficiency diseases is almost the same as in 1992. Imagine the rationalizations an unprejudiced virologist, who is aware of the heterogeneity of AIDS-defining diseases, must make to accommodate an AIDS virus. Ever since Cook and Pasteur, microbiologists and virologists were taught that a specific microbe or virus would cause a specific disease e.g. polio, flu, measles, chickenpox, hepatitis, etc., just like a particular musical instrument would make specific sounds. To accommodate the AIDS virus, the concept of a specific microbe causing a specific disease had to be abandoned for the following bewildering scenario. By picking up the AIDS virus from a diarrhea patient, a person would get Kaposi's sarcoma. The Kaposi patient would then be able to cause dementia or pneumonia in others by passing on the diarrhea virus, just as the CDC's sexual contact study had claimed in 1984, Auerbach et al., 1984. As of 1993, any one of these patients could have also caused a clinically undetectable depletion of T cells in others, again by passing on their diarrhea, dementia, Kaposi's sarcoma, and pneumonia virus. Moreover, the unprejudiced virologist would have to reconcile this bewildering pathogenic potential of HIV with the fact that HIV is one of the most primitive viruses in terms of genetic information that exist, carrying only 9,000 nucleotides. This is the viral equivalent of a musical instrument that is said to sound like an orchestra, although it only has the repertoire of a bell. 215. If HIV is the cause of AIDS, the percent incidence of AIDS diseases will be the same in all risk groups. However, the percent incidence of AIDS-defining diseases is very different in different risk groups. For example, Kaposi's sarcoma in America and Europe is almost exclusively observed in male homosexuals, Birol et al., 1990. Intravenous drug users have a proclivity for tuberculosis, weight loss, and pneumonia, Duesberg, 1992, and a very high mortality dying at 30 years, Lockman et al., 1995. Pneumonia and candidiasis are virtually the only AIDS diseases ever diagnosed in hemophiliacs, Duesberg, 1992, Duesberg, 1995b. And bacterial infections other than tuberculosis are almost exclusively diagnosed in babies with AIDS, Centers for Disease Control, 1987, Centers for Disease Control and Prevention, 1992, Duesberg, 1992, see Chapter 6. Thus the percent incidence of an AIDS diseases is very different in different AIDS risk groups. In view of this, some AIDS researchers cite CO factors of HIV, such as recreational drugs and immunosuppressive transfusions, as explanations for risk group-specific diseases. Evans, 1989, Duesberg, 1992, Ludlam, 1992, Root Bernstein, 1995a. However, they fail to provide evidence that such cofactors depend on the cofactor HIV to be pathogenic. The CDC and other mainstream AIDS researchers insist that recreational drugs and transfusions solely enhance the risk to get infected by HIV, rather than playing causative roles in AIDS. Cohen, 1994a. It is for this reason that the CDC refers to drug or transfusion risk groups as exposure categories, Centers for Disease Control and Prevention, 1994a. In fact, the CDC obscures the existence of risk group-specific AIDS diseases by reporting only the percent incidence of AIDS diseases in all risk groups combined, Centers for Disease Control and Prevention, 1994a. It is evident that the HIV/AIDS hypothesis is unable to make even one verifiable prediction, the hallmark of a failed hypothesis. Even if a hypothesis fails to make valid predictions in terms of established scientific criteria, it could by chance lead to a valid prevention or treatment. But the HIV/AIDS hypothesis has not led to any public health benefit. Instead, it has harmed not only AIDS patients but also healthy persons who are at risk for AIDS or just antibody positive. C6. 3. HIV, a harmless passenger virus. Confronted with the evidence that the HIV/AIDS hypothesis has failed to make valid predictions and failed to lead to public health benefits, many agree that HIV may not cause AIDS. But then, they wonder, what does HIV do? Indeed, all viruses are generally assumed to be pathogenic by an unsuspecting public, because a few of them actually are. Likewise, a whole nation is often stereotyped by the characteristics of a minority. For example, the French are considered great lovers and the Italians great singers, because a few of them are great lovers and singers. The same is true for viruses. In reality, most viruses cause no disease at all. Viruses are not here to kill their hosts, not even to cause disease. Instead, viruses are here for exactly the same reasons we are, to continue their species. This goal can only be achieved by keeping the host species alive, and it is achieved best if every host survives the infection. That is the reason that most viruses never cause a disease in their host. Therefore they are called passenger viruses. Passenger viruses are those that take a ride on the host, but demand no more from the host than a passenger demands from an airplane, see Chapter 9. Since HIV is not the cause of AIDS, the simplest and most plausible HIV hypothesis postulates that HIV is just a passenger virus, Duesberg, 1994a. A passenger virus is defined as follows. 1. The time of infection is irrelevant to the onset of any disease. 2. The passenger virus can be either active or passive, either rare or abundant during any disease. 3. The passenger virus can be entirely absent during any disease. 4. If the passenger virus is repressed by a failing immune system, as for example during a disease, the passenger virus may or may not contribute to the disease. For example HHV6, Cone et al., 1994, or cytomegalovirus may contribute their specific pathogenic properties to an immunodeficient patient. HIV meets all these criteria with regard to its relation to AIDS. 1. HIV infects at totally unpredictable times prior to or even after the onset of AIDS, see 8 below, or not at all, Duesberg, 1992, Fair et al., 1992, Duesberg, 1993f. 2. HIV is typically passive and rare during AIDS, hence the notorious difficulties of leading AIDS researchers in isolating HIV from AIDS patients, Duesberg, 1992, see Chapter 6. 3. There are thousands of HIV-free AIDS cases, e.g. HIV-free homosexuals with Kaposi's sarcoma and HIV-free intravenous drug users with tuberculosis, Duesberg, 1993f, see Chapter 7. 4. There is no report in the literature that AIDS patients are clinically distinguishable from each other because HIV is active or passive or not present at all, Duesberg, 1993b, Duesberg, 1994a. Thus HIV is a harmless passenger virus, even when it is active in some rare immunodeficient persons, Duesberg, 1993e. If this is true, there should be many more HIV carriers than AIDS patients. Indeed, there are 1 million healthy HIV-positive Americans, Duesberg, 1992, Duesberg, 1994a, and there are 17 million healthy HIV-positive humans, Merson, 1993, National Institute of Allergy and Infectious Diseases, 1994, World Health Organization, 1995. Nevertheless, there is some tenuous evidence that HIV can function as an autonomous pathogen, causing a mild flu-like or mononucleosis-like condition, prior to antiviral immunity, Albert et al., 1987, Duesberg, 1987, Kessler et al., 1987, Gaines et al., 1988, Marcus and the CDC Cooperative Needlestick Surveillance Group, 1988, Tyndall et al., 1988, Peterson et al., 1990, Duesberg, 1992, New, Stein and Schnittman, 1993, see Chapters 1, 6. However, in millions of HIV positives, HIV infection has gone unnoticed, because they do not experience a characteristic HIV disease prior to antiviral immunity like measles, mumps, or flu, which all occur prior to immunity against these viral infections. The rare cases in which HIV infections, prior to antiviral immunity, have been linked with mononucleosis or flu-like symptoms or restricted to prospective studies of male homosexuals at risk for AIDS. These cases could either be coincidences with a common cold or with intoxications from recreational drug use. See below. Gaines et al., 1988, Tyndall et al., 1988, Peterson et al., 1990, rather than evidence for HIV disease. 4. HIV a marker for AIDS risks. Even those who understand all virological arguments against HIV as the cause of AIDS, and for HIV as a passenger virus, have misgivings about dismissing the HIV/AIDS hypothesis, because HIV is more common in AIDS patients than in the healthy population. This sounds like an ominous connection, but only if it is taken out of its trivial microbiological context. This trivial context is that AIDS risk behavior is synonymous with collecting microbes. The common denominator of all AIDS risk groups in America and Europe is that they collect microbes either from unsterile drugs injected with unsterile equipment, or from the thousands of drug-mediated sexual contacts that are required to transmit HIV sexually, or from transfusions received for the treatment of illnesses. Jaff et al., 1983, Auerbach et al., 1984, Lauritsen and Wilson, 1986, Haverkas and Doherty, 1988, Rappaport, 1988, Adams, 1989, Callan, 1990, Lifson et al., 1990, Archibald et al., 1992, Duesberg, 1992, Jones, 1994, Mullis, 1995. This is the reason that numerous uncommon microbes are common not only in AIDS patients but also in healthy persons from AIDS risk groups. For example, the bacteria that cause syphilis, gonorrhea, and tuberculosis, the hepatitis virus, rare strains of herpes virus, putative leukemia virus, genital papilloma virus, and even HIV are all common in AIDS risk groups and AIDS patients but uncommon in the general population. Duesberg, 1992, see Chapter 6. Thus HIV is just one of many microbial markers of AIDS risk behavior. Since AIDS is defined as one of 30 diseases in the presence of HIV, rather than any other microbial or viral marker, the correlation between HIV and AIDS is in theory 100%. 5. The myth of infectious AIDS unconfirmed. If a hypothesis is unproductive, and unable to make verifiable predictions, the scientific method calls for alternative hypotheses. To find the correct AIDS hypothesis, we need to decide first whether to look for other viruses and microbes or for drugs as causes of AIDS. In other words, we need to know whether AIDS is infectious or not. There are 5 classic criteria to define an infectious disease. In individuals. 1. The causative microbe/virus is abundant and very active in target tissues during the course of the disease. 2. The disease follows within days or weeks after infection, because microbes/viruses multiply exponentially with generation times of 0.5 to 48 HRS, unless they are stopped by immunity, see above. In populations. 3. The disease spreads, according to Farr's law, exponentially in an unimmunized population within weeks or months, and subsequently fades away as antiviral immunity builds up. Bregman and Langmuir, 1990. The bell-shaped curve of a seasonal flu epidemic is the model. 4. Infectious diseases are equally distributed between the sexes. 5. Infectious diseases are most commonly observed in those under 20 and over 60 years of age. This is because after birth the immune system builds up a wide repertoire of antimicrobial resistances that is nearly complete at 20, and over 60 the system begins to decline. But American/European AIDS does not fit one of these criteria. 1. There is no abundant microbe common to all American AIDS cases. If HIV is present, it is typically rare and hibernating. 2. If dated from the time of HIV infection, AIDS occurs at entirely unpredictable times ranging from less than 1 year to over 10 years or never. It has now been 10 years since 1 million Americans were found to be HIV infected. Most of these and 17 million healthy, HIV-positive non-Americans are still waiting for HIV to cause AIDS. 3. American AIDS has slowly increased over 10 years. Although AIDS affects annually only a small fraction of susceptible persons, less than 100,000 out of a susceptible pool of 250 million Americans, it has now almost plateaued for 4 years after adjusting for new additions of diseases to the AIDS definition, Centers for Disease Control and Prevention, 1994b. Thus AIDS in America has increased steadily over years, just like an occupational disease, as for example lung cancer from smoking. There is no evidence for immunity and no evidence for a bell-shaped AIDS curve. 4. American AIDS is 87% male, Centers for Disease Control and Prevention, 1994c, which is epidemiologically as far from equality between the sexes as the sun is from the earth. A similar sexual bias has been observed early in the epidemic of smoking-related diseases in the 1960s, before women picked up smoking at the same rate as men. 5. 98% of all American AIDS cases are over 20 and under 60, Centers for Disease Control and Prevention, 1994c. Only 1% each are under 20 and over 60. Such an age bias is typical of occupational diseases, like bullet wounds for soldiers. Thus AIDS in America and Europe, Duesberg, 1992, does not meet even one of the criteria of infectious disease. Indeed, even proponents of the HIV/AIDS hypothesis, such as Jaap Goudsmit from the University of Amsterdam, grant that AIDS does not have the characteristics of an ordinary infectious disease. This view is incontrovertible, Goudsmit, 1992. The AIDS epidemiologists Eggers and Weyer from the University of Cologne state that the spread of AIDS does not behave like the spread of a disease that is caused by a single sexually transmitted agent, Eggers and Weyer, 1991. To reconcile AIDS with infectious disease, they simulated a cofactor that cannot be identified with any known infectious agent. The epidemiologists Anderson and May from the University of London had to invent assortative scenarios for different AIDS risk groups to match AIDS with infectious disease, Anderson and May, 1992. Until we have scientific evidence, infectious AIDS is just a myth and, in view of the facts, a very implausible myth indeed. 6. The HIV/AIDS hypothesis is costly, unproductive, and harmful. For 11 years now the world has fought the war on AIDS united by the HIV/AIDS hypothesis. But despite its enormous popularity, the virus AIDS hypothesis has been a complete failure in terms of public health benefits. No vaccine has been developed that prevents AIDS, no drug that cures AIDS, no policy that stops the spread of AIDS. Bendit and Jasny, 1993, Fields, 1994, Swinbanks, 1994, Wade, 1995. Whatever the reasons are for the complete failure of the HIV/AIDS hypothesis to produce public health benefits, one thing is clear, it was not for lack of trying. The passionate complaint of Schilt's 1987 book, and the band played on, that indifference was the only obstacle against a solution of AIDS, Schilt's, 1987, has long become profoundly obsolete. Since 1984, an unprecedented $35 billion has been paid by the U.S. taxpayer alone in support of HIV/AIDS research and treatment, more than for all other viral and microbial diseases combined. AIDS Weekly, 1995, Gutknecht, 1995, Henry, 1995, Stone and Cohen, 1995. With all this spending, more research has been done on HIV than on any other virus in history, but absolutely no progress has been made against AIDS. Time, at least, has voted against the HIV hypothesis. Traditionally, such complete failures are the consequences of a flawed hypothesis. But the HIV/AIDS establishment does not only cost dearly and fails to produce positive results, it also causes irreparable, 1. Clinical, 2. Educational, and 3. Psychological damage. 1. Clinical damage. Worldwide about 200,000 HIV antibody-positive persons are prescribed, every 6 hours, the highly toxic DNA chain terminator AZT or equivalents like DDI, DDC, and D4T as anti-HIV drugs, Duesberg, 1992, Thomas, 1995. Most of these, i.e. 200,000 minus the 50,000 to 80,000 annual AIDS patients in America and Europe, are healthy HIV positives given AZT to prevent AIDS. Recently these include even unborn American and French children and their HIV-positive mothers, although the risk of such children to pick up HIV from their mothers is only about 25%, The Lancet, 1994, Farber, 1995a. AZT was designed 30 years ago to kill growing human cells for cancer chemotherapy, Horwitz, Chua and Noel, 1964, Duesberg, 1992. In view of its inevitable toxicity, AZT was approved as an anti-HIV drug only tentatively in 1987, Collada, 1987. See the warnings of a non-medical manufacturer, Sigma, on the label of an AZT bottle, Fig 2. The label points out, with skull and crossbones, AZT's toxicity to the bone marrow, the source of T cells. Indeed, AZT therapy of HIV appears harmful and irrational. Since HIV is postulated to cause AIDS by killing T cells, see above, it is irrational to kill the same HIV-infected cells twice, once with HIV and again with AZT. Moreover, it is harmful to kill numerous uninfected cells with AZT collaterally, Collada, 1987, Lauritsen, 1990, Nussbaum, 1990, Wyatt, 1994. Accordingly, AZT has failed to cure even one AIDS patient or to prevent AIDS in HIV-infected persons, Duesberg, 1992, Audun et al., 1993, Talkers et al., 1993, Beischeler et al., 1994, Godard et al., 1994, Lenderking et al., 1994, Seligman et al., 1994, Valberding, 1995, Ho, 1995. Instead, evidence is growing that AZT causes AIDS-defining and other diseases as expected from a chain terminator of DNA synthesis, see below, Mir and Costello, 1988, Lauritsen, 1990, Duesberg, 1992, Lauritsen, 1992, Beischeler et al., 1994, Cohen, 1994a, Duesberg, 1994a, Godard et al., 1994, Lewis Thornton, 1994. Yet this evidence is either denied or belittled by the AIDS establishment, as the following examples document. I. The observation that HIV dementia among those reporting any antiretroviral use, AZT, DDI, DDC, or D4T, was 97% higher than among those not using this antiretroviral therapy is interpreted by its authors with little concern for percentages. This effect was not statistically significant, Beischeler et al., 1994. Godard et al. Explain their stunning results that HIV-positive hemophiliacs on AZT have 4.5 times more AIDS and have a 2.4 times higher mortality than untreated HIV-positive hemophiliacs by saying this happened probably because zidovudine was administered first to those whom clinicians considered to be at highest risk, Godard et al., 1994. 2. Sa et al. Explain their observation that male homosexuals on AZT have a 2 to 4-fold higher risk of pneumocystis pneumonia than untreated controls as follows. Zidovudine was no longer significant after T helper lymphocyte count was considered, primarily because non-users had higher cell counts. Sa et al., 1995. The fact that an inhibitor of DNA synthesis designed to kill human cells would inhibit lymphocyte growth was not mentioned. 3. The blunt result that AZT prophylaxis reduced survival from 3 to 2 years, and caused wasting syndrome, cryptosporidiosis, and cytomegalovirus infection, almost exclusively in AZT-treated AIDS patients, was interpreted like this: the study of patients who progress from primary HIV infection to AIDS without receiving medical intervention gives insights into the effects of medical intervention on presentation and survival after developing an AIDS-defining illness. But the nature of these insights was not revealed by the authors. Poznansky et al., 1995. 4. The largest test of AIDS prophylaxis with AZT of its kind, the Concord trial, found a 25% higher mortality in AZT recipients than in untreated controls. In view of this, Seligman et al. Reached the conservative conclusion, the results of Concord do not encourage the early use of zidovudine AZT in symptom-free HIV-infected adults. Seligman et al., 1994. 5 years after introducing AZT prophylaxis to several hundred thousands of healthy HIV positives, Paul Valberting of the University of California at San Francisco, Anthony Fauci of the National Institute of Allergy and Infectious Diseases and over 100 scientific collaborators now publish in the New England Journal of Medicine, zidovudine does not significantly prolong either AIDS-free or overall survival. These results do not encourage the routine use of zidovudine. Valberting et al., 1995. In an accompanying editorial, Time to Hit HIV, Early and Hard, the journal makes the forward recommendation to treat HIV infection with AZT and other experimental drugs before antiviral immunity restricts the virus to chronic latency. Ho. 1995. The article suggests that current AZT prophylaxis is too little too late. This is said although the ineffectiveness of the proposed early and hard treatment is known since 1993, Talkers et al., 1993. 6. The occurrence of 8 serious birth defects, 8 spontaneous abortions and 8 therapeutic abortions among 104 pregnancies treated with AZT is interpreted as not proving safety, thus lending TINA's support to the use of this drug. Kumar, Hughes, and Kurana, 1994. AZT must be considered the most toxic among legal public health threats available to healthy persons, much more toxic than alcohol and tobacco. For this reason I have termed AZT AIDS by prescription. Duesberg, 1992, see Chapter 6. Even Burroughs Wellcome, the manufacturer of AZT, makes that same assessment, but expresses it in different words. It was often difficult to distinguish adverse events possibly associated with zidovudine AZT administration from underlying signs of HIV disease. Physician's Desk Reference, 1994. 2. Educational damage. Since HIV, but not drugs, is thought to cause AIDS, the HIV/AIDS establishment educates the public to use clean needles for the injection of unsterile, street drugs and to wear condoms for sex under the influence of aphrodisiac drugs. Institute of Medicine, 1988, San Francisco Project Inform, 1992, Bendit and Jasny, 1993, National Institute of Allergy and Infectious Diseases, 1994. However, the disregard, in fact explicit dismissal, of drug toxicity by the AIDS establishment, Weiss and Jaff, 1990, Asher et al., 1993, Duesberg, 1993d, Maddox, 1993a, Schechter et al., 1993b, Schechter et al., 1993c, Cohen, 1994a, encourages recreational drug use because it eliminates the fear of drug toxicity. A popular joke illustrates this point. 2 junkies are reminded by a friend not to share a syringe full of cocaine. Their response: we wear condoms and use a clean needle. Yet long-term use of recreational drugs, including cocaine, heroin, amyl, and isobutyl nitrite inhalants, amphetamines, and others has been documented in numerous studies to cause exactly the same diseases that are now blamed on HIV. Haverkas and Doherty, 1988, Stoneburner et al., 1988, Lerner, 1989, Duesberg, 1992, see Chapter 6. The list of drug-induced diseases, established long before the discovery of HIV, reads like a catalogue of AIDS-defining diseases: weight loss, fever, dementia, tuberculosis, oral thrush, pneumonia, diarrhea, mouth infections, night sweats, and many others. See below. Lerner, 1989, Duesberg, 1992. 3. Psychological damage. According to the HIV/AIDS establishment, nearly all HIV-infected, Healthy persons are claimed to die from AIDS on average 10 years after infection by HIV. Institute of Medicine, 1988, Garza, Drottman, and Jaff, 1994, National Institute of Allergy and Infectious Diseases, 1994, Thomas Jr., Mullis, and Johnson, 1994. In view of this, 1 million HIV-positive but healthy Americans, and 17 million HIV-positive but healthy humans on this planet, Merson, 1993, National Institute of Allergy and Infectious Diseases, 1994, World Health Organization, 1995, are subjected to a multiplicity of psychological and sociological pressures. Anonymous, 1992, Duesberg, 1992, Schmaltz, 1992b, Schmaltz, 1992a, Miami Herald, 1994, Yarbo, 1994. They are given a death sentence by the medical establishment for being HIV positive, they are denied coverage by health insurance companies, they lose their jobs and social status, they are denied entrance visas to many countries including the US, they are denied employment by the US Army, and worst of all they are pressured to accept the toxic AZT therapy. All of this, only because they have made antibodies against a virus that is presumed to cause AIDS. If they refuse to submit to these pressures, they are charged with denial of the HIV/AIDS hypothesis by the AIDS establishment. Moss, Osmond, and Bicchetti, 1988, San Francisco Project Inform, 1992. In sum, the public health record of the HIV/AIDS hypothesis in America adds up to a staggering deficit, for $35 billion. Duesberg, 1994b, AIDS Weekly, 1995, Gutknecht, 1995, there is no cure, no vaccine, no effective prevention, hundreds of thousands are subjected to psychological pressures resulting from positive HIV tests, and several million American drug addicts are denied available information that recreational drugs cause AIDS. Defining and other diseases, drug strategies, 1995, and about 150,000 are subjected annually to AZT poisoning, many just for being HIV positive, not for having AIDS. Duesberg, 1992. 7. The drug AIDS hypothesis. Given no evidence for infectious AIDS, the reasons for the original lifestyle hypothesis, and the logic of Sherlock Holmes that when you have eliminated the impossible, whatever remains however improbable must be the truth. AIDS must be non-infectious. In view of this, I propose that all AIDS diseases in America and Europe that exceed their long-established, normal backgrounds are caused by the long-term consumption of recreational drugs and by AZT and its analogues. Hemophilia AIDS, transfusion AIDS, and the extremely rare AIDS cases of the general population reflect the normal incidence plus the AZT-induced incidence of these diseases under a new name. African AIDS is a new name for old diseases caused by malnutrition, parasitic infections, and poor sanitation. Duesberg, 1991, Duesberg, 1992, Duesberg, 1994a, see chapters 6 and 9. Indeed, the recreational drug use epidemics, that started in America and Europe during the Vietnam War, are the only new health risk of the Western world since World War II. Since its beginnings, the drug use and AIDS epidemics in the US and Europe have coincided both epidemiologically and chronologically. Duesberg, 1992. About 33% of all American AIDS patients, nearly all heterosexual AIDS patients, are intravenous drug users. Centers for Disease Control and Prevention, 1994b. Over 60% are male homosexuals who have used psychoactive and aphrodisiac drugs orally such as nitrite inhalants, amphetamines, cocaine, and phenylcyclidine, Table 2. Many of these recreational drug users and most of the few AIDS patients who have not used recreational drugs have used AZT and cytotoxic DNA chain terminators as anti-HIV drugs. See below. Duesberg, 1992, Duesberg, 1994a. Allowing a latent period of 10 years for chronic recreational drug use to cause AIDS, the beginning of the American drug use epidemics in the late 1960s and 1970s predicts exactly the origin of AIDS in the 1980s. Unlike the virus AIDS hypothesis, the drug hypothesis has a plausible chemical and experimentally testable basis. The recreational drugs postulated to cause AIDS have strong biochemical and psychoactive effects every time they are taken. The reason for their popularity, see Chapter 6. By contrast, HIV is latent, and neither chemically nor clinically detectable in HIV antibody positives with and without AIDS. Despite 11 years of unprecedented research efforts, no biochemical evidence has been found in support of the HIV/AIDS hypothesis. Cohen 1995. The specific toxicity and dosages of recreational and medical drugs used by American and European AIDS risk groups can explain all AIDS diseases. Duesberg, 1992. AIDS drugs are either indirectly toxic or cytotoxic or genotoxic and cytotoxic. 1. Indirectly toxic. Cocaine, amphetamines, and heroin are indirectly immunotoxic. All three function as catalysts in the human body. Cocaine and heroin are natural compounds and amphetamines are synthetic adrenalines first developed in Germany during World War II to suppress fatigue and anxiety in pilots and tank commanders. Vey and Rosen, 1983. Indirect toxicity is the result of malnutrition and insomnia, which in turn are consequences of drug-induced suppression of appetite and fatigue. Leyen et al., 1984, Lerner, 1989, Pillai, Nair, and Watson, 1991, Duesberg, 1992, Larratt and Zierler, 1993, Meintjes et al., 1993, Sidonik, 1994. These problems are compounded by poverty due to the enormous costs of illicit drugs. Direct, long-term pathogenic effects of cocaine and heroin have not been studied owing to the general disregard of drug toxicity. Duesberg, 1992. 2. Cytotoxic and genotoxic. Nitrite inhalants are cytotoxic, and thus are immunotoxic in animals and humans. Godard et al., 1982, Haverkas and Doherty, 1988. A recreational dose of 1 ml per day, Haverkas and Doherty, 1988, Duesberg, 1992, corresponds to about 15 ppm in a 75 kg person, and corresponds to 107 nitrite molecules for every one of the 1014 cells in the human body. The cytotoxicity of nitrites on the epithelial tissues of the lung are enhanced by the toxins of cigarette smoke, which also suppresses the immune system. Neiman et al., 1993. In addition, nitrite inhalants are among the best established mutagens and carcinogens. National Research Council, 1982, Lewis, 1989, Winter, 1989, Mervish et al., 1993. In view of the toxicity of nitrite inhalants, a prescription requirement was instated by the U.S. Food and Drug Administration in 1969, Newell et al., 1985a, and because of an AIDS link, Cox, 1986, the sale of nitrites was banned by the U.S. Congress in 1988, Public Law 100-690, Haverkas, 1990, and by the Crime Control Act of 1990, Duesberg, 1992. Moreover, the U.S. Food and Drug Administration limits nitrites as food preservatives to less than 200 ppm, because of direct toxicity and because they have been implicated in an increased incidence of cancer. Lewis, 1989, National Research Council, 1982. 3. Genotoxic cytotoxic. AZT, DDI, and other DNA chain terminators are directly toxic by killing all growing cells, in particular the fastest growing ones, the hematopoietic and epithelial cells. Fig. 2, Merck Research Laboratories, 1992, Chu and Duesberg, 1995. In addition, AZT prevents mitochondrial DNA synthesis in non-growing cells, such as neurons or muscles, and can be carcinogenic by mutating cells. Pluta et al., 1990, Duesberg, 1992, Parker and Cheng, 1994. The key to the drug hypothesis is that only long-term consumption causes irreversible AIDS-defining diseases. Occasional or short-term recreational drug use causes reversible diseases or no diseases at all. With drugs, the dose is the poison. Yet, most studies investigating the effects of recreational drugs are concerned with their short-term psychoactive rather with their long-term clinical effects. Duesberg, 1992. For example, it takes 20 years of smoking to acquire irreversible lung cancer or emphysema, and 20 years of drinking to acquire irreversible liver cirrhosis. In contrast to drugs, infectious agents are self-replicating toxins. By multiplying exponentially in the body, infectious agents may generate sufficient doses of toxic substances to cause diseases within days or weeks. Since currently no experiments are being done in America to test the drug hypothesis, I have evaluated the drug AIDS hypothesis on the basis of its predictions. In contrast to the HIV/AIDS hypothesis, the drug hypothesis can predict all parameters of American/European AIDS. 9. The drug hypothesis predicts the American/European AIDS epidemic completely. 9.1. AIDS is restricted to intravenous and oral users of recreational drugs and of AZT, because drugs cause AIDS. Since 1981, 94% of all American AIDS cases have been from risk groups who had used such drugs. Centers for Disease Control and Prevention, 1994C. About one-third of these were intravenous drug users, Centers for Disease Control, 1993, and two-thirds were male homosexuals, Centers for Disease Control and Prevention, 1994C, Centers for Disease Control and Prevention, 1994A, who had used oral recreational drugs and AZT. Duesberg, 1992, Asher et al., 1993, Duesberg, 1993C, Duesberg, 1993A, Duesberg, 1993D, Park, 1993, Schechter et al., 1993b. HIV-positive hemophiliacs and transfusion recipients also receive AZT as an antiviral drug. Duesberg, 1992, Duesberg, 1995b. However, a small percentage of hemophiliacs annually develop a specific subset of AIDS-defining immunodeficiency diseases, mostly pneumonia and candidiasis, only from the long-term transfusion of foreign proteins that contaminate commercial factor VIII. Duesberg, 1992, Duesberg, 1995b, see Chapter 11. European AIDS also correlates with drug consumption. Duesberg, 1992, see Chapter 6. 9.2, American/European AIDS predominantly affects adult males, because they are the predominant users of recreational drugs and AZT. The CDC reports that 87% of all American AIDS patients are males. Centers for Disease Control and Prevention, 1994C. This number is the sum of the following constituents. The National Institute on Drug Abuse and the Bureau of Justice Statistics report that over 75% of hard, recreational drugs are consumed intravenously by males. Duesberg, 1992, see Chapter 6. According to the federally supported Drug Strategies Program, women account for the fastest growing population in jails and prisons, in large part because of drug offenses. Drug Strategies, 1995. Therefore, the CDC reports that women are now the fastest growing AIDS risk group. Centers for Disease Control, 1994, Centers for Disease Control and Prevention, 1994A. The CDC and independent investigators report that nearly all male homosexuals with AIDS and at risk for AIDS are long-term users of oral drugs such as nitrite inhalants, ethyl chloride inhalants, amphetamines, cocaine, and others to facilitate sexual contacts, particularly anal intercourse. Lifson et al., 1990, Duesberg, 1992, Asher et al., 1993, Duesberg, 1993d, Schechter et al., 1993a, Schechter et al., 1993c. The drug use of male homosexuals with AIDS or at risk for AIDS reported by the CDC, Jaff et al., 1983, Darrow et al., 1987, Lifson et al., 1990, and others, Asher et al., 1993, Duesberg, 1993d, Schechter et al., 1993c, Ellison, Downey, and Duesberg, 1995, as of 1983 is listed in Table 2. Ostro reported that nitrite inhalant use in a cohort of over 5,000 male homosexuals from Chicago, Baltimore, Los Angeles, and Pittsburgh showed a consistent and strong cross-sectional association with anal sex. Ostro, 1994. In addition, many HIV-positive homosexuals are prescribed AZT as an antiviral drug. Duesberg, 1992, Duesberg, 1993d, Ellison, Downey, and Duesberg, 1995. Since intravenous drug users, who are 75% male, make up one-third of all AIDS patients, and male homosexuals make up almost two-thirds of all American AIDS patients, the drug hypothesis explains why 87% of all American AIDS patients are males. 9.3. Pediatric AIDS occurs because of maternal drug addiction. Indeed, about 80% of pediatric AIDS cases in America and Europe are children born to mothers who were intravenous drug users during pregnancy, see below. 5, and 8, Mock et.al., 1987, European Collaborative Study, 1991, Duesberg, 1992. The remainder reflects the normal low incidence of AIDS-defining diseases among newborns. 9.4. American AIDS is new and increasing steadily, because the American drug epidemic is new and increasing steadily. In the U.S., recreational drug use is epidemiologically new, as it has increased over the last decades from statistically undetectable levels to epidemic levels at about the same rate as AIDS. Duesberg, 1992. For example, cocaine consumption increased 200-fold from 1980 to 1990, based on cocaine seizures that increased from 500 kilograms in 1980 to 100,000 kilograms in 1990, Duesberg, 1992, see Chapter 6. During the same time, cocaine-related hospital emergencies increased from 3,296 cases in 1981 to 80,355 cases in 1990, and to 119,843 in 1992 and to over 120,000 in 1993, Duesberg, 1992, Meddies, 1994, Drug Strategies, 1995, Fig 1b. In the last 3 years, the increase of cocaine consumption has slowed down at the expense of increases in heroin consumption, which were accompanied by increases in heroin-related hospital emergencies. Getman, 1994, Meddies, 1994, Drug Strategies, 1995. Heroin-related hospital emergencies doubled, from over 30,000 in 1990 to over 60,000 in 1993, Fig 1b, Drug Strategies, 1995. Amphetamine consumption has increased 100-fold from 1980 to 1990, Bureau of Justice Statistics, 1991. Non-scientific reports describe new upsurges in the consumption of amphetamines, Sedonic, 1994, and the gay drug, nitrite inhalants, Merkin, 1995, among male homosexuals. According to a recent report from the National Institute on Drug Abuse and the CDC, nitrite use has increased in the 1990s in gay men in Chicago and San Francisco after a decline in the 1980s, Haverkas and Drottman, 1995. Drug offenders are now the largest and fastest growing category in the federal prisons population, accounting for 61% of the total, compared with 38% in 1986. The number of federal drug offenders increased from about 5,000 in 1980 to about 55,000 in 1993. In 1993, between 60 and 80% of the 12 million prisoners in the U.S. had been on illicit drugs. Drug Strategies, 1995. The German Rauchgiftbilanz reports an 11.2% increase in the consumption of illicit recreational drugs in 1994 compared to 1993, Rauchgiftbilanz 1994, 1995. Consider a grace period of about 10 years to achieve the dosage needed to cause irreversible disease, and you can date the origin of AIDS in 1981 as a consequence of the drug use epidemic that started in America in the late 1960s during the Vietnam War. Indeed, AIDS increased from a few dozen cases annually in 1981 to about 100,000 in 1993, Fig 1a, Centers for Disease Control and Prevention, 1994c. Note the parallelisms between the spread of AIDS and the spread of cocaine and cocaine-related hospital emergencies since 1981, Fig 1a and b, and the contrast with the non-spread of HIV, the hypothetical cause of AIDS, since 1984, Fig 1a. Thus both, the newness and the increase of the AIDS epidemic are predictable by the drug AIDS hypothesis. The growth of the epidemic has been accelerated by AZT. Since its introduction in 1987, AZT is now prescribed to about 200,000 HIV positives worldwide, Duesberg, 1992, Thomas, 1995. 9.5. Only a small fraction of drug users develop AIDS, because only the highest cumulative drug doses cause irreversible diseases. The cumulative total of 401,749 American AIDS cases since 1981 that were reported in June 1994, Centers for Disease Control and Prevention, 1994c, have been recruited from a much larger reservoir of drug users. There are currently between 3 drug strategies, 1995, and 8 million cocaine addicts, Duesberg, 1992, and 0.6 million heroin addicts in the U.S., drug strategies, 1995. In 1980, 5 million Americans had used nitrite inhalants. In 1989, 100 million doses of amphetamines were consumed in the U.S., Duesberg, 1992. According to a 1994 survey of the National Institute on Drug Abuse, more than 5%, 221,000, of the 4 million women who give birth each year use illicit drugs during their pregnancy. Drug strategies, 1995. These mothers are the reservoir from which most of the 1,017 pediatric AIDS cases reported in the U.S. in 1994 were recruited, Centers for Disease Control and Prevention, 1994b, C10. Unfortunately, scientific documentation of recreational drug use is extremely sporadic and inaccessible, not only because these drugs are illegal, but more importantly because the medical scientific community is totally uninterested in drugs as a cause of AIDS, see above. In addition, about 150,000 HIV-positive Americans were on AZT between 1992 and 1995, Duesberg, 1992, Thomas, 1995. Probably because drug toxicity is generally ignored, there are also no national statistics available on how many HIV-positive Americans are on AZT and other anti-HIV drugs, that, like AZT, are designed to kill human cells, Duesberg, 1992. The small percentage of AIDS patients among the many American drug users reflects the highest lifetime dose of drug use, just like the lung cancer and emphysema patients reflect the highest lifetime tobacco dose among the 50 million smokers in the U.S. The long latent period of HIV is a euphemism for the time needed to accumulate the drug dosage that is sufficient for AIDS. Indeed, it takes about 10 years of injecting heroin and cocaine to develop weight loss, tuberculosis, bronchitis, pneumonia, and other drug-induced diseases, Leyen et al., 1984, Schuster, 1984, Savona et al., 1985, Donahoe et al., 1987, Espinoza et al., 1987, Weber et al., 1990. The time lag from initiating a habit of inhaling nitrites to acquire Kaposi's sarcoma has been determined to be 7 to 10 years, Newell et al., 1985a, Birrell et al., 1990, Lifson et al., 1990, Duesberg, 1992. Blaming Kaposi's sarcoma on HIV after inhaling carcinogenic nitrites for 10 years is like blaming lung cancer and emphysema on a slow virus after smoking 2 packs of cigarettes a day for 20 years. AZT, at the currently prescribed high doses of 0.5 to 1.5 grams per person per day, causes many of the above described AZT-specific diseases faster than recreational drugs, i.e. within weeks or months after administration, Duesberg, 1992, Lewis Thornton, 1994. 9.6. Risk group specific AIDS diseases occur because of risk group specific drugs. Group specific drug use explains the following risk group specific AIDS diseases. I. Kaposi's sarcoma specific for male homosexuals. Kaposi's sarcoma as an AIDS diagnosis is 20 times more common among homosexuals who use nitrite inhalants than among AIDS patients who are intravenous drug users, or hemophiliacs. Haverkos and Doherty, 1988, Birol et al., 1990. Due to the carcinogenic potential, nitrites were originally proposed as causes of Kaposi's sarcoma. Marmer et al., 1982, Haverkos et al., 1985. Aggressive and life-threatening Kaposi's sarcoma, particularly pulmonary Kaposi's sarcoma, is exclusively observed in male homosexuals. Sloan, Kumar, and Pierce, 1993, Maduri et al., 1986, Garay et al., 1987, Gill et al., 1989. Since the lungs are the primary site of exposure to nitrite inhalants, the evidence that up to 32% of Kaposi's sarcomas of homosexual men can be diagnosed as pulmonary Kaposi's sarcoma, Gill et al., 1989, Irwin and Kaplan, 1993, lends additional support to the nitrite Kaposi's sarcoma hypothesis. Pulmonary Kaposi's sarcoma has never been described by Moritz Kaposi, nor anywhere else prior to the AIDS epidemic. Kaposi, 1872. It appears that the nitrite-induced AIDS Kaposi's sarcoma and the classic Kaposi's sarcomas are entirely different cancers under the same name. The HIV-associated Kaposi's sarcomas observed in male homosexuals are aggressive and life-threatening. Sloan, Kumar, and Pierce, 1993, fatal within 8 to 10 months after diagnosis, and often located in the lung. Maduri et al., 1986, Garay et al., 1987, Gill et al., 1989, Irwin and Kaplan, 1993. The classic indolent and chronic Kaposi's sarcomas are diagnosed on the skin of the lower extremities and hardly progress over many years. Maduri et al., 1986, Drottman and Haverkos, 1992, Cohen, 1994a. Maduri et al. Point out that the pulmonary involvement by the neoplasma has been an unusual clinical finding in the Kaposi's sarcomas of male homosexuals compared to all classic Kaposi's sarcomas. Maduri et al., 1986. Nevertheless, the distinction between classic and AIDS Kaposi's sarcoma is hardly ever emphasized and may have escaped many observers due to the difficulty in premortem diagnosis, because pulmonary Kaposi's sarcoma was indistinguishable from opportunistic pneumonia. Garay et al., 1987. The immunotoxicity and cytotoxicity of nitrites also explains the proclivity of male homosexual nitrite users for pneumonia, which is the most common AIDS disease in the US and Europe. Haverkos and Doherty, 1988, Duesberg, 1992, Table 1, Chapter 6. Moreover, the immunotoxins and cytotoxins of cigarette smoke explain why in two groups of otherwise matched HIV-positive male homosexuals cigarette smokers developed pneumonia twice as often as non-smokers over a period of 9 months. Neiman et al., 1993. 2. High mortality of intravenous drug users. Intravenous drug users suffer from long-term malnutrition and insomnia, which are primary causes of immunodeficiency worldwide. Seligman et al., 1984. This explains the tuberculosis, pneumonia, and weight loss that are typical of these risk groups. Layan et al., 1984, Stoneburner et al., 1988, Pillai, Nair, and Watson, 1991, Duesberg, 1992, Meintjes et al., 1993. Injection of unsterile drugs combined with immunodeficiency also cause septicemia and endocarditis that are common in AIDS patients who are intravenous drug users. Duesberg, 1992. As a result, intravenous drug users only achieve a very low average age. A German study found the average age at death to 29.6 years for HIV-free and 31.5 years for HIV-positive addicts, Lockman et al., 1995, and an American study showed that both HIV-positive and negative intravenous drug users died from the same diseases, Stoneburner et al., 1988. 3. Low birth weight and mental retardation of AIDS babies. 80% of American/European babies with AIDS are born to mothers who were intravenous drug users during pregnancy. They acquire low birth weight, mental retardation, and immunodeficiency through maternal drug use. Duesberg, 1992, Drug Strategies, 1995. The B-cell deficiencies and certain bacterial infections that are both only considered AIDS-defining in children are also specific expressions of their acquired immunodeficiency. Centers for Disease Control, 1987, Centers for Disease Control and Prevention, 1992, Duesberg, 1992. 4. Anemia and wasting of AZT recipients. Anemia, leukopenia, pancytopenia, diarrhea, weight loss, hair loss, impotence, Duesberg, 1992, hepatitis, Freiman et al., 1993, and pneumocystis pneumonia, Sa et al., 1995, that are observed in recipients of AZT and other DNA chain terminators, are predictable consequences of the cytotoxicity of these drugs, see Chapter 6. In addition, non-renewal of mitochondrial DNA causes muscle atrophy, hepatitis, and dementia, and carcinogenic activity causes cancers such as lymphoma in AZT recipients, Pluta et al., 1990, Duesberg, 1992, MacLeod and Hammer, 1992, Freiman et al., 1993, Beischeler et al., 1994, Parker and Cheng, 1994, Physician's Desk Reference, 1994. Compared to untreated controls, AZT recipients die 2.4 times more often, Goddard et al., 1994, 25% more often, Seligman et al., 1994, or live only 2 years instead of 3 years with AIDS, Poznansky et al., 1995. 9.7. Non-correlations between HIV and AIDS, because drugs, not HIV, cause AIDS. I. Long-term survivors or non-progressors. Persons infected by HIV for more than the 10-year latent period from HIV to AIDS who are studied by HIV researchers are termed long-term survivors and more recently non-progressors, Scolaro, Durham, and Pychanik, 1991, Learmont et al., 1992, Cao et al., 1995. David Ho et al. Recently gave a key to long-term survival. None had received antiretroviral therapy, Cao et al., 1995. Likewise, Alvaro Munoz reported that not one of the long-term survivors of the largest federally funded study of male homosexuals at risk for AIDS, the MAX study, had used AZT, Munoz, 1995. And several survey studies document that in addition to abstaining from antiviral drugs, long-term survivors are those who have given up or never taken recreational drugs. Wells, 1993, Gavzer, 1995, Ruth Bernstein, 1995b. Indeed, the vast majority of HIV positives are long-term survivors. Worldwide, they number 17 million, including 1 million HIV positive but healthy Americans and 0.5 million HIV positive but healthy Europeans. Merson, 1993, World Health Organization, 1995. Most of these have been HIV positive for at least 10 years now, because their numbers have not changed since the time between 1984 to 1988, when the epidemic of HIV testing began in the respective countries. Duesberg, 1992. Only about 6%, or 1,025,073, of these 17 million HIV positives have developed AIDS diseases since AIDS statistics are kept. World Health Organization, 1995. Since no more than 6% of HIV carriers worldwide have developed AIDS in 7 to 10 years, the annual AIDS risk of an HIV carrier is less than 1% per year. However, even this low figure is not corrected for the normal occurrence of the 29 AIDS-defining diseases in HIV-free controls. There is no evidence that HIV-positive people who are not drug users have a higher morbidity or mortality than HIV-free controls. Duesberg, 1995a, see Chapter 10. 2. Intravenous drug users and male homosexuals losing their T cells prior to HIV infection. Prospective studies of male homosexuals using psychoactive and sexual stimulants have demonstrated that their T cells may decline prior to infection with HIV. For example, the T cells of 37 homosexual men from San Francisco declined steadily prior to HIV infection for 1.5 years from over 1,200 to below 800 per L, Lang et al., 1989. In fact, some had fewer than 500 T cells 1.5 years before seroconversion, Lang et al., 1987. Although recreational drug use was not mentioned in these articles, other studies of the same cohort of homosexual men from San Francisco described extensive use of recreational drugs including nitrites. Darrow et al., 1987, Moss, 1987, Asher et al., 1993, Duesberg, 1993d, Ellison, Downey, and Duesberg, 1995. Likewise 33 HIV-free male homosexuals from Vancouver, Canada, had acquired immunodeficiency prior to HIV infection, Marion et al., 1989. Again, this study did not mention drug use, but in other articles the authors reported that all men of this cohort had used nitrites, cocaine, and amphetamines. Archibald et al., 1992, Duesberg, 1993f, Schechter et al., 1993c. The largest study of its kind reported that about 450, 16% of 2,795, HIV-free, homosexual American men of the MAX cohort from Chicago, Baltimore, Pittsburgh, and Los Angeles had acquired immunodeficiency, having less than 600 T cells per L, prior to HIV infection, Caslow et al., 1989. Many HIV-positive and negative men of this cohort had essentially the same degree of lymphadenopathy, although seropositive men had a significantly higher mean number of involved lymph node groups than seronegative men, 5.7 compared to 4.5 nodes, p 0.005. The numerical difference in the means is not striking. Caslow et al., 1987. According to previous studies on this cohort, 71% of these men had used nitrite inhalants, in addition to other drugs. Caslow et al., 1987, 83% had used one drug, and 60% had used two or more drugs during sex in the previous 6 months. Ostro et al., 1990. Another study of the same cohort observed that the risk of developing AIDS correlated with the frequency of receptive anal intercourse prior to and after HIV infection. Fair et al., 1992. Other studies have shown that receptive anal intercourse correlates directly with the use of nitrite vasodilators. Haverkas and Doherty, 1988, Duesberg, 1992, Park, 1993. Thus, in male homosexuals at risk for AIDS, AIDS often precedes infection by HIV, not vice versa. Since the cause must precede the consequence, drug use remains the only group-specific choice to explain acquired immunodeficiencies prior to HIV. If male homosexuality were to cause immunodeficiency, about 10% of the adult American male population should have AIDS. Duesberg, 1992, Seidman and Reeder, 1994. Prospective studies of intravenous drug users also document T-cell losses prior to infection by HIV. For example, among intravenous drug users in New York, the relative risk for seroconversion among subjects with one or more CD4 T-cell count 500 cells/L compared with HIV-negative subjects with all counts greater than 500 cells/I was 4.53. Desjarlais et al., 1993. A similar study from Italy showed that a low number of T cells was the highest risk factor for HIV infection. Nicolosi et al., 1990. Again, a decrease in T cells is a risk factor for HIV infection, and not vice versa. This confirms the hypothesis that HIV is a marker of drug consumption, rather than the cause of AIDS. C4 The more drugs are consumed intravenously or for sex, the higher is the risk of HIV infection. Duesberg, 1992. 3. HIV-free AIDS. One summary of the AIDS literature describes over 4,621 clinically diagnosed AIDS cases who were not infected by HIV. Duesberg, 1993f. See Chapter 7. Additional cases are described that were not in this summary. Caslow et al., 1987, Lang et al., 1987, European Collaborative Study, 1991, Weiss et al., 1992, Ellison, Downey and Duesberg, 1995, Moore and Chong, 1995. They include intravenous drug users, male homosexuals using aphrodisiac drugs like nitrite inhalants, and hemophiliacs developing immune suppression from long-term transfusion of foreign proteins contaminating factor VIII. Duesberg, 1993f, Duesberg, 1995b. Each of these non-correlations between HIV and AIDS are predicted by the hypothesis that recreational drugs and other non-contagious risk factors cause AIDS. 9.8. Discontinuation of drug use either stabilizes or cures AIDS and other diseases even in HIV positives. AZT. 10 out of 11 HIV positive, AZT-treated AIDS patients recovered cellular immunity after discontinuing AZT in favor of an experimental vaccine, Scolaro, Durham and Pychynek, 1991. 2 weeks after discontinuing AZT, 4 out of 5 AIDS patients recovered from myopathy, Till and McDonnell, 1990. 3 of 4 AIDS patients recovered from severe pancytopenia and bone marrow aplasia 4 to 5 weeks after AZT was discontinued, Gill et al., 1987. 2. Heroin/cocaine. The incidence of AIDS diseases among HIV-positive intravenous drug users over 16 months was 19%, 23/124, and only 5%, 5/93, among those who stopped injecting drugs, Weber et al., 1990. The T-cell counts of HIV-positive intravenous drug users from New York dropped 35% over 9 months, compared to HIV-positive controls who had stopped injecting, Desjarlais et al., 1987. 3. Recreational drugs and AZT. The health of male homosexuals is stabilized or even improved by avoiding recreational drugs. For example, in August 1993 there was no mortality during 1.25 years in a group of 918 British HIV-positive homosexuals who had avoided the experimental medications on offer and chose to abstain from or significantly reduce their use of recreational drugs, including alcohol. Wells, 1993. Assuming an average 10-year latent period from HIV to AIDS, the virus AIDS hypothesis would have predicted at least 58, 918/10x 1.25x 50%. AIDS cases among 918 HIV positives over 1.25 years. Indeed, the absence of mortality in this group over 1.25 years corresponds to a minimal latent period from HIV to AIDS of over 1,148, 918x 1.25 years. On July 1, 1994 there was still not a single AIDS case in this group of 918 HIV-positive homosexuals. J. Wells, London, Personal Communication. The T-cells of 29% of 1,020 HIV-positive male homosexuals and intravenous drug users in a clinical trial even increased over 2 years. Hughes et al., 1994. These HIV positives belonged to the placebo arm of an AZT trial for AIDS prevention and thus were not treated by AZT. It is probable that under clinical surveillance the 29% whose T-cells increased, despite HIV, have given up or reduced immunosuppressive recreational drug use in the hope that AZT would prevent AIDS. 4. AIDS babies, born to drug-addicted mothers, recover after birth. HIV-positive babies, born to mothers who were intravenous drug users during pregnancy, provide the best examples for the prediction that termination of drug use prevents, or cures AIDS despite the presence of HIV. For example, Blanche et al. Have observed for 3 years 71 HIV-positive newborns who had shared intravenous drugs with their mothers prior to birth. 10 of these children developed encephalopathy and AIDS-defining diseases, of which 9 died during their first 18 months of life. The study points out that the risk of a newborn to develop AIDS was related directly with the severity of the disease in the mother at the time of delivery. Based on the severity of their symptoms, about 60% of the children were treated prophylactically, but apparently briefly, with AZT for at least 1 month, and 50% were treated with sulfa drugs. Blanche et al., 1994. Despite HIV, 61 of the 71 HIV-positive children either developed only intermittent diseases from which they recovered during their first 18 months or developed no disease at all during the 3 years of observation. The T cells of these children increased after birth from low to normal levels despite the presence of HIV. A very similar picture emerges from a collaborative European study of HIV-positive newborns, the European Collaborative Study, 1994. The study reports that about 20% of the HIV-positive children had died or developed long-term AIDS during the first year after birth, and another 20% during the second and third year. About 10% of the children were treated with zidovudine AZT before 6 months of age and 40% by 4 years, the European Collaborative Study, 1994. But over 60% of congenitally infected children proved to be healthy up to 6 years after birth despite the presence of HIV. Most of these had experienced transient AIDS diseases, such as pneumonia, bacterial infections, candidiasis, and cryptosporidial infection during the first year after birth. Although this study does not even mention the health risks of the mothers, previous reports from the European Collaborative Study Group have documented that nearly all children were born to mothers who are intravenous drug users. Mock et al., 1987, Duesberg, 1992. In 1991, the European Collaborative Study Group reported that 80% of the children with pediatric AIDS were born to mothers who were intravenous drug users. European Collaborative Study, 1991. The 1991 study further points out that children with drug withdrawal symptoms were most likely to develop diseases, and that children with no withdrawal symptoms but whose mothers had used recreational drugs in the final 6 months of pregnancy were intermediate in their risk to develop diseases. European Collaborative Study, 1991. According to the HIV hypothesis, every infected baby should have developed AIDS and progressively lost T cells, and according to an HIV plus cofactor hypothesis, at least all those with intermittent diseases should have progressed to AIDS. This was not observed. According to the drug hypothesis, the AIDS risk of the children is a function of the drugs consumed. Those who received the highest doses of drugs before birth would have acquired irreversible diseases, and those who acquired diseases from sublethal thresholds would be able to recover after cessation of maternally administered drugs. Indeed, both, the European Collaborative Study Group and Blanche et al. Show that the majority of children gained T cells and recovered from transient diseases after discontinuation of maternal drug input despite the presence of HIV. The children's risk for AIDS was related directly with the severity of the disease in the mother. Blanche et al., 1994, which is an expression for the extent of drug consumption by the mother. Moreover, the harm of maternal drug consumption to sick babies was compounded after birth, because prophylactic treatment with sulfamethoxazole and zidovudine AZT was started earlier and was more frequent among the 16 children born to mothers with Class 4 disease, AIDS. Blanche et al., 1994. The Blanche study did include mothers with AIDS who were not intravenous drug users. The European Collaborative Study Group reports that 10 to 40% of HIV-positive children were treated with AZT. It follows that discontinuation of recreational and antiretroviral drug use stabilizes and even cures AIDS in HIV-positive persons. Likewise, the T cells of HIV-positive hemophiliacs increase after removal of immunosuppressive foreign proteins from their factor VIII therapy. Duesberg, 1995, see Chapter 11, and the T cells of African HIV-positive tuberculosis patients increase after standard anti-TB treatment and improved nutrition. Martin et al., 1995. In sum, the drug AIDS hypothesis correctly predicts all aspects of American/European AIDS, while the HIV hypothesis predicts none. 10. A possible solution at last. Testing the drug hypothesis should have a very high priority in AIDS research, because this hypothesis makes verifiable predictions. Cohen, 1994a, Danoon, 1995. Drug toxicity could be tested experimentally in animals, and in human cells in tissue culture. In addition, drug toxicity could be tested epidemiologically in humans who are addicted to recreational drugs or are prescribed AZT. Such tests could be conducted at a fraction of the cost that is now invested in the HIV hypothesis. If the drug hypothesis proved to be correct, AIDS would be an entirely preventable disease. Here is how. 1. AZT use would be banned immediately. 2. AIDS from illicit recreational drugs would be reduced or prevented by education against drug use. Hemophilia AIDS would be prevented by the use of pure factor VIII, Chapter 10. 3. AIDS therapy would be achieved by termination of recreational drug use and treating AIDS diseases for their specific causes, e.g. tuberculosis with antibiotics, Kaposi's sarcoma with conventional cancer therapy, and weight loss with good nutrition rather than treating each of these unrelated diseases with the same cell killer AZT. In addition to saving about 100,000 lives per year from AIDS, the drug hypothesis could save the American taxpayer up to $20 billion annually. Currently, the federal government spends annually $7.5 billion on AIDS treatment, research, and education. AIDS Weekly, 1995, Gutknecht, 1995. And the federal drug budget currently costs $13 billion, mainly for supply control, interdiction, methadone treatment, and education. Drug Strategies, 1995. But neither AIDS education nor drug education ever target the health effects of long-term drug use. They focus on the legal and social consequences of drug use and on the effects of drug use on transmission of HIV via unsafe sex and without clean needles. Instead of studying the unknown, and warning against the known health hazards of recreational drugs, the medical establishment turns a blind eye to drug toxicity in its single-minded pursuit of HIV with safe sex and clean needles. Project Inform, 1992, Asher et al., 1993, Cohen, 1994a. The Clean Needle program of the AIDS establishment would appear to encourage rather than discourage intravenous drug use. Reflecting this state of mind, science recently rejected the drug AIDS hypothesis, quoting a drug researcher that heroin is a blessedly untoxic drug, Cohen, 1994a. And described nitrite inhalant AIDS links as another hatched theory, Cohen, 1994b. The failure to warn against the health risks of drug addiction is certainly one of the reasons that drug use among young people has risen substantially for the first time in more than a decade. Drug Strategies, 1995. Nitrite use continues to remain popular and has even increased recently, particularly among male homosexuals. Asher et al., 1993, Duesberg, 1993d, Mansfield and Owen, 1993, Park, 1993, Schechter et al., 1993b, Schechter et al., 1993c, Bethel, 1994, Gorman, 1994, Hodgkinson, 1994, Lauritsen, 1994, Sidonik, 1994, Valbrechtshausen, 1994, Brandley, 1995, Haverkas and Drottmann, 1995, Merkin, 1995. There is no report that nitrite bans are ever enforced or that nitrite warnings are taken seriously. Bethel, 1994, Merkin, 1995. And the number of intravenous drug AIDS patients has increased steadily for years in America. Centers for Disease Control and Prevention, 1994b, Drug Strategies, 1995, probably because drug control in America is primarily focused on supply control efforts. Drug Strategies, 1995. However, if AIDS and drug education were based on the health consequences of long-term drug use, it would be as successful as the federal anti-smoking program. Based on education that smoking causes lung cancer, emphysema, and heart disease, smoking has dropped in the U.S. from 42% of the adult population in 1965 to 25% in 1995, Associated Press, 1995b. The solution of AIDS could be as close as a very testable, very affordable, and very practicable alternative hypothesis*. End of article.